are human supraorganisms. We are not only a single organism, we are a constellation of organisms, and therefore our metabolic features are a coalescence of these human and microbial traits. So what is a microbiome? Microbiome is something beyond the gut. We all study the microbiome of the intestine and the gut. Microbiome is something beyond the gut also, although that's the most important part. We have microbiome in skin, in oral cavity, in ocular. After the COVID pandemic, we know about the nasal also. Vaginal and penile microbiome studies are also underway. Now this host microbiome interactions, that maintains the homeostasis of health. And any perturbations in these host microbiome interactions can lead to a disease, allergy, metabolic syndrome, obesity, infections. The microbiome studies are broadly divided into two different parts. One is amplicon-based, 16S rRNA gene sequencing studies for identifying all the culturable and non-culturable bacteria. If you do 18S, you get the fungus. But you don't get the gene families and the pathways, and the bacteria, how they function, you don't understand that. For that, you have to go a bit deeper. Whole genome shotgun sequencing. These are done by random primers. It's an unbiased kind of sequencing where you can identify both the, up to the species or the strain level. You can identify the fragments of the microbial genomes that are enriched in gene families and microbial pathways. Nowadays, metatranscriptomic studies have also started. They are pretty hard, as you know, isolating RNA from the human sample of the microbes and then doing the sequencing. It's a pretty hard, aim, but people are now starting to do it. The analysis is heavy when you do these whole genome shotgun sequencing. It's easy when you do 16S rRNA gene sequencing. Now, microbiome is also having a cold rush, like the GWAS studies had. You name a disease and everybody is studying microbiome in that. And some of the diseases like autism and cancer are also autoimmune kind of diseases are also in the block. Now, Nature and Science are now flooded with these reviews and new issues on microbiome recently. So, while talking of respiratory infections, viral infections now are very important to study because of these pandemics, and we have learned that there is a connection, the lung and the gut axis. There is a lung-gut axis which is very, very important. The human body, there is an impact of the host microbiome interactions that reaches far beyond the local environment in the lungs. So there are some changes that happens in the microbiome of the intestine by the food you eat or some other kinds of perturbations, the medicines you take, and that can have havoc on the lung microbiota and vice versa. How does it happen? It happens because microbes don't travel. The short-chain fatty acids, the metabolome, they actually travel and they create inflammatory responses in the host gut and lung. So schematically, there are many studies that have come up. They are reviews mostly, they are yet to prove because it's all correlation-based studies now. Functional studies are yet to come, but there are few studies that have shown that, high fiber foods produces SCFAs which acts in the lungs as signaling molecules. And also, if you have a SCFA receptor deficient animal, that has showed increased inflammatory cytokine response in the experimental models of asthma. Antibiotic treated animals and in influenza infection also, it has been shown that from the lungs, these things travel to the intestine and it creates a Th17 activation. Now, for this study, I have just, identified some of the COPD microbiome studies. These are the synopsis of some of the COPD microbiome studies that I have been able to, collate. I'm not an expert in this, but I thought that this forum will be it will be very interesting for this forum. So as you can see, there are many studies. I can name 17, 18 studies, but these first 6 studies are done in very reputed journals. NEJM, then American Journal of Respiratory and Critical Care Medicine, Journal of Allergy and Clinical Immunology. All of them, if you can see, have found similar kind of bacteria, Haemophilus, Velionella, Staphylococcus. They have got some distinct pneumotypes. And everybody all these studies have been done in different populations, although Asia or India is completely underrepresented or absent in this study. So they have found out the core microbiome present in the COPD patients and asthma patients, and these impact factors are very high, as you can see. And most of them are studied with the sputum. Only a few of them are studied with the bronchoalveolar lavage. Now there are studies coming up. It's a very recent study in Nature Communications from Australia. They have taken stool samples also. They are trying to understand the role of the intestine in lung changes in the chronic obstructive pulmonary diseases. So this is something that they are studying, not only the microbiome, they're also studying the metabolomics in the intestine. Yeah, in COVID times, there are some studies that have come up that alterations in the gut microbiota in patients with COVID. They have taken 30 COVID patients, 24 influenza patients, and 30 healthy controls. They have seen that the intra-individual variability has a change, and there are some bacteria in the intestine of the COVID patients. They are dominated by opportunistic pathogens compared to those that are present in the healthy gut. Now, I don't have much time, but we have also performed a study of the URT microbiome profile in the SARS-CoV-2. So NIBMG is actually one of the active members of the INSACOG. They study the virus from different parts of India. There are several organizations. NIBMG is leading in that. And now we are also doing, besides the sentinel surveillance, now we are also doing wastewater surveillance of COVID. And I am the nodal officer from West Bengal, from the government of India. We get all districts' raw data, their wastewater, we do these surveillance actions. So when it was lockdown, we were also studying all these things. And so from that it came out, we studied a URT microbiome profiles in Delta and Omicron infected patients, because people were studying the healthy and the COVID patients, what is the difference. But we thought to study what is the difference between healthy and COVID, and also between the Omicron and the Delta. Now, this is my lab team, the students who are working on that, and our theme is, to that, although we understand that microbiome performs essential functions by interacting with the epithelial cell types in different organs, so we want to understand broadly the contribution of ethnicity, host biological factors, and external parameters like food habits, geography, lifestyle, behaviors, how they shape the healthy reference microbiome in the Indian population. And then go by organ to organ, host cell and microbiome interactions in these different organs, how that aids in the sustenance of health. And thirdly, how the perturbations in the microbiome structure in different organs of the human body leads to chronic complex kind of ailments. And that also in differential host genomic profiles. We don't study microbiome in unison, we study it with reference to the host risk alleles already identified in GWASs or other kinds of studies. So these are the 4 types of diseases we are focusing in. We don't have any opportunity to study COPD or others, but we are constantly in connection with Dr. Bhattacharya to study a nationwide study on COPD or asthma. So we study mostly atopy, psoriasis, the skin diseases, the autoimmune. We also study foot ulcers, diabetic foot ulcers, and preterm birth and childbirth failure is one of the emerging nationwide program that we are into. Well, we have published these 3 papers from our lab in the last 5 years, and as you can see, they have got formidable citations. And we have been the first, we are the first lab in India to study the healthy skin microbiome. And now we are studying their perturbations. We have identified SRES and some of their gene families, how they kill the filaggrin protein, how they kill the filaggrin protein by their histidase and urocanase enzymes, and how that leads to atopic eczema. And in the nationwide vaginal microbiome program, we have studied that how these changes in the vaginal microbiome in India leads to preterm birth. Now, in last few minutes, I'll just browse through our COVID study. It's now under review in M Systems in an ASM journal. So we have taken COVID patients, 24 Delta and 19 Omicron, and 20 healthy controls. So we have taken from the VTM. I will not go to the details of the big data analysis. That is NovaSeq. That is the largest genomic sequencer present on Earth. We have a couple of them. And you can do 384 whole genomes every day on this in Kalyani. So we do the operational taxonomic units, because when the lots and lots of sequences are done, we can then identify them 97% or 98% sequence similarity, and then put them into OTUs. Now from each OTU, we can take a representative sequence by which we can then align to some Silva or green genes or some shotgun microbiome databases, from where we get the idea of, these are the OTU looks like. So these are the names of the OTUs. They are all de novo OTUs, and each samples are there. So total number of sequences from each sample are present there, and then you get the bacteria up to the genus or the species level by 16S or whole genome. Then you do Shannon and Chow intra-individual diversity indices. I don't have so much time to explain. We do something called rarefaction plots to see that the inter-individual variability in sequencing has not had any bias on the calling of the microbiome. So what we have found that, as you can see, the controls are closer to each other based on their genus and species level profiles. And as you can see, the cases, they have formed two different clusters. And they are like Omicron and the Delta based on their microbiome levels. And now, when we introduce that type of heterogeneity on the Omicron and the Delta, this is actually the Omicron. This one, this blue one is the Omicron, and this is the control, and this is the Delta. As you can see, Omicron and healthy controls overlap, but the Delta is completely separate. So I'll move beyond it. So I just take 1 more minute. So Delta and Omicron, we have compared by some statistical analysis. We have found that commonly reported commensal organisms are more in the controls and non-commensals in the cases, as we all know. But in the Delta individuals, the non-commensals are more, and the commensals overlap in the Omicron also. We have found a signature of 8 bacteria, both in the genus and the species levels, that have a gradient, that in control they are higher, in Omicron they are significantly lower, in Delta they are again significantly lower. So we have done a random forest-based prediction model by which we have found with 90% accuracy with this microbiome, we can annotate a control or a Omicron or a Delta successfully. So we have, hosted a India EMBO Symposium on human microbiome in 2019 in October and November, and that was in collaboration with JCVI USA. And we had brought all the microbiome stalwarts from Europe and America in Kalyani just before the pandemic, and we had a 4-day-long workshops and hands-on training and posters from different parts of India. So these are some of the glimpses of that symposium. We have now a human microbiome facility built up in NIBMG. We already had the sequencing facilities, but now we have this human microbiome facility also, where we have separate compartments for isolation of DNA, RNA, microbial DNA, RNA from the human samples like stool, body fluids, swabs, scrapes, and plaques. We have anaerobic workstations. We have these cell culture units where we can do human cell culture and microbial co-culture. And these are my students, and this is NIBMG, where we have a fantastic model of a 3-dimensional DNA helix. So I would invite all of you to the NIBMG campus. If you can get some time, you can come, and we hold regular outreach programs for schools and colleges, and we are all open to clinicians collaborating with us, because our mantra is accelerating genomics for health. Thank you.
So let us thank our first speaker one more time. We will have a panel discussion later on. So let me introduce the next speaker from this session, Dr. Sudipto Saha, who is from Bose Institute, Kolkata, and his topic is omics in pulmonary disease. So let us welcome Dr. Sudipto Saha on the stage.
Am I audible? Okay. So on the onset, I must thank Dr. Banerjee and Dr. Bhattacharya for inviting me and selecting a topic which is also a part of my lab. So the topic is omics in pulmonary diseases, and I have a lab where we work on lungs, and we use different omics approaches, including computational tools to integrate the omics approaches. So this is how healthy and diseased lungs looks like, and there are several factors associated with the disease prognosis. Some are aging, genetic factors, exposed to environmental factors, lifestyle, and the microbial infection. So we can study lungs in a different angle. You can study lung in a gene level, protein level, in a cell level, in a histopathology level, and at a patient level. So there are different ways you can dissect, and you can target in studying the lungs. So what is omics? So omics means study the inter-complement of a specific type of biomolecules. Suppose if you study DNA, then we call it genomics. And then if you study proteins or mRNA, we term as the transcriptomics. Oh, it's okay. So if we study mRNA, we term as the transcriptomics, and if we study proteins, we term as the proteomics, and metabolites, metabolomics, and lipidomics. So the aim of omics study is to identify, describing, and quantifying the particular type of molecules, and study the molecular process that contribute to form and function in a cell or a tissue. However, the single omics contribute to a better understanding of single type of molecule. It doesn't capture, so it doesn't capture the entire biological complexity of a pulmonary diseases. So for that, we need multiple omics approaches. For example, genomics, transcriptomics, proteomics, metabolomics, and also side by side we study epigenomics, which doesn't directly relate with the sequence. And we also study radiomics, which is medical imaging, and then we integrate all these omics approaches using machine learning tools and come up with some specific model which can able to predict for diagnostics purpose, or to predict the model whether it can be applied to another different population level. So here I have jotted down some of the biomarkers for across the lung diseases, mainly asthma, COPD, IPF, TB, COVID-19, and lung cancer. The first for omics, like genomics, transcriptomics, epigenomics, proteomics, and metabolomics, is coming from the host factors. But metagenomics is coming from the microbiome, specifically the lung microbiome. And this I jotted down from a book which is coming in the month of September, where me, my student, and Dr. Bhattacharya, we compiled and wrote a book in the Springer. So the detailed information you can see in this particular book, which is coming in the month of September this year. So the interesting part here is that if you look at the metabolomics part, the majority of the biomolecules, they're coming from the lipids. So the lipids like ceramide and the sphingomyelin, they has been known to be involved in the inflammation process. So in our lab, we did some analysis and we compiled all the whole genome metagenome sequencing coming from the asthma, bronchitis, COPD, and cystic fibrosis, including healthy smokers. And we come up with two plots, and Sabik just explained you about the Saran index, which is nothing but telling the diversity of the richness within a population, like how many different types of microbes are present. So if you look at the healthy smokers, which is represented in the blue box, the diversity is higher as compared to the cystic fibrosis, which is present in the green, or the bronchiectasis. Next, we thought of doing the beta analysis, where to see whether the microbiome within a disease state, whether they are different or not. So here each dot represents a metagenome sequencing of a single patient, and we can show that the COPD patient samples are totally different from the bronchitis, and it has a little overlap with the healthy smokers, which is represented in the blue color node. So to study the lung microbiome and host interactions, there are two concepts are there. So one is the target, where you can use the genomics, epigenomics, transcriptomics, proteomics, and metabolomics. On the other hand, if you want to study the microbiome in a lung, you can use metagenomics, metatranscriptomics, metabolomics, and we can integrate and see how the metabolites coming from the microbiome, they interact with the host. So the interaction can be twofold. It can be the host DNA microbial metabolite, which is present here. And it can be host metabolite protein interactions. The host metabolite, they may interact with the receptors present in the immune cells, and they may cause the inflammation process. They start the inflammation process. So this is how it looks like. The lung can have bacteria, fungi, and they produce metabolites, and the metabolites passes to the epithelial cells, and they interact with the receptors present on the immune cells, and also they interact with the ion channels and the lipids and lipoproteins, and by this way they can start or initiate the inflammation process by inducing the pro-inflammatory cytokines, and in the long run there is a destruction in the epithelial cells and causes airway inflammation. So here there are few metabolites coming from the microbe, and has been reported in different journals, and people have come up and shown that the trimethylxine, the short-chain fatty acids, the palmolite, the oxylipines, they have known to be associated with the disease pathogenesis of COPD and asthma in human as well as in the mice model. This is one paper which came up last year in Nature Microbiology, where they have shown that in case of COPD patient, mainly in the neutrophilic COPD, where the tryptophan metabolism is altered in the lung microbiome, they have used induced sputum samples, and they have shown that the indole 3-acetic acid is downregulated in case of COPD patients as compared to the healthy, which is shown here. And they conclude that this, due to the reduction level of indole 3-acetic acid, it induces the IL-22 signaling pathway, and they have shown that the proteins which interacted with the indole 3-acetic acid coming from the metabolites interacting with the host factors using the transcriptome and proteome-based approach. They use two data sets. One is the discovery data set, which is represented by ZZ, and they use a validation data set, which is represented by SZ. And they have shown that that particular species, mainly the lactobacillus species by Lactobacillus salvarius and Lactobacillus aureus, they are the main cause of producing the indole 3-acetic acid. And they use a mice model, and they shown that if they use supplement in indole 3-acetic acid in the mice COPD model, then the model recovered, and they have shown that indole 3-acetic acid is the sole responsible for the disease pathogenesis in COPD model, in animal mice model. But however, there are some pros and cons using microbiome and metabolome analysis. First thing is that if you want to use lung microbiome, you have to use deep sequencing, because the microbial mass in the lung is very low. Second thing is that most of the lung microbiome is confined to the bacteria, but however, papers are coming up that the fungi also plays a very important role, but the databases are not still established yet for the fungi and other virus disease as compared to the bacteria. Next is for metabolites. So it's very confusing whether the metabolites are coming from the host or coming from the microbiome. Still there is a confusion. And finally, in case of proteomics, still if you use multiple studies, you may get a different set of proteins, and still it's not very standardized yet. So there are some pros and cons for using multiomics approaches in the lower airway studies. So what we can do? So if you use generate on the omics approaches from the host factors, and if you use the microbiome from the lung, then we can integrate using machine learning based method. And once we develop the model, that model can help us to assess and compare the data of different domains for the diagnostics and prediction utility. We can also test the model for the patient population, and also we can use in an iterative way and apply in the clinical applications. And finally, you can test the effectiveness of the interventions of the model informed treatment and modifiable task factors. So finally, the omics approach may or may not be the total scenario. We need to integrate your data with the non-omics data, for example, spirometry or two-chair test, and then come up with a holistic view to come up with a precise or precision medicine. So this is a very important slide. So most of our understanding of the genetic risk of OPD, as Sobi was mentioning, most of the data come from the European ancestry. And as we know that the European ancestry account only for 16% of the world population, yet if you go to the NCBI gene bank, the most of the data are populated with 80% of the data are populated with the European population data. The genetic susceptibility and the environmental exposure interactions in the Indian population largely varies. And so the immune phenotypes and the disease microbiomes is also different than what we will observe in the European populations. So it needs a collaboration of different expertise to generate and analyze the omics data for the translational discovery. So this is my lab, including my family. Last year we went to Meghalaya, and I want to acknowledge some of my students who have contributed few of the slides. Thank you.
Thank you, Dr. Saha. Now, may I invite Dr. Saha as well as Dr. Mukherjee on the stage, and let us open it for discussion. So if any participant had any questions, and you can have your sheet and we can discuss and question answer session. So anybody has any question regarding, so one question.
Dr. Sumit Sengupta, panel member, Sumit Sengupta.
Okay. So also I am now introducing Dr. Sumit Sengupta. So Devkona, you can go ahead and ask the question.
We know in respiratory disease,
the sterile organ, but nowadays this lung microbes is an important thing, like that of the gut microbes, and we should know about this, and it can change the disease process, and it can change the treatment scenario also, because in asthma, COPD, in cystic fibrosis or bronchiectasis, these changes of this concept, the new concept that is the lung microbes, is a important thing, and this is on research oriented. Mainly this is the research oriented, but still we should know about this.
Thank you. Anything you would like to add from what we have given?
So question, can you repeat the question? The first question was exacerbations-based study. So till now, I haven't come across any study which specifically studying the exacerbation versus normal. But generally, in case of COPD and asthma, they group patients according to the COPD. I think there is a gold standard-based method 1, 2, 3, 4. But in case of asthma, they just differentiate into two types: exacerbation versus normal asthma. So, but in terms of that, how microbiome gets affected, I haven't come across any study as such in India.
the reason I ask this question is that there are
It can. It can. Yeah, so that's what I've showed you, that the microbes interact with the immune cells through metabolites. So if a particular microbes if the diversity is less, the Shannon alpha diversity is less, it means that particular microbes, they are dominant, the set of microbes. So if they are dominant, then they release the metabolites. That metabolites can interact with the immune cells. And many of the metabolites, they interact with the lipids, the lipoproteins, which are present in the receptors of the immune cells. And once they get bound to it, then the cascading events of like activating the IL-22 pathway and some other pathways which induce a lot of cytokines, like pro-inflammatory cytokines. Those pro-inflammatory cytokines can exaggerate and damage the epithelial cells. So that can be one factor, yeah, which is already known. Yeah.
So I remember this study, one study that I jotted down in this Excel sheet that calls biological exacerbation clusters that demonstrate, third one, asthma and COPD disease overlap with distinct mediator and microbiome profiles. In 2018, Journal of Allergy and Clinical Immunology in UK, what I have read is that they have taken 32 asthma and 73 COPD patients and tried to look into their overlap and non-overlap, as doctor was saying. So they have found cluster 1 has, so as you can see, the microbiome has a ratio of proteobacteria is to Firmicutes. Now they have found in cluster 1, proteobacteria is more than the Firmicutes, and cluster 3, Firmicutes is higher than the proteobacteria. These are two distinct clusters. And cluster 2, it has bacteroidetes, completely different than proteobacteria or Firmicutes, and that's the overlap between, so these are the two groups that are, one is COPD and one is asthma. Some of them are overlapping with each other, that is the cluster 2. Similar kind of study, they have identified pneumotypes in the 6th study, although they have not told about exacerbations, but they have also found Veillonella, Prevotella kind of pneumotype in this supraglottic taxa. So to answer your question, yes, there is one study on these exacerbations, but we have to look into it. And to answer broadly, as is the theme of these questions, that is it a cause or an effect, or is it available for diagnosis of patients or as a prognostic marker? Well, we have to understand that we are a long way to actually identify the baseline microbiome first. We have to do some studies to address the interindividual variability in healthy humans. As the panelist said, that we knew that lung is sterile. Similarly, we knew that placenta is sterile. Now we are getting placental microbiome in pregnant mothers, not in the preterm, but also who are not preterm, they also have a resident microbiome. So paradigms are shifting. So we have to understand that in Europe and US, they have already established their baseline. Now they can afford to ask those questions. We have not invested much money in microbiome. We are doing microbiome studies 15, 20, and as you can see, these Excel sheets also, sample sizes are very small, and they will have these sample size effect. We have to do a nationwide study to first identify the baseline, because ethnicity having a role, is geography having a role, because as we know, the lung-gut axis is also there. The food you eat can also impact your lung microbiome. It's a very complex thing, but you know, the 16S sequencing is not so expensive. Shotgun sequencing is. But with these kind of studies, if we can build a panel, like a gene panel we now have for genomics of cancer. Similarly, one day will come when a microbiome panel can be made for these kinds of diseases. And as a prognostic or predictive biomarker, we can identify that, but this has to be done in connection with your host genotypes, and as Dr. Saha told, host multiomics is also very important. If you already have a risk allele which is predisposing you to COPD, you better change your lifestyle and make your microbiome healthy so that your risk factor is lessened. All these can be done when we develop a score, something like we now do in the genomics thing, polygenic risk score. It will be a multiomics risk score kind of thing. So AI, statistics, big data, everything has to be integrated. But first we have to identify the baseline. Is the baseline same? I am doing a study, he is doing a study, everybody is doing a study. Are we finding a unified core microbiome? If yes, then we can study the perturbations on that to identify cause-effect relationship in animal model studies or in vitro cell biology co-culture studies.
Yeah, so the first report of that lung is not sterile, it came in 2010. And the paper came in PLOS One journal, so which is not considered to be a very good journal. But believe me, the same group, the next paper came in Nature. So since it was the first paper in 2010 that the asthma and COPD contains some microbes, so it was published in a PLOS journal, that was PLOS One only. And second thing is that, as Sobik was telling, that in China, the paper which was shown in Nature Microbiology, it came from China. So what Chinese people are doing, or scientists are doing, they're integrating with the hospitals. And they're getting data from the hospitals and the clinicians and the basic scientists. They are integrating, and in that particular paper, more than 30 people are there as authors. So they are integrating the data as it was done in the European-based studies. So China, if China can do it, why not India? The question comes here.
Yeah, please.
I'd just like to react to what my colleague talked about, the importance of the lung microbiome in stable COPD and exacerbations. So by now we know that lung microbiome is basically very important in COPD. And there is a normal equilibration of the pathobiota and the symbiota that we normally see in the lung microbiome. That is in an equilibrated status. And once this equilibrated status changes, it is a very dynamic process. And the dynamicity actually has been related to the possibility of the microbiome change leading to an exacerbation. Early days has been seen with, particularly with Haemophilus influenzae, but this is the way that people and the researchers like you think about it, and you're going to give us further answers. The answer to the question is yes. There is a dynamic change in the microbiome between the stable COPD and the exacerbation.
Thank you.
May I make a small comment? Yeah, because this is a clinical question. It's very important to understand the microbiota is changed when there is exacerbation or if there is an infection. Almost all microbes can synthesize one or other antibiotics. There's no antibiotic under the sun which is not synthesized by the microbes. And microbes control microbes. The good microbes, to my mind, controls the bad microbes. What happens, we do more harm by killing the good microbes most of the time. And that's the reason we have a cycle of infection coming up one after another. Exacerbation leads to another exacerbations. There has been a study I cannot quote right now. It has shown that just changing the gut microbiota reduces the prevalence of ventilator-associated pneumonia. So it's integrated to gut microbiota. The whole issue is wonderfully placed by my friend Dr. Parvez, that the balance is disturbed, and the gut-lung microbiota relationship is also disturbed in diseases. But we have to identify exactly what is the specific change for a specific disease and what is a quantum change for the degree of disturbance that leads to a disease.
I have one question. I'm Dr. Sushmita Roy Choudhury. So see, we have some publication also where we've done BAL and sent and looked at fingerprinting for asthma, COPD, and the so-called ACOS. And there is difference in them. But we definitely need microbiota in stable disease, in asthma and COPD patients. Do you think you are all going to be able to help us in collating data if we were able to give you samples of stable COPD asthma patients in Eastern India? Similarly, it could be done in Western India. We have data from the western part of the world, but we don't have data from our country at all about what the microbiota here is, because that would be so much more helpful when these patients who are frequent exacerbators come to see whether microbiota has a role to play in it or not.
The answer is yes, because in NIBMG we can do sequencing, and in both institutes like us, we have a state-of-the-art mass spec facility. which can allows you to do metabolome studies and proteomic studies. So what we need is to integrate and talk among each other.
Yes. So do we
Cross-talk
take
among each other.
do we take it that you will initiate it, or
Yeah,
will we need
we can. We can.
Okay, great. Thank you.
One more thing I want to say that people have known already that the microbes alters, but now the big question is that how microbes interact with the immune cells. So if there is a differential expression in the microbe, that is already reported, and for the last 10 years people have reported. The big question is that, as I was mentioned in one of the paper in 2020-22, it came in the Nature Microbiology, that which product coming from the microbes and how they influence the immune cells or particular pathway, like signaling pathway, which signaling pathway. So that is a big question nowadays.
Any more questions?
Another thing is that taking sample is a problem in to know about the lung microbiota, because there are many times the contamination occurs because, by doing that, and when we are sampling the lung microbiota, that is the lung samples, this contamination occurs frequently from the nasal areas, from the oral areas. This is the problem. The sampling is important thing. We have to take the samples correctly.
I think most of the studies here have looked at sputum, which is obviously contaminated, but that is the baseline we are doing about. One thing we are not touching on, I don't think we have enough information, is difference between causation and correlation. I mean, you're talking about exacerbations being correlated with changes in microbiota. Are they causing it? That is the question which we don't know. And I think it's going to be very difficult, but as you said, the first step is to get a level playing field. This is the normal microbiota in Kolani, West Bengal, India, and so on and so forth. We don't have that data, and we really cannot extrapolate from Western data. It would, you know, it's a huge difference in interaction. So I think we look forward to interacting with you all, and as Shushmita says, we'll be on your backs very soon with various proposals. If there are no further questions, we can call this session to a close. Thank you very much to our distinguished speakers. Thank you.
Thank you so much.
Thank you, sir. Thank you, everyone. Before we proceed to the next session, I would like to say that tea is being served outside. You're all invited to go and have it. And with this, we proceed to our next session, that is physiology and assessment. I would like to invite the coordinators for this session, Dr. Dhiman Ganguly, Dr. Ganguly is a devoted to academic activities and an eminent orator across the country and abroad. Currently, he is associated with the Vivekananda Institute of Medical Science, Kolkata. He has also adored the chair of the East India chapter of ACCP and is also the president of Indian Chess Society. Our next eminent speaker, chairperson, is Dr. A.G. Ghoshal. Dr. Ghoshal is the ex-president of Indian Chess Society and is a consultant pulmonologist for more than 30 years of clinical and teaching experience. His areas of special interest are airway diseases, lung-resistant TB, sleep disorders. Currently, he is associated as the medical director of NAABI, Kolkata. I welcome you, sir. I welcome you, Dr. Ghoshal and Dr. Ganguly, and request you to kindly proceed over the session. Thank you.
Should we begin right now?
We can start.
We can begin right now.
We can start. Thank you.
There is some overlap. Better to start. Okay. Good morning and welcome to this summer symposium on a fine morning. Now let me begin by congratulating the scientific committee of this summer symposium for using the term airways diseases rather than our usual fascination with asthma and COPD and the overlap and so on and so forth. So that's an improvement. It's a confession in a sense that the nosological problems with airways diseases have not been solved yet. We would rather talk about non-specifically about airways diseases rather than giving them a diagnostic level, which is a great improvement. I don't think this has been done in the past 30, 40 years. We usually restrict ourselves to the terms asthma, COPD and overlap. Secondly, assessing lung is a rather different problem compared to assessing, say, liver or kidney. Now these organs of ours, they mainly are concerned with maintaining the milieu interior, maintaining our internal environment in the right place. And therefore, when it comes to assessing liver or kidney, we depend on metabolic parameters, metabolic measurements to find out organ damage. But lungs are quite different. Firstly, the act of breathing is mechanical. You know, this is partly true about heart also, but because this is a mechanical function, so measuring lung mechanics is a part of assessment of lung, which is not seen in some of the other organs I was mentioning. Secondly, if we come to metabolic parameters, after all, the heart and lung physiology are supposed to maintain our oxygen supply and hanging on to the right dose of carbon dioxide. Lungs are supposed to saturate our hemoglobin to 100%, and they are supposed to keep hanging on to the right volume of carbon dioxide in the system so that our acid-base balance remains intact. Now, if we consider that, then measuring arterial blood gas only should give us a good idea about cardiopulmonary physiology. But that's not true either. Apart from the mechanical part, apart from measuring lung mechanics, there are other problems with lung, mainly the airways and the lung parenchyma. They produce different kinds of problems with lung mechanics, and they are not mutually exclusive. That means there are patients who have both airways disease and lung parenchymal disease and contributing to their quality of life differently. Quality of life and our fitness level in day-to-day life also depends partly on cardiac function, but what is special about these two organs is, you see, they have to match our lifestyle all through 24 hours. We are sleeping, we are talking, we are sometimes running around, and the cardiopulmonary physiology will have to match our activity level all the time. So measuring lung function means looking at the metabolic effect of lung function, looking at lung mechanics, looking at different kinds of mechanical trouble depending on whether the disease is predominantly airways or predominantly lung parenchymal, and last but not the least, it has to match our lifestyle. That means our fitness level. So all these things make assessing lung much more difficult and complicated compared to organs like liver or kidney. So what we'll do in next 2 hours or so is to look at our present armamentarium of assessing lung physiology, and then we will have a question-answer session where you will have a chance of addressing specific problems in specific situations that we face in our day-to-day practice. So with that introduction, I will ask Dr. Ghoshal to begin the session, asking for the speakers. I believe there are 3 speakers, and they will be covering different aspects of lung function, and we'll then have an hour to answer questions from the audience and some from our coordinators. Thank you.
Thank you, Dimanda. So, good morning, everybody. This session is focused on physiology and assessment of airway diseases. We have 3 speakers, 3 very good speakers. First, functional anatomy and physiology of airway diseases, Dr. T. Balamuges. Then assessment of airway diseases, Dr. Sojol De. And the third, HRCT in airway diseases by Dr. Sumit Sharma. So these 3 speakers will speak in tandem, and then we have our 5 panelists, Debraj Josh, Sivarajmi Unitan, Pranav Mondal, Sudeep Ghosh, and Saurabh Maji. I think they are all already here. So we'll take the lessons, informations from the speakers, and for the next half an hour we'll go to the panelists. What is their take of these messages from the speakers? And though it's a session for all, I particularly intend on the next generation to focus on the theme of the topic. So first I invite Dr. T. Balamuges, Functional Anatomy and Physiology of Airway Diseases. Dr. Balamuges, please. I think the CV is there. Yes. Okay. We'll go to the...
Thank you, sir.
Have a good time.
Good morning, friends, respected sirs who are the chairpersons. I'm grateful and privileged to be part of this summer symposium. I express my gratitude to the organizing committee and Dr. Partha for inviting me to this wonderful city. My talk is anatomy and physiology, often the boring topic during our MBBS days. We just mug up something so that we can pass the exams and go to the more interesting stuff. But the knowledge of anatomy and physiology is important and relevant with the onset of newer methods of assessing the both the diagnosis of the disease and managing. So I will be trying to give importance to the clinical relevance of the anatomy and physiology. I bring greetings to you from Christian Medical College, Vellore. A few months back, some of the specialty departments moved to a new campus, which is much more spacious and well equipped. I invite all of you to visit Vellore. What happens during normal breathing? As Sir told, breathing is a mechanical function, and when we breathe, in a day, 10,000 liters of air go through the respiratory tract, which exchanges around 400 liters of oxygen and carbon dioxide across the alveolo-capillary membrane. And when the air needs to flow, it can flow across pressure gradients. So it has to flow from a high pressure to a low pressure gradients. And this gradient is provided by the work of breathing, the respiratory muscles. And countering this work of breathing is the resistance to the flow in each segment of the respiratory tract, as well as the compliance of the lung, as well as the chest wall. So there is a close interaction between all these things which results in the normal breathing. We all know the Poisson's law. The resistance to a laminar flow of air through a rigid tube is related to the length and diameter. Greater the length, the greater the resistance. Smaller the diameter, there is a 4 times increase in the resistance. So the airway diameter has a more significant role than the airway length. This Poisson's law cannot be directly applied to all segments of the respiratory tract because respiratory tract is not a rigid tube. It has a dynamic lumen which changes during inspiration and expiration, as well as the flow is not always laminar. It is often turbulent at places. And this diagram is very important for us to understand the pulmonary function test, which will be dealt in the next talk. And some amount of air, even after forceful expiration, remains in the lung. That is called the residual volume. And this volume of air is important because this prevents the alveoli from collapsing during expiration, also maintains diffusion of gases even during expiration. So some amount of air is normally trapped in the lung even after forceful expiration. What is complaints of the lung? Complaints of the lung, in simple words, is the ability of the lung to distend. And that varies from various lung zones, or from the upper lobe to the lower lobe, from the apex of the lung to the lower bases of the lung. The ability to distend is dependent on 2 factors. One is the intrapleural pressure, which is more negative in the upper part of the lung. It is -8, and it is less negative in the bases of the lung, -1.5 centimeter of water. And this is because of the gravity. This means that the alveoli at the upper part of the lung is already distended than the alveoli at the lower part of the lung. So for each given amount of the distending pressure, the alveoli at the apex of the lung, because they are already distended, they can distend only less. So the compliance is less at the upper part as compared to the lower part. So preferentially during normal breathing, the air goes to the lower zones because the alveoli are more able to distend. So this is explained in this graph. As you go from the apex to the base, the ventilation increases because the alveoli are able to distend. As compared to the blood flow, because of the blood flow is more affected by the gravity, there is disproportionate increase in the blood flow to the bases of the lung. So both ventilation and blood flow increase from apex to base, but there is more increase in the disproportionate more increase in the blood flow, and that is reflected by the VQ, ventilation perfusion ratio. Coming to the respiratory tract, and this is a confusing field because lower respiratory tract, conducting zones, small airways, are they all synonymous? No. There is various terminologies used by various authors or different organizations, and that confuses people what the divisions of the respiratory tract. I will go through each one of them. The first division is anatomical divisions, which is upper respiratory tract and lower respiratory tract. And this is divided by the larynx. Anything above larynx, including larynx, is upper respiratory tract, and it is affected by the barometric pressure, the atmospheric pressure impinging on the external surface. Lower respiratory tract include extrathoracic and intrathoracic portions of the trachea and the rest of the bronchial tree. And this is not directly impacted by the barometric or atmospheric pressure. This is impacted by the intrapleural pressure, especially the intrathoracic airways, and which is normally negative but become positive during forced expiration. The intrapulmonary airways are surrounded by alveoli, and the diameter of these airways are affected by the alveolar tension as well as then the alveolar septa, which tethers the airways. So there are multiple factors which affect the intrapulmonary airways. And this is an important diagram which you all know. The changes in the flow volume loop in different types of obstruction. So in extrathoracic obstruction, extrathoracic variable obstruction, they are outside the thorax. They are affected by the atmospheric pressure or the barometric pressure. During inspiration, the barometric pressure is more than the inspiratory pressure, the pressure inside the airways, and then the airway collapses, the upper airway collapses, and that is shown by flattening of the inspiratory loop in the flow volume loop. And in variable intrathoracic obstruction, it is the intrapleural pressure which is affecting the pressure, affecting the airway diameter. And during forceful expiration, as shown here, because the intrapleural pressure is more than the pressure inside the airways, the airway narrows and causes flattening of the expiratory limb. Okay? And then in fixed obstruction, it is not affected by all these things or the pressure gradients. It is both, it's like a box-like graph or a square graph where both flattening of the inspiratory and the expiratory limb. The upper respiratory tract plays an important role. The nasal cavity has important function in filtering, warming, and humidifying ambient air. The nose accounts for approximately 50% of the total resistance to airflow through respiratory system during nasal breathing. So that has important clinical implication because that's the reason why patients who are dyspneic, they don't breathe through nose, they open their mouth and breathe. What they are basically doing is to reduce the work of breathing by negating the effect of nasal airflow resistance. Another implication is if you are giving nebulizer through a face mask and you are asking them to breathe through nose, there is increased resistance and also there is reduced deposition of the aerosols to the bronchial tree if they ask the patient to breathe through nose. So if possible, you allow them to ask them to breathe to mouthpiece rather than the face mask. Or even if they you give a face mask through a nebulizer, ask them to breathe through mouth rather than through the nose to improve the deposition in the lower airways. Another important practical thing is, one of the mechanisms by which tracheostomy help is rather than this thing is tracheostomy bypasses the resistance to the airflow through the nasal passage. The long, narrow oropharynx surrounded by adipose tissue and also the negative pressure during inspiration can cause the tendency for the oropharynx to collapse. And this is negated by neural reflexes which stiffen the pharyngeal muscles. And here also there is implication because when there is obesity, there is excess adipose tissue deposition in that fixed space and there is reduction in the airway size in the oropharynx and this predisposes to sleep apnea. Because at some point of time, the stiffening of the pharyngeal wall to prevent the airway from closure is not sufficient during, especially during sleep, and then the airway significantly narrows, results in snoring, and then subsequently sleep apnea. Now coming to the next terminology. So we talked about upper respiratory tract and lower respiratory tract. The second group of terminology is conducting and gas exchange zones. Here it is easy to understand. Conducting airways, their function is only conduction of the air up and down. They don't take part in gas exchange because they lack alveoli. The gas exchange zone, otherwise called transitional or respiratory zone, has alveoli in their walls, and they consist of respiratory bronchioles, alveolar ducts, alveoli. And if you number the divisions, generations, up to 16 divisions is conducting zones, that is up to terminal bronchioles, and beyond that up to 23 it is the gas exchange zone or the respiratory zone. Now coming to the third division of airways is large and small airways. And this definition of small airway was done in a workshop which happened in France in 2011. And in that workshop, they defined small airways, or otherwise called peripheral airways, as non-cartilaginous airways with an internal diameter of 2 mm. So small airways do not have cartilage. So that means that they are bronchioles and they have an internal diameter of less than 2 mm. So from 8th generation of airways down to alveolar sac, it is called small airways. The rest of them up to 7th generation, it is called large airways, or called more than 2 millimeters in internal diameter. So then there is confusion. So what is large and small airways, and how is it related to conducting zone and gas exchange zone? So this diagram explains that. So there is some overlap between conducting zone and small airways, because conducting zones includes terminal bronchioles, and the bronchioles are considered small airways less than 2 mm, and there is some overlap here. The respiratory or gas exchange zone starts only from 17 and below, while the small airways starts from 8. As the airways divide, their internal diameter reduces. So as the number of generation, 1 bronchi becomes 2, then they divide again 4, 8 and 6, and their individual diameter decreases. But up to 4 divisions, their total cross-sectional area remains the same. So although they divide and they become smaller, the sum of all the cross-sectional area almost remains same till 4th division. After that, you can see that there is gradually and then subsequently exponential increase in the total cross-sectional area. So like in the respiratory bronchiole, although the individual bronchioles are very small, because there are so many divisions, so many respiratory bronchioles, their total cross-sectional area is significantly high. And that is well depicted here. Till around 4 divisions, the total cross-sectional size remains same, and then it subsequently increases as the divisions increase. And this is again depicted in this diagram. And at the bronchioles, alveolar duct level, there is significantly high cross-sectional area. The area is so high, but the volume of air going through its limit is same. That means that the velocity is significantly reduced in these distal airways. Because the velocity is decreased, it is called silent zone. And these area is responsible for 20% of the respiratory resistance, total airway resistance. Because the area is so huge, they contribute only 20% to the total airway resistance as compared to the central airways, which contribute 80%. These small silent zone or the small airway airways are, although it is called silent zones, they are very important in health as well as in disease. Because the flow of air is very slow, there is more chance that the pollutants or the irritants which reach that site, they can deposit in the airway walls and cause damage. And also, it starts any obstruction or pathology, because the total area is so large, they remain silent for a considerable duration of time till it is very severe. It cannot be easily detected. Only when 75% of the small airways are occluded, it will manifest as abnormal spirometry or FEV1 value. So these small airway small airways, which are otherwise called silent zone, are very important in the pathogenesis of some of the diseases. The airway diameter has also implication in the deposition of inhaled particles. As we all know, the bigger the particles, they don't enter the smaller airways. The coarse particles, otherwise more than 5 to 10 microliters of diameter, the term used in literature is mass median aerodynamic diameter. So that means that 50% of them will be above that particular value and 50% below that particular value. The coarse particles gets filtered in the upper respiratory tract, nose, throat, as well as the pharynx. The fine particles, which are commonly used in the traditional or conventional inhalers, are between 2 to 5 microns, and they get deposited in the conducting airways, which predominantly has receptors for bronchodilation. The extra fine particles, less than 1 to 2 micrometer in diameter, can penetrate to the small airways. The ultra fine, less than 0.1 micrometer, can reach the distal alveoli, and they are so small that they can even cross the alveolar capillary membrane and enter the systemic circulation. So this graph depicts what I just told. The more than 10 micrometer diameter, it gets filtered in the oropharynx. 2 to 5 is what reaches the bronchial and conducting airways. Less than 2, it can reach the alveoli. So the lesser the particle size, the more distally it can go. That's the implication why we use for one of the parameters of air pollution, PM, particulate matter, 2.5 micrometer, whatever it is, less than 2.5 micrometer, they have significant health effects, adverse effects due to the pollution. Coming to treatment implications, the extra fine particles, as defined by less than 2 micrometers, if we use as an inhaler, will they have beneficial effects as compared to the usual fine particles in the traditional inhaler? The data is conflicting, various studies showing various things. What to do if there are conflicting results? You put them everything together and call it as a systematic review or meta-analysis. So this looked at that, and they found out that extra fine inhaled corticosteroids is better and have achieved better asthma control, better exacerbation ratio, and lesser ICS dose. But when they looked at ICS-LABA combination, beclomethasone, formoterol, extra fine particle versus non-extra fine particles, again this is a meta-analysis, they found out that they did not show any significant benefit in small airway dysfunction parameters like FEF 25 to 75%, no difference in FeNO, asthma control test score, rescue medication use, or exacerbation rate. So there is no consistent effect to say that extra fine is definitely superior. You have to consider your patient in front of you and see how he responds to the traditional inhaler, and if it is not responding adequately, you may need to assess his small airways and subsequently target the small airways. So what happens in COPD? The smoke and the noxious particles reaches the distal airways and the alveoli. The alveolar macrophage is stimulated, neutrophil is activated, and the cytokines cause alveolar disruption, mucus hypersecretion, and mucosal inflammation. So there is disruption of the alveolar walls, excess mucus secretion, inflammation, there is submucosal fibrosis, and this results in what is called hyperinflation, dynamic hyperinflation. The patient is unable to expire his inhaled gas fully, and that results in increase in the functional residual capacity. So his baseline functional residual capacity itself is increased, and when he exercises, this respiratory rate increases, there is inadequate time for expiration, and then the air trapping worsens. There is continued dynamic hyperinflation, and his inspiratory capacity is significantly reduced. So the lungs get progressively inflated. His exhausting his breathing reserve, his inspiratory capacity is reduced, and he feels very breathless. He is unable to take his breath. Another effect of hyperinflation is, suppose in this, the left upper lobe is hyperinflated because of emphysema. You can look at it, the hyperinflated left upper lobe is compressing and compressing the left lower lobe, which is otherwise healthy. So the lower lobe, which is healthy, is unable to function to its full capacity, as well as the diaphragm is getting flattened. So the length tension relationship of the diaphragm is altered. So for the diaphragm to function well, there has to be an optimal length and the tension, which is altered when it is flattened. Diaphragm is unable to function to its optimal capacity, and then patient feels breathless. And another mechanism why the patient is breathless is because of the neuromechanical dissociation, otherwise the afferent-efferent mismatch. Usually we are not breathless sitting in this hall because there is good match between motor output and mechanical response. For a given amount of stimulus, there is a given amount of volume of air which goes in, given amount of respiratory muscles shorten. But that is altered in COPD and asthma and airway diseases. There is dysynchrony between efferent output and the afferent impulses, and that is perceived as dyspnea by the patients. The last concept is the equal pressure point. When a patient, normally when we breathe out, there is positive pressure inside the alveoli as well as the bronchi. The positive bronchial pressure gradually decreases, and then it becomes equal to the atmospheric pressure when it comes out. And when it gradually decreases, at some point of time, the pressure inside the bronchi will become equal to the pleural pressure, and then when it reaches there, the airway tends to collapse. And in normal individuals, it happens in the airways which has cartilage. So the pleural pressure becomes equal or more than the intrabronchial pressure in airways which have cartilage in normal individuals. So the airway doesn't collapse. But in COPD, because of the dynamic obstruction, narrowing of the airways, there is increase in the velocity of air, and there is precipitous fall in the intrabronchial pressure, so that the equal pressure point at which the intrapleural pressure becomes equal to the intrabronchial pressure is at the distal airways which do not have cartilage in their walls. And because of that, there is collapse of the airways, aggravating the air trapping. One of the methods by which pursed lip breathing helps is to prevent this happening by moving back the equal pressure point back to the bigger airways which have cartilage. Though when you constrict the mouth like a purse and then ask them to expire, and there is increase in the pressure of the airways and prevent the airways from collapsing. And many patients, without even us teaching them, they get to learn this and they utilize this to help their breathing. So in conclusion, the basic knowledge of anatomy and physiology is important to understand the mechanism of the disease, and it helps in the diagnosis and appropriate implementation of management strategies in airway diseases. Thank you.
Dynamic hyperinflation, and lastly equalization of pressure point. These are all really very intriguing and interesting, but he has covered it very well. No questions at this stage because we would have a panel discussion at the end which may answer most of your thought process. And I also request our speakers to please, if possible, stick to the allotted time of 20 minutes. And naturally the continuum is next one, assessment of airway diseases by Dr. Sajulde. Dr. Sajulde, please.
Good afternoon. Thank you, Chairperson, for the nice introduction. It is my pleasure to be here. I'd like to thank Dr. Parthasarathi Bhattacharya for inviting me. For next 20 minutes, I will talk on the assessment of airways. My previous speaker already described the physiology, and some of the slides may be similar, so I would like to skip those slides. Already has described there is the airways, maybe small airways and the large airways. And in spirometry, we are usually look for the FEV1 values. But if the diseases are heterogeneous, it primarily involves the small airways, the spirometry may not able to pick up the airflow obstruction. As it happens in pre-COPD patients, where the patients are very symptomatic, but spirometry is absolutely normal. So they do not classify as a COPD as per GOLD guidelines. So in this group of patients, the other lung function parameters may be useful to diagnose the obstructions. What are the techniques available to diagnose airway diseases? Most commonly you use the spirometry, where we look for the FEV1 FVC ratios, maximum need expected flow rates, FEV3 divided by BC. In the body plethysmography, we can look for the specific RAW and the GAW, and we can use also IS or forced oscillation techniques. What are the less commonly used techniques are available? That is the tidal breath analysis. It usually done in case infants or neonates. We can go for area under expiratory loop measurements, capnovolumetry, which is also part of spirometry you can do during the spirometry, methacholine or bronchoprovocation test, and estimation of nitric oxide at different flow rates. Not only the airflows, airways, also you can measure nitric oxide at the alveolar level. We know that we have to use the FEV1 by FVC ratios to diagnose the airflow obstruction. But there are a lot of controversy. How we'll use the ratios? Most commonly use the FEV1 FVC less than 70% as advised by the GOLD guidelines. But GOLD guideline has used that it should be used the post bronchodilators. Whereas ATS, ERS and most of the guidelines has mentioned that you should use the less than lower limit of normal. And there is a new approach which is the parameter D, I will discuss later on. What are the challenges to use fixed FEV1 FVC ratios? It is the most simplest and most commonly used in our day-to-day practice because it is basically independence of the predicted equation, because in Indian we do not have any uniform predicted equation. So it is very easy for us to use the fixed values. But what the problems? The problems are the ratio is arbitrarily selected. And with ages, the FEV1 FVC ratio declines, and it also varies between the male and female. So if you use the fixed values of FEV1 FVC, you may misclassify approximately 20% of the patients. And misclassification may occur mostly in case of younger adults and in the elderly. So can we use the lower limit of normal? But what are the problems? Because if you need the lower limit of normal, you have to need the predicted equations, and we don't have any predicted equation. And adjustment of the ethnicity and race is required for the reference equations. And we have observed that self-reported ethnicity is not sufficient. Suppose that Indian born in US and brought up in US, the same Indian values cannot be used for him. And also the lower limit of normal is not very good parameters to detect the mild airway disease. And if you need to use properly, you should have baseline values, but it is not possible in practical issues that every person should have a normal baseline values. Because even there is a decline from 95 to 10 percentile, it may be normal, but the patient has actually have the disease. So very difficult to diagnose airflow obstruction by available parameters. Next is the parameter D. It is basically the volume of rise of during expirations. There is increase in volume over time. So these can be measured, but it is not usually available in our spirometers at present. In healthy individuals, these parameters basically there is where flow will be increased, volume rise will be high, so it will be negative or little positive. And it has shown that D parameters has a significant association with the FEV1 values. So these can be a possible parameters in future. Next, the severity of classification. Again, there is a lot of changes between the severity of classification. What ATS-ERS guidelines has mentioned in 2022-21, and the gold guideline, the severity is again different. Again, there is a difference in the Indian guidelines. And sometimes what happens, people used to use the FEV1 FVC values to use the severity of obstruction, but you should not do it. Okay? You should avoid use. You should always use the FEV1 values to classify the severity of obstruction, but there is a wide variability across the different recommendations. And the latest guidelines, 2022, they have mentioned the Z score. You should use the Z score. Z score, how will measure the Z score? You have to measure values minus the reference values and divided by the standard deviations. So that means the patient's value is how much difference from the normal values in terms of standard deviations. So 1.64 is up to 1.64 standard Z score is normal. If it is more than minus 1.64, that is abnormalities. And this classification has been made mild, moderate, and severe depending upon the Z score. But here again the point is you need the predicted equations. Otherwise, you cannot use the Z score. Whenever we compare the spirometry reports, we have to compare with the normal predicted equations. If you are using the predicted equations of other populations, there will be considerable changes. You may misclassify. Dr. Chabra et al. Has shown that if you use the Indian values, Indian patients' values and compare with the European values, there is considerable increase in the changes. There will be more, you will more misclassify obstruction and also restriction. So you have to use patient-specific race and gender-specific equation to classify the severity of obstructions. So because the spirometry reports depends upon the race, so that has been addressed for last 1 year. There is a race-neutral approach is proposed. In GLI equation, what we are using in mostly spirometer that do not have any Indian values. Okay, but these values, these approach has mentioned that you can use, there is some race-specific weight can be applied to get the predicted equation. And this time, what we have observed, we are using the standing height. But it is observed that the sitting height is the most predictive power to develop the predicted equation, not the standing height, what we are using till today. Next is the bronchodilator response. Bronchodilator response is a qualitative term. Reversibility means that is the normalization of the FEV1-FVC ratio after giving the bronchodilators. And ATS 2022 guideline has mentioned that bronchodilator response must not be confused with the airway reversibility. And bronchodilator response means integrated physiological response of airways, nerves, epithelium, mediator, and smooth muscle cells, along with structural and geometrical factors affecting the airways in the conducting airways. And the clinical implications and significance of bronchodilator response is still not known. Traditionally, the absence of bronchodilator response means airflow obstruction is irreversible. But bronchodilator response is not a specific parameter to differentiate between bronchial asthma and COPD. Large cohort has shown that bronchodilator response between the bronchial asthma and COPD is not much different, around 18%. So the bronchodilator response has limited value distinguishing between the asthma and COPD. It should be considered as a phenotypic marker. And also it has observed that bronchodilator response can change after giving the bronchodilators. In some patients may have changed, absolutely become normal. Some may be partially reversible, and some may remain irreversible. And the difference in bronchial asthma and COPD, it is observed the bronchodilator response in bronchial asthma is more in FEV1, whereas in COPD it is more in FVC. So that is a criteria to differentiate with bronchial asthma and COPD, but it is not advocated at current scenario. What is the ATS guideline says about the bronchodilator reversibility that we are using in day-to-day practice? That is the post-bronchodilator response, post-bronchodilator values, minus pre-bronchodilator divided by the pre-bronchodilator multiplied by 100. But there are some limitations. It is influenced by the baseline lung function. If it is very high or very low, bronchodilator response may be altered. And it is associated with height, age, and sex in both healthy and disease. What is the recent guidelines mentioned? They have said that bronchodilator, post-bronchodilator minus pre-bronchodilator divided by predicted values multiplied 100. And predicted values as per GLI equations. And here the change is more than 10% of the predicted is considered as a bronchodilator response. So there is a significant changes in the recent guidelines about the bronchodilator response calculations. Suppose this patient. So this patient has FEV1-FVC, the airflow obstructions. The FEV1 shows there is a more than 12% increase in FEV1. But if you see the absolute values, there is no increase, not more than 200. So though the patient has airflow obstruction, this airflow obstruction is not reversible. These patients, again there is an airflow obstruction, and severe airflow obstruction, and FVC there is a bronchodilator response not only in the 12%, there is also increase in 200%. But if we use the new criteria, that is 2.29, that is the post-bronchodilator, minus 2.04, divided by predicted equation, that is 2.08 into 100. That means 8.4%. So as per latest guidelines, the patient do not have any bronchodilator response. Next, one patient who is 83-year-old smoker having exertional dyspnea, and this patient has the spirometry reports. So here you can get there is a moderate obstruction, FEV1 is 48%. Okay. And what happens after giving the bronchodilators? There is a decrease in FEV1 and FVC. So there is a paradoxical bronchodilator response. So it is not very uncommon. So not only you will get the positive response, you can get also the negative response. That is the paradoxical bronchodilator response. And around 4.5% you can get in the clinical practice. There are several mechanisms has been advocated, that is adverse reaction to the propellant or the beta receptor polymorphisms. This patient, This patient has FeV1 FVC 68%. As per gold guidelines, you can say they are not 73% after post-bronchodilator, they do not classify as a obstruction. But pre-bronchodilator is 68%, and the values are near normal. And what is this? So this is basically, if we use the gold guidelines, you may misclassify this patient as an obstruction. Because you have to look care of this, this patient is 75 years old. So the lower limit of normal in 75 years old may be less than 70. Okay. So fixed criteria may misclassify these patients. This is one patient who is 11 years old male. Here you can get that FeV1 FVC ratio is 60%, and the FVC FeV1 is 100%, 145%. So you can say definitively that this patient has airflow obstruction. There is no doubt. But does it have airflow obstruction? No. This is a dysnapsis. What is dysnapsis? Defined as abnormal FeV1 FVC despite having normal FeV1 and FVC. Due to unequal growth of the airway caliber and the lung parenchyma, that is dysnaptic growth. And these patients are high risk for obstructive lung disease. These risk factors are male, younger age, and taller statures, and associated increased morbidity. Another patient. This patient has airflow obstruction. You can see FeV1 FVC ratio is normal, so this does not qualify for the airflow obstruction, but FeV1 is less, 69%. So what is the diagnosis? Any comments? So the patient may have restriction or may have non-specific patterns. So you have to understand what is the non-specific pattern. Non-specific pattern is defined as a normal FeV1 FVC ratio and a low FeV1 and a low FVC, or both with a normal total lung capacity. So this patient, you may suspect the patient may have restriction or non-specific pattern, but you have to go for a total lung capacity estimation, then only you can say it is a restriction or non-specific patterns. Non-specific pattern is basically the patient failure to exhale or inhale completely, and you can go for a measurement for the slow vital capacity. If there is a slow vital capacity more than 100 ml from the FVC, it implies the airway collapse during the post-exhalation procedures. And the prevalence of non-specific pattern is approximately 6.6% in general populations, and one study was conducted over a period of 3 years. This non-specific pattern may remain same in non-specific in 64% of the patients, but rest of the patients, they may convert into restrictive pattern, obstructive pattern, normal, and even mixed. Another patient, that is a 46-year-old male smoker. This patient do not qualify for the obstruction as well as gold guidelines, but this patient also has FeV1 is less than normal or less than 80%. So what is this? So that is a preserved ratio impaired spirometry. The term is introduced by Wallen in 2014, which is described as post-bronchodilator FeV1 FVC ratio is normal more than 0.7 and FeV1 less than 80% of the predicted or less than lower limit of normal. Prism is associated with thicker airway wall relative to the smoker with normal spirometry. Prism associated significant increased risk for mortality, adverse cardiovascular, and respiratory outcome. And Prism is unstable group with frequent transition to both obstruction and normal spirometry over period of time. What are the other parameters we can use other than FeV1 FVC to classify for the airflow obstructions? One is the PA measurements, peak expected flow rate, but they cannot be measured, you should not be used reliably because they have weak specificity, depends upon the patient's cooperations and effort. Maximum mid expected flow rate is highly variable, boldly reproducible, not specific for small air diseases, and it depends upon the FVC. Normally, if the mid expected flow rate is less than 60%, that means the patient probably have small air dysfunctions. The other parameters that can be used to assess the small eye dysfunction in the spirometry, that is the FEV3, over a period of 3 seconds divided by FVC, and FEV3 divided by FEV6. And next is the area under the expeditive flow volume curve. This can be used to classify obstruction and the severity of obstructions. Next, one interesting thing is the capnovolumetry. These parameters can be measured during, if you have ultrasonic spirometer, these are additional features you can measure capnovolumetry. What is the capnovolumetry? That is basically the spirometer machines will measure the exhaled EtCO2 during tidal breath. As you can see, there is two phases, S1, phase 1, phase 2 and phase 3. From the slope of the phase 2 and the phase 3, you can determine the inhomogeneity of ventilation and airflow obstructions. There are some parameters, but there are not much literature about the use of capnovolumetries. But from the capnovolumetry, especially the slope of phase 2, phase 3 indicates the inhomogeneity of ventilation, and the slope of the phase 2 indicates the FEV1s. One interesting case, one 26-year-old female who complained of shortness of breath. Here you can see the FEV1 FVC is 74.6%, so does not qualify obstruction. This patient probably has a restrictive pattern. So restrictive pattern in spirometry is very non-specific. So you have to have, if you get restrictive pattern in the spirometry, you have to have confirmed by the lung volume estimations. Most commonly confirmed by the body plethysmography. So decrease in FVC in spirometry may be in two conditions, either decrease in total lung capacity or increase in the residual volumes. That can give rise to pseudo restrictions. So for this patient, we have gone for the total lung volume estimations. You can see their patient has FRC has 121%, TLC is normal, and RV by TLC is 49%. That is, usually it should be up to 40% is normal, and percentage of predicted is 179%. 140% is the normal. So this patient has increase in FRC because of the airflow obstructions. If you go for the simple spirometry, you may not be able to identify the airflow obstruction. Only after going for the total lung capacity estimation, you can confirm this patient do not have a restrictive pattern. It is basically the patient has pseudo restriction. So in body plethysmography, we basically measures two things. One is the FRC and the specific RAW. And specific RAW is calculated. Specific RAW is basically flow standardized work that required to complete the maneuvers. And one by specific RAW is the GAW. That means the conductance of the airways. And we have seen that the RAW and GAW can use as a parameter to assess the airflow obstruction and also the severity of obstruction. Normal, the normal values of specific RAW is approximately 0.38 kilopascal per liter per second. So using the specific RAW and GAW, you can also able to diagnose the airflow obstructions. Suppose these patients. This patient, you can see the flow volume loops, sorry, not the flow volume loop, the specific RAW loop is opened up, so that means the patient definitely have airflow obstruction and the FRC is increased. What's about the RAW and GAW? That is, the RAW and GAW has decreased after giving the post-bronchodilator. So more than 20% decrease in the specific RAW has a good sensitivity and specificity to diagnose reversibility by using the body box. Next, one equipment we mostly talk about, that is the IS or forced oscillation techniques. There are several parameters are available, but most commonly used parameters is the R5, X5, and both R5 and X5. What are the implications? If only R5 is abnormal, that means the patient has airflow obstructions. If the X5 is abnormal, there are three possibilities. One is the airflow obstruction, the patient may have restriction, there may be inhomogeneity of ventilations. And if both R5 and X5 is abnormal, that means the patient has severe obstruction. like spirometry, we can also assess bronchodilator response in the IOS or FOT. If there is 40% decrease in R5 or 40% improvement in X5 and 80% decrease in reactance area, you may classify the patient has bronchodilator, the small areas functional or have bronchodilator response. So I can show some examples. This patient has FEV1 FVC ratio is 62%. Okay? FEV1 is 76%. That means the patient has mild airflow obstruction. And after giving the bronchodilators, there is a 4% increase or minus 5% decrease. So this patient has mild airflow obstruction but do not have any bronchodilator response. But if you go for the FOT reports, you can see there is a severe obstruction, R5 is increased, X5 is also more negative. And after giving the bronchodilators, the improvement of R5 is more than 40%, and improvement in X5 is more than 50%. And also the reactance area has been improved from 86%. So though the patient has mild airflow obstruction, do not have bronchodilator response in the spirometry, but the patient has significant involvement of the small areas that responded with the bronchodilators. So always there may be some discrepancy between the spirometry and the FOT. Next, another patient, that is a 57-year-old female with history suggestive of poorly treated bronchial asthma. What we have observed that FEV1 FVC is 50% and FEV1 values is 29%, very severe airflow obstruction. And after giving the bronchodilator response, you can see there is an improvement in the FVC. So the patient has severe airflow obstruction with significant bronchodilator response after in the spirometry. But what happens in the IOS or FOT? Here the patient definitely have airflow obstruction. The R5, X5 has high, and after giving the bronchodilator, there is no improvement. So though the patient has severe airflow obstruction and significant bronchodilator response in the spirometry, in FOT there is no bronchodilator response. So small areas and large areas, if there is a heterogeneous, so you cannot able to pick up by single test. Next one thing is, here you can see there is an FEV1 FVC reduced, the patient has airflow obstruction, and also there is an basically the patient has FVC is also reduced, there is pseudo obstruction is there, and significant bronchodilator response. But what happens after giving the bronchodilators, the R5 has increased, X5 also become more negative. So that is a paradoxical response, like the bronchodilators, here also you may get paradoxical response to bronchodilators in case of FOT also. One patient, that is the 43 years old female who complain of occasional wheeze. You can see the spirometry is absolutely normal, there is no bronchodilator response. But here you can see there is an R5 and X5 is increased, there is significant bronchodilator response. So here, by just going for the spirometry, you may not able to diagnose the airflow obstruction, but if you go for the FOT, you can able to confirm your diagnosis.
Dr. De, do you have many more slides?
I will skip some slides. So this patient has airflow obstructions. There is a significant improvement, but there is no significant improvement in the FOT parameters. I am just keeping one or two. This is a 12-year-old boy referred to us for having suspected bronchial asthma. If you go for the FOT, you can get the FOT is high, and there is no significant bronchodilator response. But if you go for the spirometry, you can see there is a pseudo restriction because the FEV1 FVC is both are reduced. Here, if we go for a spirometry, we cannot able to diagnose, but if we go for both the tests, we can able to confirm the diagnosis. And during the COVID period, there are a lot of talk was, can we use the FOT as an alternative to spirometry? So because there is a chance of aerosolization during the spirometry, so many people advocated for the use of FOT. But we studied before the COVID, and we published also before the COVID. We compared the spirometry parameters with the FOT parameters, and we have observed the correlation is very weak. And if we use the FOT parameters to diagnose the airflow obstruction or severity of airflow obstruction, the area under curve is 0.75. So they are in a good, neither good sensitive or specific. So single test is not sufficient to diagnose both the small and larger abnormalities, and lung function do not make any diagnosis. The diagnosis is based upon your clinical symptoms, and only the lung function test will confirm the airflow obstruction or restriction. Interpretation based on the pretest probability of the lung disease like history, other investigation, and what is known as clinical correlation is necessary. And IOS is not a substitute for spirometer, rather a complementary test. Thank you.
So, thank you, Dr. De. That was very elaborate, and our students have definitely taken a lot of lessons from this. Now, what Dr. De said is about physiologic assessment of airway diseases. And we will have the next, the HRCT assessment in airway diseases. Now, before that, I want to show at least one information. This regression equation, reference equation from Eastern India, Dr. De and the audience. Initially, it was run by Chatterjee long back in the late 80s. In the 90s, Dr. Kamath did it for India, but again, the population was really in Madras. So, Dr. Kamath had one of his students' postgraduate dissertation, which was done in Chennai. And we from National Allergy Asthma Bronchitis Institute, this is in my hand, and this is from the website of National Institute of Health Government. Reference equation for spirometry interpretation for Eastern India is there, published in Lung India in 2015. So for all of you, there is a regression equation for spirometry interpretation from Eastern Indian population, Lung India 2015. It's a free article, anybody can download it. So thank you, Dr. De. Questions or comments, if any, we'll have in the panel discussion. And now I invite Dr. Sumit Sharma to talk on HRCT in airway diseases. Please keep your questions and comments in store. We'll discuss them in the panel.
Okay. So good afternoon, everyone. So I'll be talking on the topic of radiology of obstructive airway disease. So I'll be only presenting the HRCT perspective of it. So I head the department at Imaging Interventions for HCG Eco Cancer Center in Newtown. The content of this topic will be anatomy of the airway, the small airways, direct and indirect radiological signs, the common airway pathology in obstructive airway disease, asthma and COPD in bit, and bronchiolitis, inflammatory and fibrotic a bit in detail. So I'll skip through the slides for anatomy. I would Yeah, I would like to show this to you, which is a secondary pulmonary lobule, and reads definition. You can see the contact radiograph over here. So in this you can see this is the interlobular septum, and these are the bronchus, the terminal bronchioles that you can see, and this is the pulmonary artery. So we'll be dealing with these more in detail. So as we all know, the airway is accompanied by the pulmonary artery even at the level of secondary pulmonary lobules. The area around the terminal bronchiole and the first order respiratory bronchiole is called centrilobular portion. Now the pulmonary vein runs between the pulmonary segment as well as it connects to the interlobular septum. So this forms the connective tissue, and the thickening of this is mentioned as interstitial lung disease. So distance from the centrilobular portion to the peripheral structure, including interlobular septum, pulmonary vein and pleura, is kept constant at about 2.5 mm. Now here you can have a look at the centrilobular area, which we'll be dealing in detail in HRCT. So the peripheral area that you see is the pulmonary vein. This forms the interstitium, and these area over here, as you see, is the centrilobular area which is surrounding the pulmonary artery branches. So this is visible till the first respiratory bronchiole at 2 mm. Now coming to scope of HRCT, we have already dealt in detail with the GOLD, that is the Global Initiative for Obstructive Lung Disease. Now if we come to relate it with HRCT, we know that identical GOLD stages will have different morphological appearances at computed tomography. So HRCT has this ability to actually visualize airway till a diameter of nearly greater than 2 mm, but below that we will not be able to visualize it. So that corresponds to subsegmental bronchi. Now therefore a centrilobular region can be recognized as an area around the tip of the visible pulmonary artery, as I have already shown it to you. Again the same image, as you can see over here, we have the secondary lobule. Now this is at the tip, we can see this is the smallest the pulmonary artery that we can see over here. Area surrounding it is the centrilobular area. Let's have a look at the first HRCT. This is a normal lung, and you can see this is the pulmonary artery, and these are the distal branches. Around it, what you see as this is the centrilobular area, and this is the minimal amount that we can actually visualize in the HRCT. You can actually visualize that there are no bronchial airways that you are able to see at this level, because they are beyond the scope of HRCT. Now, having a look at the small airways disease, we have to now, since we can't visualize them proper, we'll have to have some direct signs and indirect signs to pick up the pathology. So direct signs are what we see as thickened peripheral airways. They can be seen in the proximal portion, and only in case of chronic bronchitis or asthma is when we see them. Indirect signs can be very non-specific, and they can present with overlapping features like air trapping, subsegmental atelectasis, mosaic lung attenuation, which has to be differentiated from crazy paving pattern, and centrilobular nodules, which when they are in a branching pattern are known as tree in bud opacities. So coming to air trapping, in a case of bronchiolitis obliterans, which is a kind of a constrictive bronchiolitis, you will see there are areas which look very patchy. So this is not like a normal lung. So what you see over here, these are the normal lung tissue, and what you see over here in the surrounding areas, these are low attenuation areas. So these are the areas of air trapping in a case of bronchiolitis obliterans. Have a look at subsegmental atelectasis. Are you able to pick it up? So you see some thin linear flattened structure over here. This is the collapsed lung segment. Apart from that, you can also see it over here. Coming to mosaic lung attenuation, this is another case of bronchiolitis obliterans, you can see. So in this case, you see there are patches of lung attenuation differentiations. So you can see this as a higher attenuation, another patchy area over here which is of low attenuation. Similarly on the left lung as well. So this you have to differentiate from crazy paving pattern, which we usually see in case of pulmonary edema, ARDS, or infective pathology. We also see it in case of alveolar proteinosis. So this is another image where we are seeing tree bud or centrilobular nodules. So centrilobular nodules, which you can see over here, these are at the center of our secondary lobule. So when you see them in a branching pattern, so in case of TB and most of the infective pathology, so you will find them looking like this. This is the branching pattern of TB. Now coming to common airway pathologies in obstructive airway disease, asthma and COPD. Now asthma per se is not visible to us as radiologists. We have to pick up on the indirect signs. And asthma, most of the times it comes out as a complication for us. So most of the asthma patients come with complications of ABPA, and which is why we don't say that we can diagnose asthma on HRCT. However, HRCT features of asthma which we do come across are non-specific and overlapping. We see bronchial wall thickening, expiratory air trapping, inspiratory decreased lung attenuation, which is quite non-specific, small centrilobular opacities, bronchial luminal narrowing, reduced bronchoarterial ratios. Also some, in very few cases, we will find subsegmental bronchiectasis. So this is a case of ABPA where we have complication of asthma. You can see over here, this is the branching pattern over here, and mucoid impaction is seen. Along with that, we see the V and Y shaped configuration. Apart from that, you are also able to see some kind of bronchiectatic changes over here. So this is a central varicose bronchiectatic changes. To confirm, we obviously have to correlate with clinical findings, and there were elevated IgE levels and a mildly raised eosinophil count. Apart from that, fungal hyphae was found on microscopy. Coming to how do we go about COPD? So major CT phenotypes, we can see a spectrum of 3 components which might coexist or overlap. Firstly, emphysema. These are areas of low attenuation. Bronchitis, which are airway wall thickening. Bronchiolitis, air trapping. So this air trapping is the greatest challenge as we all come across. How do we assess emphysema? We have qualitative as well as quantitative assessment. So coming to qualitative assessment, we have severity, type and distribution. There are multiple ways in which we can describe it. How we go about it is centrilobular emphysema, which could be trace, mild, moderate, confluent, or advanced destructive emphysema. So advanced destructive variety can lead to lung collapse. Then we have panlobular emphysema, which is mostly in case of alpha-1 antitrypsin deficiency. We also see paraseptal emphysema, which could be mild or substantial. Rest of the airway disease that we see are bronchial disease, which is thickening of the walls, and small airway disease, which will be coming in detail now. What are the types of emphysema? To put simply, we have centriacinar, panacinar, paraseptal and parasecretional. Let me explain to you with the images. You can see this is a centriacinar variant, and this is the most common type in case of smoking. So there is destruction of parenchyma around the terminal bronchiole, as you can see here. This is a larger one, but it is yet to form a bulla. More marked in the upper end, it is mostly symmetric. Coming to panacinar, it's a difficult one to pick up because the entire lung and all the acini are involved in the lobular segments. So it is mostly found with alpha-1 antitrypsin deficiency, as mentioned before. Coming to paraseptal emphysema, so paraseptal emphysema is quite easy to pick up for everyone, and it is the one which causes a lot of complications for us, as in whenever I as an intervention radiology have to go for some kind of biopsies, these are the ones which actually collapse and form maximum pneumothoraxis. Coming to parasecretional, so these are the ones which actually are overlapping most of the times and very difficult to differentiate from honeycombing pattern. So honeycombing pattern, as you all know, is seen in case of IPF, usual interstitial pneumonitis. So this pattern, as you can see over here, these are subpleural, and actually these are not the paracikatricial ones. This actually is a paracikatricial emphysema. This is still honeycombing. Coming to quantitative assessment, we have CT density mask and we have parametric response mapping. Going in bit of a detail over here, CT density mask is one of our texture analysis methods. What we do is, if you see A to C, we have a chronic heavy smoker. So in this you can see some kind of emphysematous, mostly pan-acinar variety of emphysematous changes. This we will put across in our density mapping, and you see the density mask being placed. Over here you can see the yellow portion, which is the moderate variant of emphysema. Anything that is in the red zone is actually a high grade of emphysema. So a lot of lung destruction in this red zone. And we can actually compare both the lungs and come up with this data to explain how the emphysema is destroying the lung. Another method for us is parametric response mapping. In this, what we do is we have the Hounsfield units marked at minus 950 and above minus 856. So if you see the green zone over here, this is the normal lung. And if you see the yellow part, this is areas of air trapping. Okay. So these are areas of air trapping in case of an emphysematous lung field. And you see over here, these are red areas. These red areas are the emphysematous areas. So if you see this case, this has more, actually in this case, you see air trapping is more. So this is a pathology which is in the small airway disease. Over here you can see this has a combination of both the diseases. Okay, so this was an image of a 61-year-old man with gold stage 3 emphysema. Now, if you want to combine both qualitative and quantitative assessment of emphysema, so there is a new technique which was published in 2019, dark field chest radiography. What they are doing at this level, I have not come across it in my practical knowledge. So what they do over here is take coronal image of CT and an axial image, do the marking on the CT and represent it on a chest X-ray. So as you can see over here, they actually correlate, and the correlation actually is known as dark field coefficient. So once it correlates and they are able to differentiate between mild, moderate and severe variant of emphysema. So as you can see, the p-value is quite significant. Coming to a small discussion about bronchiolitis. So again, as in emphysema, as also in bronchiolitis, we have direct signs and indirect signs because we are not able to visualize them again. So indirect signs, we have thickening of the bronchular wall by inflammatory cells. What we see as central lobular nodules again. The tree in bud pattern, as I've already explained, Y or V pattern. Poorly defined central lobular nodules, which I'll be showing in images, and bronchiectasis. Very rarely we are able to pick them up close to the pleural surface. Coming to indirect signs, we have mosaic attenuation. As I've already explained, we have to visualize them on expiratory CT. Air trapping, easily detected when focal, difficult to detect when diffuse. Okay. So coming to a bit of a detailed study about bronchiolitis, we have two variants, inflammatory and fibrotic. In the inflammatory variant, we have infectious hypersensitivity pneumonitis, respiratory bronchiolitis, follicular lymphocytic and panbronchiolitis. Add to it also bronchiectasis that we can see in some. So the fibrotic or the constrictive variant is usually seen in case of post-infectious toxic fumes, transplantation, post-transplantation patients, cryptogenic collagen vascular diseases, and also in drug abusers. So what is the difference that we are able to notice? Most of the times what we see are central lobular nodules. Sometimes they are ill-defined, sometimes they are well-defined, sometimes they are with tree in bud opacities in the branching pattern. But in case of constrictive, one very important thing that you should notice is air trapping on expiratory CT. Now, in case of inflammatory or any of these variants infectious, so you don't see any kind of air trapping. So that is something unique to constrictive bronchiolitis. So here we have instance of a 35-year-old man with cellular bronchiolitis secondary to mycoplasma infection. You can see over here, these are very poorly defined central lobular nodules and which are presenting in a branching pattern. Another instance of a 57-year-old cigarette smoker with respiratory bronchiolitis. You can see some peripheral central lobular nodules over here, and there are patchy ground glass opacities, which are not very clearly seen, but yes, you can see, focus over here some places. 30-year-old man with post-infectious constrictive bronchiolitis and with history of mycoplasma pneumonia. You can see some patchy areas. As I've already shown to you, these are areas of mosaic attenuation. So entire lung on both the sides, you can see some areas which are with low and high attenuation, but there are no septal thickenings surrounding it, which actually differentiates from crazy paving pattern. Also note some bronchial wall thickening and cylindric bronchiectasis. Another case of a 55-year-old man with cellular bronchiolitis secondary to subacute hypersensitive pneumonitis. So this pattern is very pathognomic of hypersensitive pneumonitis. You can see the entire lung segment has patchy areas of low attenuation and high attenuation over here. There are multiple central lobular nodules which are not very clearly defined. So these are poorly defined central lobular nodules. Another case of a 37-year-old lady with rheumatoid arthritis and follicular bronchiolitis. So we see a few larger nodules over here, right? We see a few larger nodules as well as we see some bronchiolitis and bronchiectatic changes. So this is a case of a rheumatoid arthritis with follicular bronchiolitis. Another case with rheumatoid arthritis and bronchiolitis obliterans, you can see mosaic attenuation pattern. So now what you notice over here is the air trapping. These air trappings are very much pathognomic of constrictive bronchiolitis. Another case with constrictive bronchiolitis, what you see is a worker in a microwave popcorn flavoring factory. Now this patient had severe obstructive lung disease of Gold stage 4. You can see this entire area, this is with low attenuation. Very difficult to pick up unless you have seen a lot of images which show you normal lung. Now let's understand what are the recent advances. We have been trying to enumerate the health, composition, number and location of the small airways in an important goal for chest imaging and research purposes as well. So recent studies we have found that full-scale airway network. Till now we have been using the vascular channels to try to make a central lobular pattern to understand the pathology, anatomy, but over here we are using the computational fluid dynamics. So we are actually going down to the level of the bronchioles to find out what exactly is the structure of the bronchiole, and this method is even now in research and has not been put into conventional use. Micro CT has been used for the ex vivo visualization of inflated frozen human lung specimens. This is also in research level and we are not able to use it at a commercial level. And finally we have use of fractals. This is a mathematical method for determining the branching pattern of structures, directly enumerating the simplification of airways. Much research is going on this field, but yet it has to come into practice. To conclude, so individuals with identical Gold stages may have different morphological appearances at HRCT. These reflect very important differences in the underlying pathophysiology as well as in the genomic profile of COPD, as we have already discussed in the prior presentations. Combined quantitative and visual assessment of CT may contribute to improved personalized care for patients in COPD. Bronchiolitis may be classified, as I have already mentioned, as inflammatory and fibrotic. And when it comes to high resolution CT findings, correlate with the histology of different forms of bronchiolitis. When high resolution CT features overlap, we are not able to actually come up with a diagnosis. So what we do is narrow down using the differential diagnosis and use our clinical knowledge to come up with the appropriate diagnosis. To acknowledge, I really thank Dr. Parthasarathi Bhattacharya for giving me this opportunity, and Dr. Sumit Mukhopadhyay and Olga Chatterjee from Tata for making sure that I stay interested in interventional radiology apart from HRCT of the lung. Thank you so much.
Thank you, Sumit.
Thank you.
Sumit Sharma, তাই তো তোমার সাথে পরে যোগাযোগ করো চলো প্যানেলটা দাও So this is recapitulation time and we have 5 panelists. I'll tell them to quickly দেবান আপনি ওদিকটাতেই বসুন যেটা আপনাকে দিক থেকে দেখতে পাই আমি Dr. Devraj Josh, Dr. Sivarasmi Unithan, Dr. Pranab Mondal, Dr. Sudeep Ghosh and Dr. Saurabh Maji. All the panelists. So, what happened? The speakers have spoken enough, okay? Not much more them to speak for. I will see how much our panelists have digested this and what is the take-home messages. That means we need the distillate. We are lay people, so we need the distillate from them. What is the take-home messages? So, no, not this one. Hello? Not this one. Not this one. Change it. দিয়ে দিয়েছি দেখো বাবা আমাকে ওটা নট দিস ওয়ান আমাকে দেখাও কি করছো Start with, who is the senior most here? Jivanda Kena? Pranab. Dr. Pranab Mondal? Okay. Pranab, let's get to a very clinical question. They were talking of all these things. Now, no, it's okay, you go there. I know all this. Pranab, when do you ask for a repeat spirometry? I say repeat spirometry in a patient with diagnosed obstructive airway disease. Again, for all the panelists, we have a little sort of time, so very focused, preferably within 90 seconds, 1.5 minutes. So, Pranab, when do you ask for repeat spirometry?
When do you ask for repeat spirometry? Actually, spirometry, what the role of spirometry in obstructive airway disease? It is required for diagnosis and for follow-up. In diagnosis, there is variable airway obstruction in asthma and persistent RAFO limitation in COPD, in classical cases. Repeat spirometry in asthma is done after 3 to 6 months after starting controller therapy to know the personal best FEV1 of that person and to show the lung function improvement with therapy. And periodically thereafter, actually every 1 to 2 years, to see the decrease in lung function, if any. A low FEV1 is an independent risk factor for exacerbations, so more frequent spirometry is needed in particular persons of asthma patients who are at risk of fixed airway limitation and increased exacerbations. In some asthma patients, we start, the initial spirometry report is inconclusive and we start controller therapy on clinical ground. In that particular cases, the repeat spirometry should be done after 3 months of controller therapy to see any improvement in the lung function. In case of COPD, it's a progressive disease in spite of treatment. There is gradual fall of FEV1. So, every 1 to 2 yearly spirometry should be done to identify those persons when there is rapid decline in lung function. And again, spirometry is repeated in COPD patient when there is discrepancy between the symptoms and the level of lung function to see the any extra therapy which should be given or the presence of any comorbidity like heart failure in case of COPD. And obviously, repeat spirometry should be done in COPD patients when there is any interventional procedure like lung and heart operation or any upper abdominal operation. Thank you.
Pranab, so as I take it, so repeat spirometry in COPD you said about a year, in asthma you said about 6 months.
Initially 6 months, then yearly.
Any disparity between symptom and the spirometry?
In case of COPD,
and also
sir.
probably borderline cases.
In borderline cases.
Dr. Sajalde, any one-liner on this topic?
I fully agree with him, because COPD, it is generally progressive. Even after doing spirometry, we are not able to say any improvement, not only in the FEV1, FBC, and also we have observed there is no improvement in also small airways function that we have observed in our ongoing studies. Though the patient has symptomatic improvement, CAT score are improved, but physiological parameter does not improve.
Actually, improvement in COPD is not improvement in the lung function.
Yes.
Okay, no conversation through. Okay, it's always through the moderator. Okay, now, Sudeep, so will you please summarize both together? Actually, the first one was functional anatomy and physiology. The second part was assessment of airway diseases. Suppose I was sleeping, and tell me the take-home messages from these two topics together. I was not sleeping anyway. Please.
So, Dr. Balamuke sir nicely talked about the physiological anatomy and physiological aspect of airway diseases. So, what I think the most important part is what is small airway, that's less than 2 millimeter or beyond 8 generation of our branches. Another important is this is, although it's a silent zone, but in COPD, small airway may be the first affected area. And the clinical implications of small airway involvements are like sir already told, dynamic hyperinflation. So due to early closure or shifting of the equal pressure point more distally to the less non-cartilaginous portion of the small airways leads to air trapping and dynamic hyperinflation, leading to those length and tension abnormality of diaphragm, which leads to the which changes mechanical properties of lung, leading to respiratory failure and other consequences. So these are important anatomical and physiological aspects of the airway diseases.
Take home messages. Okay, so for a clinician, yes, out and out clinician who doesn't know about all these things, do you summarize like this, that so-called, when you talk of airway diseases, for a clinician, predominantly it is obstructive airway disease? And when you talk of obstructive airway disease, the main concern is on small airway disease. Do you agree with this?
Yes.
Divanda, any comment?
Well, I have a question for Dr. Balamukesh. Do we have beta-2 receptors all through the airways? What is the anatomy of beta agonists, beta receptors in airways?
That's a very good question, sir. Beta-2 receptors are predominantly present in the conducting airways. They are very scantily present in the small airways. But the corticosteroid receptors are present throughout.
Okay. So if that is so, our bronchodilators are of no use for small airways dysfunction.
Yes, that's right.
Okay.
Only inhaled corticosteroids predominantly.
Okay. So, Divanda, a good political answer to your question would be, number one, you said, are there beta receptors in the small airways? Is it? So, number one answer would be, it's a good question. And number two is, it varies according to the person. Okay? Now coming to...
Some of us are lucky to have beta receptors in our terminal airways.
Exactly. Okay. It depends. So, now we'll go to Naturally, it's to Debraj. Dynamic hyperinflation. This is take-home message, Debraj, not elaboration. Exactly the point. Dynamic hyperinflammation. Why and how? Go on.
Dynamic hyperinflation, we will talk in ventilated and in non-ventilated patient. As you told about the assessment and management part. In
Good topic. Sorry to interrupt. Take out the ventilated out. Now it's only in the non-ventilated.
The non-ventilated part, the ward dynamic means it is somewhat related with the exercise related part, because with more and more passage of the, when we increase our respiration rate, the duration of the expiration decreases and more accumulation of the gas. So it is measured by end expiratory tidal volume, which is somehow reciprocated by residual volume by total lung capacity, but it is best measured by the cardiopulmonary exercise testing, where the significant contribution of the dynamic hyperinflation can be assessed. Regarding the management part of the dynamic hyperinflation, obviously the bronchodilators, because it will reduce the resistance, it will open up the airway and it will decrease the end expiratory flow volume. The second one is due to some of the studies have showed that inhaled furosemide or those kind of things which will, but predominantly the management is through bronchodilators.
Okay. So practically, dynamic hyperinflation has been reinvented. It was there in the 1980s, Divanda.
Well, O'Donnell's paper was quoted by Bala Mukesh, I think. O'Donnell was the main author describing what, in simple words, our tidal breath is right in the middle of our total lung capacity. In advanced patients with airflow limitation, if we exercise, then they have no way of increasing their tidal volume. So what they increase is their frequency of breathing. If you increase your frequency, you have less time to exhale. So if you have less time to exhale, you have more hyperinflation. So gradually your tidal breath shifts towards the total lung capacity. And as you showed with your carbon compliance, that as you move towards your total lung capacity, your lung compliance decreases. And that's what makes them so unhappy when they exercise.
So, these are learned people. Take it from me, a junior. There was a static hyperinflation, there was a dynamic hyperinflation. So, what has happened? The chest wall always tries to recoil outwards. The lung, by its intrinsic pattern, geodesic pattern of the fibers, always try to recoil inwards. So, there is a mutual. This mutual is the functional residual capacity. And any loss of intestinal recoil, lung recoil, sorry, they can hear me. Any loss of lung recoil, they can, oh, you are holding it. Leave it. Okay. Any loss of lung recoil where the chest wall will punch outwards. This is what happens in COPD, particularly emphysema, because of the loss of elastic recoil of the lung. This is static hyperinflation. Now, whenever you are taking breath, any airway, however constricted it is expanding, because the whole chest is expanding. So air will go into. And as you are expiration, you are pushing it, so some of them are very constricted, they close, they can push out their air, they will trap the air. This air trapping in totality is the hyperinflation, that is dynamic hyperinflation. Okay. Now, Saurabh, then, now standing here, spirometry versus iOS.
I will go for the iOS.
No, why you will go? You are not allowed to go unless you explain.
Patients shouldn't be able to afford iOS.
Yes. You are not allowed to unless you explain.
For the small airway, after discussing of this by the lecture, and I will prefer the iOS, because it is the effort independent. We can do very comfortably for the children also, and we can assess the small airway which are missed in our normal spirometry.
Okay, I will go to Dr. Sajalde for your answer. So, Dr. Sajalde, no, specific question for you. Do you think iOS can give me all the answers which is there in spirometry, or spirometry is complementary to iOS?
iOS is complementary to spirometry because you have to go for spirometry first. In spite of spirometry normal, if you suspect the patient may have small areas involvement which is not picked up, you should go for iOS. If you go directly iOS, you may miss many patients.
Okay, you have something to say?
We wouldn't
Devendra,
like this audience
Devendra,
to
okay,
go
both.
home with the message that you can forget about spirometry.
Okay, we'll come to that. Sort of very quick answer. Are there regressive equations for iOS in Indian population?
No.
The answer is
Not
no.
yet.
But
Wrong,
again,
wrong.
yes.
Dr. Saibal Mitra has published iOS parameters, and myself has published A40 parameters that is published in Lung India, and it is also incorporated in some instrument also. So it is a wrong conception.
You are having something.
And for pediatrics, I have also published, and Dr. Anurag Agarwal also
Actually, lack of regressive equation does not justify not doing iOS. Regression equation will be there. If not today, it will be done tomorrow. That is not the point. Okay, hello, sir. You are telling something.
Yes, so not the equations, but predictive values for forced oscillation have been published by the Chest Research Foundation in Pune. And they are not for iOS, but for forced oscillation.
What I mean, even if not today, it will be done tomorrow. That is not a valid contraindication of not doing iOS. But again, there is an age effect. At my age, I will start with spirometry. Dr. Sajalde, spirometry, then iOS, and Saurabh jumps to iOS straight.
No, not the iOS state. First spirometry, then iOS.
But again, I tell you, as Dr. Sajalde was speaking, particularly remember that 26-year female who was, you did this pseudo-restriction pattern, you did body plethysmography, you did, and you showed that there was no restriction actually. For me, the whole answer was there in the clinical correlation. Actually, spirometry is not an answer in itself. It is the clinical correlation because the graph, there was degree of reversibility. Reversibility is not an all or none phenomena. So this 15%, 12% probably will not be there in coming days. Reversibility is a continuum. I saw in that graph. So look at the curve. There was tinge of reversibility there in that curve. Yes.
I want to add one thing, that is the static hyperinflation. That can be diagnosed by the A40 parameters because the inspiratory difference between the inspiratory minus expiratory, it is more than 2.28 hectopascal. That means the patient has dynamic hyperinflation at rest, which is not able to diagnose by any other investigations.
But for the audience, juniors, static hyperinflation has to be suspected first. So make the patient strip. See him standing from the front and from the back. Otherwise, you cannot diagnose. Actually, that was the reversal of the transverse diameter versus the anteroposterior diameter ratio. Okay.
And also add to this, because the pseudo-restriction may be involvement of the small airways. That is missing by the spirometry. So, A40 will help to diagnose there is just end expected tidal flow limitation.
I fully agree. Particularly, iOS is interesting because number one, it is effort independent. And number two, small airway disease that is truly cannot be diagnosed by standard spirometry. Okay. So naturally it comes to Shiva, Rasmi, small airways. Again, you have seen both. The physiological versus the radiological test for small airway disease. Which one you prefer? Which one is more reliable for you at the present moment? The physiological one, Dr. Sajalde, or the radiological one, Sumit Sharma?
Sir, I feel that overall physiological assessment of the small airways is generally a bit difficult because it takes a lot of affection of the airways, small airways, to actually see the abnormality detectable. Because of the total cross-section area of small airways being so big. But having said that, physiological assessment like spirometry, where you are looking at the maximal expiratory flow with just 75% or 50% or 25% forced vital capacity remaining, that is not a bad parameter. It does help in showing you whether there is small airways lability or not. Coupled with that, if you have an impulse oscillometry, where because of the fact that we are giving different frequencies of sound waves, you can actually utilize the fact by, you can differentiate between the airways resistance in the large airways and small airways. So you are being able to detect that.
I fully agree. You are going for iOS and you diagnose small area disease.
But sir,
what do you do for radiological imaging?
With CT imaging, what I can do is, with this direct and indirect signs that Dr. Sharma talked about, you can actually see where exactly and whether the problem is whether you try to differentiate whether this is a small airways disease per se, or is this a small airways dysfunction as part of the early phase of some obstructive airways disease. This is difficult with physiological assessment.
Okay, clue from her. Everybody can raise hand. How many of us here always ask for expiratory CT in a HRCT chest with expiratory? How many? Raise your hands. Siba? No, you didn't raise your hand.
No, I thought I was not part of the...
Okay. So practically, this...
Should I make one comment?
Yes, please. Go on.
Sir, we should be honest. We always use physiological variables to diagnose small airway rather than radiological variables.
No. What I am saying is with physiological assessment, you can detect the extent of airways disease.
And also Debraj accessibility. That is also important. But again, for this radiological purpose, we have to have very close correlation with the radiologist. We have to talk with them that I want this exactly, otherwise you don't get the...
Sir, unless artificial intelligence takes over, it is very difficult to quantify the...
I fully agree, unless artificial intelligence take over, lower intelligence like us has to continue. And then artificial intelligence will come in time, okay? Now, okay, Debraj, then, what is the add-on knowledge from DLCO and body box? Forget DLCO, body box. What is the add-on knowledge?
Sir, body box, the most important message, it will give information regarding the larger airways also. And from the value also, we will get mainly two values. So, conductance as well as resistance. It will give all the lung volume assessment. And second, we will have a very good reversibility criteria of from the resistance also, which is taken around 50%. Which will give the reversibility on a crux of both larger airway as well as a smaller airway. In all those FUTs as well as the spirometry, it is in context, it will give more values of the larger airways also.
Just tell me one case where you were not satisfied with anything else other than the body box.
Sir, you need to have an assessment of all the volumes. This is only test which will give an exact value of all the volumes, because even in the gas dilution test, because of the communicating bullets,
Exactly. So
especially in M5 meters.
Non-communicating airways can only be assessed by body box. They are not assessed by anything else, excepting probably ventilation perfusion study.
VQ scan is a crude study. VQ scan you cannot make an ideal, and also COPD patient, it is very difficult to undergo ventilation scan because to hold
accepted. Sudeep. Lobular air trapping, specificity and sensitivity for small airway disease.
Sir, for any sensitivity and specificity, we need a comparator. So here the comparator could be a physiological test like FE2575, FOT, or single breath nitrogen washout, or it could be a histology where pathological sample can be tested for small airway changes, and that can be compared with radiological. But as per my literature review, this CT changes in small airway disease is a new coming concept. As already radiologist told about the air trapping, and diagnosing air trapping is not always easy. Just seeing the CT scan, sometimes we may miss subtle or mild air trapping. So we need to calculate the pixels, Hounsfield units in suspected area. So that needs a dedicated radiologist also. All the current studies only showing the correlation between the spirometric value like FEV1, FEF2570. So they are correlating good with small airway changes.
I agree with you, don't have a comparator. I fully agree with at the present moment. Okay, Sumit, exactly any comment on this?
Yeah. So from my point of view, I think it's better that we correlate clinically as well as radiologically. What happens next is when we have two of the components correlating, we are in a better position to actually quantify as well as quantitatively we can say this is exactly what it is. It's a small airway disease of which pathology. So, as far as I am concerned, yes, we are able to pick up a lot of findings on CT. With experience, yes, we have picked up all kinds of air trapping. It's important that it's done in the right way. Not many places, see, as a radiologist, I do imaging. I also receive imaging from multiple other centers for me to interpret. If it's not done in the proper way, can the blame lie on the person who's reporting? That is the biggest question that we have to ask.
Sir, another,
can I?
Just
Yes, please. Devanda.
If I give you 2 HRCTs done properly, 6 months apart, can you tell me if the disease is progressing or regressing?
Devanda means I was also going to ask the same question. Can it be historical control?
Yes,
previous 6 months back and 6 months later?
I would like to put across this point that as a person who's been reporting a lot of onco imaging, we are used to comparison studies, and we have been comparing even the minutest of details in a patient with cancer. So it is not something that is very difficult to pick up, and yes, we can quantify based on how much percentage of the lobule is involved, how much of the lung is involved. So we are able to do actual analysis and quantify it.
Sudeep.
Sir, actually, there are many ways for quantification of this small
No, we are
error
not
disease.
going to quantification CT at present because that's a very big talk. Everybody knows that there are new apparatus. There is known as there is a caliper and other things. Even with one CT, they can tell you the degree of fibrosis ensuing. So probably again, Devraj, artificial intelligence will come in.
Yes, artificial intelligence
Yes,
will help.
that-
Even with artificial intelligence, just last month there is an article published in the European Respiratory Journal. It is not necessary that there will be radiological abnormalities in all small airway diseases.
Yes.
So it is not necessary that you will have some kind of radiological abnormality. Second, when you have a same CT interpolated by a different artificial intelligence, the reports will be different. So the intra-observer as well as inter-observer variation is significantly high in case of radiology.
Will be there even with artificial
Even
intelligence.
with because artificial
okay,
intelligence
I fully agree
is
with you, but again, artificial intelligence is definitely a step ahead, but still now the baton is being passed, not it has been totally passed away. I'll come to, this is Saurabh. The second part, may higher resolution help further in the assessment of small airway disease? Saurabh, that means we are talking about HRCT, higher resolution, does it help in further assessment of small airway disease?
Sir, for the assessment of the small airway disease, first we have to, it is the, we have to first to put the expiratory film. If the expiratory film, if there is a air trapping, we look for the air trapping. If the air trapping is increased, then we are suspecting that whether it is it may be the small airway disease. And in case of if the air trapping is not increased, we can think about in the inspiratory film, we can think about the airway narrowing or the bronchial wall thickening, and that also we can, from that part we can clinch the diagnosis of the COPD component, that is the bronchitis component, we can clinch. And in case of the inspiratory film, the centriacinal emphysema or panacinal emphysema or the emphysema component we are looking for in the
Okay,
HRCT
no, but again, Sumit, but there is a limitation of HRCT for resolution, isn't it? We cannot go beyond that. So what is the limit there, higher resolution, we cannot go beyond that?
Sir, usually the conventional CT is taken at 3 mm to 5 mm. And once we go below that, see, we have a collimation and we go up to the level of 0.625 to 1.
Tell me from the pleura, how far it is not visible in HRCT?
It is at the level of your dichomatous branching around 12. So you are in the 14th level. So if I had shown you the image, if you remember the image, I had shown you pulmonary artery which are at the distal level. Surrounding it, what you see is the actual bronchus. So that is the terminal bronchiole, which we only see as indirect and direct signs, right? So if we are to put across the point saying that how do we differentiate between small airway diseases, I have already differentiated between the inflammatory variety, that is the proliferative, as well as constrictive variety.
Okay, done. Fibrotic and inflammatory, and also direct and indirect.
Exactly.
So peripheral portion of the airways usually are not visible in HRCT.
No, sir, that's not possible.
No, it's not.
Yeah.
Even with higher resolution, it does
with
not.
the highest of resolutions also, you will not be able to pick up. You pick it up on contact radiograph, as well as new technology developing, which is micro CT, in which you are able to see it. But those both are ex vivo. It's not in vivo.
Sumit, is anybody doing parametric response mapping in India, PRM?
I am not aware who must be doing it, but at present I have a 128 slice CT and we plan to do it.
Okay, done. Now, they will come to Shiva. Shiva, this is out and out clinical. So, how to differentiate? This is asthma or this is asthma COPD overlap? Does reversibility have, or it is a clinical spirometric correlation?
Sir, when you ask about asthma with loss of reversibility, I think this has to be a historical diagnosis, that this is a patient whose disease has followed the course of the natural history of the disease and has gradually progressed over time with gradual regression of the FEV1, and has come to a state where it is an airway remodeling, where the reduction of FEV1 and the reduction of FEV1 by FVC ratio is not being improved with even treatment. Now, when you are comparing this with an echo, with the different echo definitions, you know that you are either dealing with a asthmatic smoker or a eosinophilic COPD. Now, if it's an asthmatic smoker, either way, we are dealing with a common point, which is exposure to smoke or some noxious stimuli like pollution. So then, that way you can differentiate, and maybe there might be some
ultimately
of these asthmatics.
It's a holistic approach.
Yes.
Clinical spirometric
Yes.
correlation. Just Shivu, one question. You have got one patient of asthma, again, no reversibility by the criteria, okay? Is there any chance that you may get back the reversibility after 6 months of good treatment?
Exactly,
is the patient
sir.
basically undertreated?
Exactly, especially in those cases where they come to you at 25 to 40 years age. They may have ignored their symptoms in their childhood, and now they have come with clinical features of asthma, but when you do your PFT, you see there is irreversibility. So then this should not be a single clinical visit diagnosis.
So don't take loss of reversibility as the final, and it is always a second spirometry after say 6 months may show better prospects.
Yes.
Last one, we have to be very quick. Pranab, clinical person, 50-year male, light smoker, 8 to 10 cigarettes per day, salesman, by which I mean he has a traveling job, presents with progressive shortness of breath for last 1 year. What's your algorithm of evaluation?
The problem is progressive shortness of breath in a male smoker aged more than 50 years. The possibilities are obstructive airway disease like COPD, asthma COPD overlap, or asthma with fixed airway obstruction, heart disease like dilated cardiomyopathy, IHD, arrhythmia, or heart failure, and DPLD.
These two have been excluded, Pranab. Heart disease and DPLD excluded, trust me, okay? You go for the next.
Then, sir, apart from his proper history and clinical examination, the diagnostics which should be done, that is the chest X-ray, ECG, echocardiography, HRCT chest, and DLCO if the facility is available.
Just Pranab, straight X-ray chest PA view in this patient. How does it help you in this algorithm?
Actually, sir, if there is heart disease, there will be cardiomegaly, and in case of COPD, there will be features of hyperinflation, and in case of DPLD, there may be the reticulonodular changes or the opacity suggestive about blurring of the diaphragm.
Proceed.
Actually, the non-cardiorespiratory causes like asthenia, anemia, and thyroid disease will not be responsible for shortness of breath for 1 year, but should be excluded. Cardiac disease is excluded in these cases. So two possibilities are DPLD and obstructive airway disease. DPLD also excluded possibly by good quality
You have done the chest X-ray in the OPD, okay?
Yes,
now
sir.
will you go for the spirometry next? Yes or no?
Yes, sir.
And then what?
Then in this particular case, COPD should be considered in any person who has progressive shortness of breath with history of exposure to risk factors, and then spirometry should be done on that particular patient. A FEV1 FVC ratio less than 80% confirm the diagnosis of COPD, a less than 70% confirms the diagnosis of COPD. But in a smoker...
Just one point. Everybody listening to this, how many of you believe that still this spirometry in this 50-year male smoker may come with a good reversibility? Please raise your hand. How many of you believe still this patient may come with a good reversibility? Please raise your hand. I am raising mine. Okay. It is surprisingly common. Okay, then go on, please.
So actually, in a smoker, sometimes it's very difficult to differentiate asthma from COPD. So the other two possibilities are asthma with fixed airway obstruction or ACO. In these particular cases, history should be taken regarding the onset, duration, progression and periodicity of the shortness of breath, the history of allergy and atopy, if there is any previous diagnosis of asthma by some physician, and history of medicine intake, what inhaler he was taking previously, and how...
Okay, Pranav. So good history taking, examination, you have done straight exam, chest PAB, you gone for a spirometry. Will you go for a HRCT chest with expiratory view if not decided till yet?
Yes, sir.
And then, go on.
Then we will go HRCT test, and if the initial spirometry is inconclusive, we will go for second spirometry after giving controller treatment to this particular patient if there is suspicion of asthma. And DLCO may also help in the suspected cases. And one thing, when everything is normal but the patient is having short of breath, then anxiety disorders, panic attacks and hyperventilation syndrome should be excluded.
Dr. Balamukesh, you want to ask him anything on this case? No? Okay. So, I think we, yes, Divanna?
Can I conclude?
Please.
If you have finished.
Please. Welcome.
What saddens me a little is there is very little participation of our young postgraduate students. You see, these things are all done for you people, and if you do not participate, then the purpose is 50% lost. So we do not have any time, but I'm still waiting. If any one of you, the PG students who are present here, wants to make a comment, don't feel worried that you will probably be talking nonsense. We all have talked nonsense in younger days. In fact, I still do. So, if you want to make a comment, you are welcome. Anybody who, did you understand anything at all, what has been done?
I'm not a PG student, but I'd like to rock the boat.
No, well, you are not allowed. You see, any, okay, yes, somebody has raised his hand. Tell us very clearly if this has been of any use or it's all useless.
So I just had one question. How do we differentiate in a PFT between a non-specific pattern and a so-called prism?
Okay, yes.
In both of them they have got FEV1.
Okay, you don't. Non-specific pattern and restrictive defect. Is that what you said? Oh, prism.
Basically, prism is a part of non-specific pattern. Okay, this is basically those patients who are smoker, you suspect COPD, but you are getting FEV1 FVC ratio of more than 70 post-bronchodilator. In that group of patients, you can think about the prisms. It is a part of the, not separate.
Sir, may I add something?
Yeah, go ahead.
In the latest guideline, it will come in the next guideline, gold guideline probably, there is an article of PRISM, sir, where they mentioned also 3 criteria. So PRISM is that you are also taking the CT scan into picture, you are taking the clinical, that pack year history as well as less than 50 years age. So all these taken into factor, whereas in inadequate inspiratory effort or inadequate spirometry or mild restriction, in which you will not have other 3 criteria like pack year, less than 50, CT criteria present. So this is the difference between PRISM and the mild restriction.
Dr. Dey, you want to conclude?
PRISM is PRISM is a part of non-specific pattern. Whenever you say that the patient is suspecting COPD, you are getting normal spirometry, then you can talk it as a PRISMs. If you go for the general population, you are getting, then it is a It has to be smoke.
Okay, there is a talk on prism.
Okay, no, but again, still, please, this is a bit academic question. Most of the thing you see is poor effort in the real life. So please be careful that it's not a poor effort. People calling it non-specific or prism. And then only we can discuss this.
Yep.
And you have to, for non-specific pattern, you have to go for the total lung capacity. TLC should be normal. Either decrease in FEV1, FVC, okay, isolated decrease in FEV1, FVC, but TLC should be normal.
Thank you. We don't have enough time. You can talk to him personally. You see, as you grow older in the profession, you will see there are innumerable patterns of spirometry. You see, each individual is unique. You have to decide on clinical grounds, on other evidences about what to do about them. Anyway, it's been very lively. Dr. Ghoshal has selected right questions for the audience and for the panelists, and I hope this has been of use to all of us, not only students, and I thank Parthu Bhattacharya to have arranged such a session this morning. Thank you.
Thanks everybody.
Thank you very much. We will now break for the lunch. I request everybody to finish it quickly. We are already running a little late. We will join again after 40 minutes.
Your lunch as quickly as possible.
40 minutes is good time.
Just check.
Working.
बोलते. Hello, hello. So guests are requested to come inside the hall. We are going to start the next session. 1427 3:30 মধ্যে ছাড়তে হবে। Good afternoon. We are going to the next session now. This session is on confusions and clarifications in obstructive airway diseases. To coordinate the session, I request Dr. Shushmita Raichodhuri. We have got two speakers in this session, Dr. Ongira Dasgupta and Dr. Professor Parvez Kaul.
Good afternoon. We'll just call Ongira first to do her lecture, and then after that Dr. Kaul, and then after that we can have a panel discussion.
So good afternoon, everyone. Welcome to the post-lunch session to those in the hall and those outside the hall. Let me start my lecture by saying that everything in airway diseases might be a contentious issue. We are still not clear. So many guidelines are coming up, and so if any disease has so many guidelines which are changing, it's contentious. I'll not be talking about a lot of things, but I'll just touch upon three important things in airway diseases. That's the diagnostics, the spirometry interpretation, which has been done by Dr. Dey already. So I'll just be very simplistic, and this is a point of view from the clinician's side. Small airway function and management, especially of inflammometry and anti-inflammatory therapy. So this is a very familiar patient who walks into our clinics almost every day. A 25-year female who has cough with phlegm, breathing difficulty. She also hears musical sounds from the chest and associated with a lot of rhinitis. Preliminary blood tests and imaging is normal. So if this is the clinical scenario, I think all of us in this hall today would do a spirometry. So this is the spirometry of the patient, and we usually do a spirometry with a reversibility, right? So the one on the my right-hand panel is the spirometry which we have done before giving bronchodilators, and the other one is the reversibility with bronchodilator results. So this is reversible, right? And a lot of it 1.73 liters. With the bronchodilator, it's 2.28 liters. So it's reversible. But there was a lot of criticism at one point of time saying that FEV1 by FVC was not just a fixed 0.7 ratio, but it decreased with age. So when that is the case, a fixed ratio of 0.7 may not be the only way to interpret the spirometry. So the lower limit of normal was then proposed. And it is very clear, and Dr. Dey has made it extremely clear in the previous session, that the reports might be different if we interpreted the results in a different way, LLN versus fixed ratio. So the red columns in the graph show that it's different. So if it is a percentage predicted like we do it in a fixed ratio one, the FEV1 is 59% only, and the ratio is 0.62. Whereas if we used the LNN method, it's 81.22% with a ratio of 0.78. So it's absolutely almost different. But the trick here is not to stop by doing one spirometry and then not follow it up by others. So airway disease spirometry needs to be done more than once. You need to repeat it in order to see what the patient's spirometry is going at. So a fixed ratio is sufficient for a diagnosis of obstructive airway diseases regardless of the lower limit of normal values. This is from a clinician's perspective and not a perspective of research. The lower limit of normal delays the diagnosis, and the fixed ratio predicts hospitalizations for COPD exacerbations, COPD mortality more acutely. So when both the tests were done, not the test, when both the interpretation was done in 4,882 patients, it was seen that patients who were classified to have a COPD or an obstructive airway disease by the fixed ratio had non-significant, very small increases in the odds of adverse outcomes. When both the fixed ratio and the lower limit of normal were used, the increases in the odds for adverse effects were larger and significant. But the strongest associations were seen for the patients who had airflow limitation by both the fixed ratio and lower limit of normal, and also who had a low FEV1 to start with. So having said that, it might be a revisit for the people who are who write down the tests for the diagnosis of obstructive airway disease. The guidelines should reconsider using both spirometry abnormalities so as to reduce an overdiagnosis and maybe sometimes an underdiagnosis of airway diseases. So this is something that might happen in the future. Coming to the reversibility testing, bronchodilator reversibility, we all know it's 200 ml and 12%. But why is it 200 ml? Why is it not a 250 ml? Why is it not a 150 ml? Why is it 200 ml? Anybody from the audience? Because I don't have an answer, because the only thing that I have read the literature, it seems like it's just a consensus. It's nothing scientific or statistical. But why is this reversibility important? So, is it important to classify a patient with as asthma or as COPD? Is it important to have a label saying that you have asthma, you have COPD, and you have echo? Do we really need it? So I think this is very important and tells us that there are three components of airway diseases. As a whole, you don't need to talk about asthma, you don't need to talk about COPD. You can just tease out the three elements of airway diseases and treat accordingly. So airflow obstruction, spirometry, give bronchodilators, airway hyperresponsiveness, present, give bronchodilators, airway inflammation or inflammometry is something which we should do more often, and the treatment would depend on what the inflammation nature is like. So when we see a patient in our clinic, supposing we had done a spirometry 6 months earlier or 3 months earlier, maybe 2 months earlier, when the patient comes into our clinics, do we repeat a spirometry? I have seen many doctors, junior, senior, irrespective of that. When a patient walks into the clinic and a spirometry is advised, and most of the time, even if it is the second or third or maybe 10th spirometry that is being ordered in the patient always gets a bronchodilator reversibility test done. Now, I'm not pointing fingers at anybody, but it's just to think, what is the goal of this repeat spirometry? Why are we doing it? It's just to maintain or achieve the best lung function of that particular person with treatment. Whatever treatment we are giving, if we do a random spirometry, then maybe the FEV1 is going to increase, and if it increases, it means that your treatment is doing good. Small airway function has been dealt with extensively, and why is it important today? Because it is thought of to be the first affection in both COPD and asthma. And like we discussed in the previous panel, this is the zone of a laminar airflow. So if you have a laminar airflow, no breath sound, no turbulence there, and therefore it's a quiet zone. But how do we detect a small airway disease? So we have discussed all these things. I'm not going to go through them. Even this was discussed. if you look at the left-hand side first CT, it's an inhalation CT, and the one on the right side is exhalation. So the air trapping amount can be quantified, like we discussed in the previous session again, and we can have an estimate of the small airway disease. The centrilobular micronodules are something that which we are seeing more and more today, maybe because we are all trained by now. It's a sign of hypersensitivity pneumonitis. And of course the treen bud, some form of an infection, TB in our country. Now remodeling is something which we should all keep our eyes open to diagnose as early as possible, because remodeling is that the airways have started to have a fibrotic element. So due to remodeling, we can have a goblet and mucous cell hyperplasia. Look at the clinical manifestations: increased sputum, airway narrowing, airway wall thickness. So which means that these patients are going to have a lot of expectoration, and if we can measure the sputum MUC5B and MUC5AC levels by ELISA or maybe mass spec, then we can diagnose remodeling a little bit earlier than we usually do. And another test that has been discussed very much, and I do not have a slide for that, is the sputum supernatant hyaluronidase. So if that is increased, that equates to a remodeling having setting. Okay, so the last part of my talk today is management, anti-inflammatory therapy. So it has been said that asthma is all the way ICS, it's all the way steroids. You need steroids in the mild asthma, moderate asthma, persistent asthma, severe asthma, difficult asthma, and difficult to treat asthma. But for COPD, it's not so. You give a bronchodilator, a single bronchodilator, it doesn't work. You give a double bronchodilator, it doesn't work. You add ICS. So to me, it seems a little bit irrational and just following the guidelines. And to the extent, I quote this, the use of ICS and LABA is no longer recommended for treatment of COPD, and we should not prescribe a combination of ICS-LABA for our COPD patients anymore. Now, I would criticize this very, very severely, because all asthma, all COPDs are not the same. So you might require an ICS in a COPD patient quite early on in the disease if you have an eosinophil hidden somewhere in the body. Same thing here, we are giving as needed low-dose ICS formoterol. Here we have an ICS and a formoterol, a bronchodilator and an anti-inflammatory in one step. But what are we doing? We have not measured the inflammation, we have just measured the airway caliber, and we have prescribed a steroid. Why? Because this patient is a patient of asthma. So being labeled as having asthma is not equal to having prescribed an ICS or an OCS. We need to find out why we need to do that in a patient when we prescribe an ICS. And this is not only true for asthma, it's also true for COPD. So the steps of GINA ladder are based on symptoms and on minimizing future risks. There is no mention of measuring an airway inflammation, although there is mention of giving an ICS. Lung function also is not measured objectively even when the drug in question is a bronchodilator. And we are treating patients with ICS and oral corticosteroids by monitoring the symptoms and risk factors to adjust treatment and treating exacerbations. So is this logical? Is something that we should be asking ourselves. See, the guidelines for treatment of acute exacerbation of COPD has a step where you can give an oral corticosteroids, tablet prednisolone 30 milligrams OD for 5 days. Now nobody knows why it's 30, nobody knows why it's OD, nobody knows why it's 5 days. So it's up to us to try and find out what we are doing with our patients. Now, who are these patients in whom we should be prescribing corticosteroids? Patients with infrequent exacerbations should not be given corticosteroids, but those who have an exacerbation of more than equal to 2 in the past 12 hours, 12 months, can be given corticosteroids. Now, this is again a statement which does not have a scientific basis, but it's there in the guidelines, all right. When there are eosinophils in the blood, more than 300, why 300? We don't know. Or sputum eosinophils, 2 to 3%. Why 2 to 3%? We do know that. Because when you work out the math, the 3% comes in the median column, and plus minus 90% confidence interval on two sides. Sorry, this is a standard deviation on two sides, is what the number that we use for patients for sputum eosinophil counts are. When it's not 300, when it's less than 100, it's said that you have to give azithromycin and roflumilast. But again, the question is why 100? We have not seen what the nature of the other cells in the airways are like. What is the scope of withdrawing ICS if there are no eosinophils? Are we overdosing ICS? So these are all pertinent questions that we need to ask ourselves and try and find it out and answer for that. I'm skipping here. See, the rate of exacerbations according to the blood eosinophil levels, if plotted in a graph like this, the blue one is an exacerbation which has been caused by bacterial, viral, or a post-inflammatory exacerbation, which means it is a non-eosinophilic one. But the orange-colored place is the eosinophilic exacerbation. So without an ICS, you get the eosinophilic and you get the other type of non-eosinophilic type of exacerbation, whereas with the ICS, the orange portion has just vanished. And this is why we use a 300. The one which is orange on dots is less than 300. They are the non-eosinophilic exacerbations, whereas look at the 300 point in the y-axis. It almost is demarcates the less than 300 exacerbations from the more than 300 ones. So how do we measure? Sputum, blood, both. So when we do a cellular count, it's called a sputum quantitative assay, and that's not the end. We need to do some measurements in the fluid phase measurements like proteins, lipids, etc. So this is the man behind quantifying the eosinophils and the other types of cells in sputum. The strategy most of us have understood by now. If it's an eosinophilic bronchitis, increase the dose of the corticosteroids, but do not decrease. Neutrophilic, do not increase the ICS. Add antibiotics if the total cell count is high. Recheck after 2 to 3 weeks. And if there is no inflammation, but the patient has an airway hyperresponsiveness, decrease the ICS and add SABA or LABA. See, this is results from our lab in B.R. Singh Hospital. If we use a cutoff of eosinophil count of 300, we found that there were patients who had eosinophilia of 64% of the times, and non-eosinophilic was 36%, which means that it's a lot of eosinophils in the patients. But if the same test was done, that is an inflammatory was done in sputum, this is what it looks like. Now going back to the question where I said that are we overusing our ICS, it seems like we are. Otherwise we would not have such an increase in the neutrophil count compared to the eosinophil counts. What are the disadvantages of sputum? It needs training, of course. Everything, any test, any laboratory procedure needs training. Wet lab facilities, that's available in your pathology. But the availability is restricted, that is enough people have not been trained. No sputum despite induction. And the process needs to be completed by 2 hours. But that's not the end. If you do not find sputum, if you are unable to do an induction, does the patient go away home without a treatment? That's not true. These days we have to get started off with the fluid phase measurements. So this is some work which got published in 2021. And what we did was we worked out the sputum supernatant and we used a mass spectrometry to find out what proteins were present in the supernatant. And those with the proteins which were raised or decreased more than 2 folds between the groups were selected. We found 21 proteins to be able to predict the disease. And when we did a functional clustering, this is what we found. There were 5 clear groups. The red one is the inflammation-related proteins. Calcium was the blue one, and the most important one was the yellow one. It said that mucin-related proteins, and the oxidative stress-related proteins was the green one. So if we, by some means, if we are able to find out what the predominant type of protein is in the patient, we could have 5 different types of patients, and we could offer a new target for treating the patients in the form of antioxidative stress-related proteins. We could give an antioxidant there. When it was mucin-related proteins, we could give a mucolytic. Inflammation, of course, anti-inflammatory therapy. So if you look carefully, this graph shows us that the inflammatory proteins in exacerbators are higher as compared to the non-inflammatory person. So the black, if you go to the first column, it's azirosidin. The black one's upright, and the second black is down. So a clear differentiation between patients who had an exacerbation or an exacerbated type of disease versus those who had a non-exacerbated type of disease. And it was true for neutrophil defensin, lactotransferrin, and lysozyme also. So being a clinician, maybe all the protein names we cannot understand so well, but believe me, these are all inflammatory proteins. This is the most important graph that I have today. If you forget everything else, please remember this, that estimation of the mucins in the supernatant fluid by ELISA can tell us about these exacerbator status. Oxidative proteins, same results. And indeed, antioxidants have been proven to be useful in patients with COPD. So, summarizing, controversies in airway disease are plenty. Spirometry interpretation in diagnosis and monitoring disease needs to be revisited. Small airway diseases need to be looked at for early diagnosis of obstructive airway disease. Anti-inflammatory therapy with ICS or OCS needs to be monitored by sputum inflammatometry with cellular counts. And of course, we need to start doing fluid phase measurements of novel protein biomarkers as soon as possible. Thank you very much. Last. Mike.
So as the slides are getting loaded, I'd like to extend my thanks to Dr. Partho and the organizing team for having invited me for this very apt session, confusions and clarifications. I mean, there probably can be no more confusing a topic, and if we had not had enough Dr. Angira actually added many more questions that need answers. So I'll be talking primarily about PRISM, and we have heard a lot about it since morning, so I'll try to skip where I find there is a possibility of duplication. So Fabri's editorial in COPD guidelines, important thing is not to stop questioning, and this is a plot of individual values of percent predicted FE1s and percent FE1 FVC ratios in about 1,000 patients who had a clinical diagnosis of COPD based on interview, based on lung examinations, lung function tests, and chest X-ray. So as you can see from this, in addition to a significant proportion of patients with normal lung function, which would used to be called stage 0 or at risk, almost 14% of the patients who were clinically diagnosed as COPD could not be classified because they had an FE1 by FVC ratio of more than 70%. despite having a reduced FEV1, that is 80% or less predicted, suggesting that gold criteria miss an important subgroup of patients with clinically diagnosed COPD. And the data from the current and former smokers enrolled in the COPD gene study, nearly 10,000 patients, which was an observational cross-sectional study, which recruited subjects from 45 to 80 years of age with more than 10 years, 10 pack years history of smoking, were analyzed. And the prevalence of this abnormality that we talked about was about 12.3%. And there have been many other studies which have tried to address the prevalence of PRISM, as are given in there. It has varied from 5.3% in the Rotterdam study to as high as 19.6% once given by Merritt et al. And if you look at this, okay, so we are, no, anyway, I'll give the pointer. If you look at this part, this is the portion that we are basically talking about. And FE1 we predicted this less than 80%, whereas the normal FE1 FEC ratio. So when we talked about the COPD gene study, the important things that they concentrated were on exposure, spirometry, symptoms, and CT imaging. And based on that, they came up with no COPD, possible COPD, probable COPD, and definite COPD, depending when the person had only exposure to more than one, that is exposure and CT imaging, exposure and symptoms, exposure and spirometry, then 3 of them, and then all the 4 of them, from no COPD to definite COPD. And the hazard ratio for all-cause mortality went on increasing from no hazard to as high as 5.81%. What are the predictors of this PRISM status? The significant factors clinically include an increased dyspnea, reduced 6-minute walk distance, increased percent emphysema and decreased total lung capacity, whatever you define it by, increased segmental bronchial wall thickening, as was talked by Dr. Sharma. Although no common genetic factors have been identified on genome-wide studies, a significant association has been found with Klinefelter syndrome, that is 47XXY. There are subgroups identified which would include a clustering with COPD subtype, restrictive subtype, and a highly symptomatic metabolic subtype. This is looking at the deciles, the mortality risks according deciles of both emphysematous predominant and the airway predominant. As you can see, this is the emphysematous predominant axis, and as you can see, that hazard ratio goes on increasing. And this is the distribution of the high and moderate risk subtypes by pulmonary function. As you can see, this is the airway predominant type, the moderate risk airway predominant type only, the combination type, and the emphysema predominant type. So subtyping these PRISM by using quantitative CT imaging was discussed very nicely by Dr. Sumit Sharma. So this study, they looked at 488 patients. 94 of them were normal, 51 had PRISM, 48 had mild to moderate COPD, and 13 had severe and extremely severe COPD who were excluded. These patients underwent clinical parameters, pulmonary functional indices, and quantitative CT. I will not go into the details of this. It has been very nicely described by them. This is the upper one is the normal patient, the middle one is the PRISM patient, and the lower one is the moderate COPD patient. The nomogram, actually, there were significant differences in quantitative CT parameters between the PRISM group, normal group, and the mild to moderate group. Once we use the composite of smoking, MLD, the mean lung density or the parametric response mapping, as he talked about, and lumen area, you could differentiate PRISM from a normal group. And once we used parametric response mapping of emphysema, that was an independent predictor which was able to differentiate a PRISM from a mild to moderate COPD. The clinical and radiographic predictors of gold unclassified, which we used in the previous classifications in the COPD gene study. It defined it as a heterogeneous group with significant symptoms and functional limitations who likely have a number of underlying etiologies. We know that nearly 9% of smokers in that study had PRISM. those who had an increased body mass, a disproportionately reduced total lung capacity, and there was a higher proportion of non-white subjects and those with diabetes. However, once we used the lower limit of normal criteria, this increased representation of the African Americans went away. And Dr. Sumit talked about the increased wall thickness and other CT parameters that I'm not going to talk about. So is there any morbidity and mortality associated with PRISM? This was a study, the Rotterdam study, which looked at the trajectory and mortality of PRISM, and they looked at these patients in phase 1 from 2009 to 2014, then phase 2, 2014 to 2016, and then followed them up to 2018 December. Nearly 1,603 subjects were included in the longitudinal part of the study. In both phases, the cross-sectional studies were done. And anybody who had an interpretable spirometry both at phase 1 and phase 2 was subjected to a longitudinal study. So they found that PRISM had 3 subsets: those who developed COPD, those who had a high cardiovascular burden and early mortality, which was pronounced as early as 1 year, and those who persisted with PRISM and had normal age-related functional decline. If you look at the mortality of PRISM, you see it is somewhere between the normal spirometry and COPD. The red one is the PRISM. And if you want to subclassify the COPD, it is between the GOLD 1 and GOLD 2 to 4. What are the various comorbidities? This was an extensive Korean NHANES trial, and they found that compared to normal group, they had a higher chance of having hypercholesterolemia, obesity, thyroid disease, and number of comorbidities overall was higher, significantly higher compared to the normal group. Then there was an association that was demonstrated in the Chinese community of around 1,000 plus patients between the presence of PRISM and risk of obstructive sleep apnea with a odds ratio of about 2. They also found that the neck circumference, waist to hip ratio, highly sensitive CRP, higher proportion of males, cigarette smokers, number of current smokers, high risk of OSA, presence of nasal and ocular allergy symptoms were significantly higher in the PRISM group. Risk of diabetes and metabolics, and this study demonstrated that these patients had a higher, the patients with PRISM were 47% more likely to develop type 2 diabetes compared to those who had normal spirometry. And there was an upswing of 121 metabolites. Small airways, you can see that patients with PRISM had a definitely increased evidence of small airway dysfunction. However, the way you wanted to define it, be it spirometrically, be it by iOS, or be it by CT features. This study looked at the spirometry, the PRISM in relationship to cardiovascular outcomes. So from the UK Biobank study, they looked at people who had no cardiovascular disease at enrollment, that was 2006 to 2010, and followed them for a mean of around 16 years, and they found that they had higher chances of developing major cardiovascular events, MI, heart failure, stroke, and cardiovascular mortality. And in the German lung cancer screening intervention trial, the LUCI study, spirometry was performed in the CT screening arm, and they found that about 1/3 of smokers eligible for lung cancer screening in Germany have underlying COPD or PRISM. This is the UK Biobank study, huge cohort of people, 350,000 plus. PRISM was there in around 11% patients at baseline and was associated with obesity, doctor-diagnosed asthma, female sex, being overweight, trunk fat mass, and trunk fat percentage were risk factors for PRISM. It was strongly associated with symptoms, comorbidities, cardiovascular disease, and all-cause mortality. So what happens to PRISM over time? Something that is known to PRISM is that it can transition from one group of lung function, one character to others. It can go into a GOLD 0, GOLD 1, or GOLD 2 to 4 as defined in this study. And over time, if you look at this, this is something that is very interesting. In the Rotterdam study, people who had a normal spirometry at initial phase of 135 had PRISM and 38 had COPD. They evolved into different classifications over time, over a period of 5 years. So normal could develop PRISM, could develop COPD, could actually have died. PRISM could go back to normalcy, can go back to COPD, or can have a continuum as a persistent PRISM. And same is true of COPD also. That's something that we must note, remark, that COPD patients in this study, around 7.4% at 5 years had a normal spirometry. So something, if you probably can take care of the risk factors, maybe you can reverse COPD as well. Around 5, that is 1.5% had PRISM, and most had persistent COPD, and some died as well. So transitions of lung function categories can actually go from normal to all these, from PRISM to all these, and from COPD to all these. Though as you move from normal to COPD, your chances of reversal keep on reducing. And in this study, they looked at as to whether 80% should be the cutoff for progression to COPD, and this particular study said that no, it should be 86%. Another paper that came very recently in the scientific reports, they looked at the PRISM with or without restrictive spirometric abnormality. Around 12,000 patients, they had non-restrictive PRISM if you had FEVC of more than 80% of the predicted, and it was a restrictive PRISM if the FVC was less than 80% predicted. And they found this is the non-restrictive PRISM, and the restrictive PRISM is shown in the red. There was an instability of the lung function. One could transcend into another, and those who had a restrictive PRISM had a higher chances of mortality. So, current and former smokers in the COPD gene study with available spirometries at different phases demonstrated that over a mean period of 10.5 years, nearly 1/2 of patients transitioned in and out of PRISM. So PRISM is an unstable group with frequent significant transitions to both obstruction and normal spirometry over time. A word about lung and diffusing capacity. If you look at the diffusing capacity of less than 80%, PRISM had many more patients than mild COPD or normal spirometric patients or even moderate COPD patients who had a DLSO which was low. And it had a definite effect on mortality, as you can see. A bit of another confusion was created by this longitudinal mortality study of a Korean cohort of PRISM patients, what is called the KOJI study, a large study, 10,000 plus patients, and those defined by airflow limitation either by fixed ratio or the LLN. And you can see that the distribution between normal, PRISM, and COPD in the two groups. And what the confusion created showed that all-cause mortality risk and cardiovascular risk, mortality risk in their patients are not increased in individuals with PRISM if the populations had characteristics like middle-aged, slight smokers without any additional cardiovascular risk. This is a diagonal approach to what has been said earlier on, adding to the confusion. And I must not leave before talking about this concern about PRISM, voiced by one of the tallest leaders in the BOLD study, that's Peter Burney. They say that Since PRISM relies on measurements taken from an FVC maneuver, it attempts to circumvent the limitation of FVC by relying on FEV1 as the measure of restriction. And since FVC underestimates total lung capacity by the residual volume, which obviously we know that increases with age and is higher in patients with airflow obstruction, and FVC is inferior to slow vital capacity in patients with obstructive lung disease because of the airway compression, the collapse that occurs because of forced exhalation. Given the limitations of PRISM, Peter Burney and his colleagues suggest that efforts should be made to improve the estimation of total lung capacity in epidemiological studies, and advances in future should be achieved with simpler and cheaper methods of measuring total lung capacity. So, ladies and gentlemen, PRISM is a commonly encountered entity. It is safe to assume that it represents a stage which can transcend into COPD, is associated with increased symptoms like breathlessness, increased risk of respiratory, which means exacerbation-like or pneumonia-like illnesses, cardiovascular hospitalizations, all-cause respiratory and cardiovascular mortality is increased in most of the studies. Some recent studies, though, dispute it is increased risk for mortality, and that's a large study done in Korea. PRISM is an unstable obstructive airway disease and transitions frequently into either a more severe form of obstructive lung disease like the COPD or into a normal lung function. Risk factors for that include female sex, increased BMI, smoking, small airway dysfunction, and reduced DLCO. Data are emerging, and we need more data focusing on the therapeutic benefits of routine therapies for obstructive airway disease, because this is one group of patients who, while you are recruiting patients for therapy for COPD, would be excluded from recruitment because they don't have a ratio that qualifies them for inclusion in the study of FV1 by FVC ratio of less than 0.7. And astute researchers argue about using PRISM and suggest better measurements in future. I thank you for your attention.
Dr. Sushmita Roy Chaudhary had to leave because she has a talk somewhere else. So I shall take over. I shall start the panel discussion with by inviting our esteemed panelists, Dr. Shurojit, Dr. Shoikat Nag, Dr. Shujan Bardhan, Dr. Parvez Kaul, thank you, sir, and Dr. Angira Dashgupta. So obviously, since this is a session where we are dealing with contentious questions, first of all, I think the first question has already been discussed in details by Angira Di, but nevertheless, the lower normal limit versus FEV1 by FVC ratio, which one would you choose? Benefits. Dr. Shurojit.
Yes. The lower limit of normal and the fixed ratio, that is 0.7, has already been discussed by Dr. Angira Di. So, according to her, the fixed ratio is enough to diagnose presence or absence of obstructive airway disease. And lower limit of normal, that can delay the diagnosis. But one thing that this FEV1 by FVC ratio, that decreases normally over age, and this is more in COPD patients, so that is not included in the criteria. So if sometimes in extremes of ages, say for elderly people, we can, by calculating this FEV1 FVC fixed ratio, we can sometimes overdiagnose that COPD or the obstructive airway disease, or in children we can underdiagnose it. So that part should be taken in consideration.
Yes, so age specific and probably even gender specific lower limit of normal, and we can probably be circumvent the possibility of high rates of false positives, isn't it, in older individuals, especially when we come across COPD in this age category.
I think in the lower age group also, we tend to underdiagnose if we are taking a fixed ratio. So in the lower limit of normal will also help in giving a better diagnosis in case of the both extremes of age group.
Right. So we go on to the next question. The wording is a bit different. So it's, how frequent is mild asthma? Gina this time has talked a lot about mild asthma and how it should be apparently mild asthma. So how frequent do you see mild asthma? This question will be to Ongiradi.
Hello. Okay. So this is something that has not been addressed so far. The mild asthma frequency is around, I would say, 12 to 20% in our clinics. I hope the number matches with everybody else.
Yes. Yes, it does.
So I'm through.
So there is this asthma insight and reality survey that was conducted among 29 countries over about 11,000 asthmatic patients, and they found the percentage of intermittent asthma to be 22 to 54%, and mild persistent asthma was 12 to 20%. So you were talking about mild persistent, but Gina does not differentiate between mild intermittent or mild persistent. Thankfully, they have brought this, the concept of the severity in Gina this time, they have brought according to the concept of difficult to treat. So now we know mild asthma as somebody who has no symptoms, well controlled, with the least dose of the medication. I go on to the next question, how to identify remodeling. This question will be to Dr. Shujan Bardhan.
Remodeling, for identifying remodeling, you have both invasive and non-invasive tests. So in non-invasive tests, the most accurate would be induced sputum test. From the induced sputum, you do a, you can separate it into the sol and the gel phases, and there are certain markers in the sol phase which will identify that remodeling has occurred. Also, we can use the, in cases where the fractional exhaled nitric oxide is high, they have a tendency to have increased remodeling. Recently, the modality of EBUS has come, and EBUS can identify almost 4 to 5 layers of the bronchial wall, can identify whether the remodeling has taken place on it. But the sine qua non is the histopathological diagnosis by a bronchial biopsy.
Yes. Yes. So, Dr. Shujan, would you agree if we say that histopathological diagnosis the mainstay for diagnosis of remodeling? Maybe you can identify remodeling with the non-invasive processes.
The confirmatory diagnosis would be histopathological, no doubt.
So, Ongiradi.
I just wanted to add,
yes.
And the quantitative CT is also helpful anatomically to diagnose the remodeling.
Yes. So that in that way we can actually measure the airway wall thickening in a much accurate fashion. Ongiradi, is it why biologics are not recommended prior to remodeling occurring?
I would not say that biologics are not recommended. The biologics are usually targeted at certain things that we can measure and identify as early as possible in any disease. For asthma, remodeling can be identified from certain clinical symptoms and also from the sputum supernatant measurements of proteins which indicate remodeling. I would not recommend a biopsy because it's something that is not feasible in clinical medicine. So in the absence of diagnosis of remodeling, I wouldn't expect that biologics are also not recommended. If we can diagnose remodeling early on, if the symptoms match with remodeling, for example, if there is a lot of MUC5AC and MUC5B in the sputum, that is one reason why we can give a patient an anti-IL-4. But again, these are all things that have not been tested so far, so understanding remodeling is sometimes quite difficult.
Which is why it is a contentious issue. Yes.
I just wanted to ask maybe you or maybe the House, should there be a repositioning of the biologicals earlier on in the treatment of asthma? Because once we are being guided to institute at a particular point in time, by then is there a significant advancement of the disease?
I think biologicals we can position in step 4 at least, if not step 5.
See, I feel that GINA is coming forward maybe in the next 2-3 years probably because they are being preemptive. We are being preemptive about inhaled corticosteroids at least now, trying to phase out SABA and trying to phase out the reliever part. Then probably in the next 2-3 years we might be seeing some studies which are telling us that preemptive use of biologics might be helping.
Agree to that, but then we need to have the correct tests in place.
Yes. That is why I'm asking whether your sputum MUC5 is that enough? Or of the
prevent the progressive airway deterioration or progressive deterioration of FEV1? What is the thought?
Arudha, our last question is almost similar to what you are asking. So we can have a detailed open house discussion about that at that time. Okay. The next question is to Shoikadda. Do we need more than one spirometry in diagnosing COPD?
Yes. Preferably we should do at least spirometry in 2 occasions because it is a natural phenomenon that a person's spirometry varies physiologically in over the times, and it is so if one spirometry may not be helpful, it may not be always indicative of the disease, whether the presence of reversibility or not, and also the the maximum exercise capacity of the patient also may be variable over time.
Do you think, Shekhar, that this could be a chronological diagnosis? Maybe you might find some amount of reversibility in some patients, and maybe we can change the diagnosis in the next few visits.
In our clinical experience, we get this type of variation in spirometry data. If the patient, we do PFT in 2 occasions, we can get in sometimes we get reversibility in second setting.
In that same patient, is there any annual variability? Maybe even if the patient is not treated with whatever we give.
That can be done. We all sometimes we see that this type of annual variability.
So there is a reported annual variability of FEV1 and FVC to the tune of around 15% in up to 22% patients, and you see a change in diagnosis by 2 years in around 24 to 32% of these patients. My next question will be to Dr. Shurojit. What is the role of low-dose CT and DLCO in diagnosing emphysema?
Yes. So, CT, especially the HRCT, is the modality of choice in diagnosis of emphysema for radiological diagnosis. And low-dose CT, that includes the low radiation dose that will be safer for the patients also. So, for diagnosis of emphysema, one is low-dose CT scan is obviously important in the anatomical point of view, and for DLCO, yes, the amount of emphysema that correlates linearly with the value of DLCO. That lower the DLCO, the higher the emphysema is. So both are important in the diagnosis of emphysema. Yes, both anatomically and physiologically.
So on HRCT, I think the emphysema index, that is a good measure.
Very important measure. The Hounsfield unit probably minus 960.
Yes,
that is indicative of emphysema index in the HRCT.
Right,
that
and
we can calculate by HRCT.
Exactly. And with DLCO, along with the spirometry below the lower limit of normal values, that suggests the presence of emphysema. In fact, as you said, DLCO is proportional to the extent of emphysema.
Yes, that's true.
So are we talking of just low-dose CT, or are we talking of a HRCT, a regular HRCT? Or are we talking of a quantitative? Sorry, I mean that maybe we have some radiologists in the audiences. So what would be the? Shall I?
Yes, of course.
So, I mean, a quantitative CT, low-dose CT, and an HRCT. HRCT, which can actually have a quantitative measurement as a part of it. So, where would you position each?
So, what I would like to inform to the entire place is there are two ways of taking CTs. So, one is segmental sequential acquisition. What happens in that is we take acquisitions at a difference of 1 centimeter. So once we take that kind of a CT, there is automatically low dose. But in case of HRCT, it is volume acquisition. It is a spiral CT, and we have a volume acquisition also with the collimation of 1.5 to 3 mm. So obviously we would like to suggest an HRCT instead of a low dose CT. But low dose CT does not mean an inferior CT. It's just in case of few patients that we can suggest a low dose CT.
So is it possible for you to assess, say, airway predominant disease or an emphysema index or an emphysema as clearly as you would do in, say, a narrow collimated CT or HRCT?
HRCT is preferable, but yes, we can pick up even on low dose CT.
Would you prefer CT densitometry over HRCT if you want to quantify the emphysema?
Yes, that's
so ultimately it's CT densitometry over HRCT over low dose CT.
Low dose CT.
So CT densitometry is one of the softwares that you use in a regular HRCT.
Yes, it's not always available at all the centers. So
obviously,
definitely
it's
HRCT is
whichever center wants to
thing.
procure it. That's right.
My next question is to Dr. Parvez Kaul. Does including symptoms in COPD severity overdiagnose the severity?
I think your 2023 guidelines have actually increased the confusion, and you've seen that how confused the gold people are as well. I would say yes, because we are moving from symptom-based diagnosis to spirometry-based diagnosis to imaging help diagnosis to stage 0 to stage stage 1, 2, 3, 4 to A, B, C, D, E, C, D to now A, B, E.
Yes.
That's it. So that is a lot of paradigm, I would say a tectonic shift that has happened over the years from 2007 onwards to this time. Yes, I think if you include symptoms and if you include, say, CAT score or MMRC, there is a possibility of overdiagnosing them. However, if you look at the COPD gene study, the ideal is having all the 4 things, that is exposure history, spirometry, symptoms, and imaging. And if you have any of these missing, then you make what we used to call it a probable COPD or a possible COPD. Yes, I would agree with you, probably yes.
So would you, in the group B, the ones that have more symptoms, are we going to overtreat these patients just because it's there in the guidelines that you have to include that also?
That is a fair, in fact, that is one of the limitations that has been put forth as one of the limitations in the group B. There is a possibility that there's too much of emphasis on symptom-based diagnosis, and you might overdiagnose them.
And
but that is what it is.
Maybe there must be some other pathology also along with that COPD. Maybe there is bronchiectasis.
Those have got to be excluded. You have to factor in other things that would contribute to these symptoms, like dyspnea or...
Yes, Dr. Shujan.
Yeah, I think the associated comorbidities will definitely increase the diagnosis if we take into account the symptoms, because GERD itself or patients of obstructive sleep apnea, these patients, these comorbidities will definitely add to the diagnostic yield.
The next question is a genuinely contentious issue. So I'll start off with Ungiradi, and we will move on to the audience. Should we check maximal dilatation or progressive dilatation at initiation of therapy in COPD? What should we aim at? Ungiradi. All the difficult questions are directed towards Ungiradi.
So first of all, I'm a bit not sure of what you're exactly trying to ask me.
That's maximal dilatation means the change in the FEV1 values?
Yes, yes.
Change in FEV1 value?
Yes.
And the other one? Progressive?
Progressive dilatation is where you are expecting that this patient will improve over the over time.
At a single point or during progressive?
Okay, longitudinal.
Yes.
So if that is what is meant, then the first thing that we should try and discuss is how frequently are we doing these spirometries in the patient. So do we need to do it at every visit, or we need to do it at a fixed interval of 6 months to 1 year? So I'll tell you what I do with my patients. Every time the patient comes, because I have the facility of doing tests free of cost, so what I do is when patients come to me, I always do a spirometry and then try and match up with the previous one or the one before that, so that I can give a treatment to my patient which is going to give the patient a maximum, the best lung function that he ever has. So if there is a decrease in FEV1, then I go looking for factors that are causing a deterioration in the lung function. And usually I find one because we do a sputum test also. And in that, the cause of FEV1 being decreased is mostly an uncontrolled inflammation going on in the airways.
So my question is, if we do get an improvement in the FEV1 the next time around, are we going to stick to that dose and be happy with it, or would you, if you see that the symptoms are still persisting, and would you aim at, would you try to be a bit more greedy and want more symptom improvement and FEV1 improvement?
So usually it's not a disconnect. Like if the FEV1 has increased, then the patient's expectoration and cough has also increased. So very rarely I have seen patients who are discordant, and if they are discordant and symptoms persist, then it's probably that the treatment is not adequate. So we need to treat the patient with more of drugs that the patient is responsive to.
Any comments from the audience?
The nature of the disease is such that if you take COPD, the lung functions are going to deteriorate. So to expect that there is a reversible cause or there is an inciting cause as a reduction in the FEV1 when you see somebody in a follow-up process may not always be there. You may not always find an infection or a suboptimum treatment strategy or something, because the nature of the disease is such that you might find the lung functions to have deteriorated after a few months or after a few years.
Yes, but with ultra-LABA combination, there are some patients who do come with improvement in the FEV1.
I think it's a question of in the beginning, and therefore to try and see if that stability is maintained. And when we talk about stability, we're talking about, you know, the range of difference in the FEV1 if you take that as a parameter, rather than a drastic improvement or a drastic drop. So what I'm trying to say is, first, I think we are reading too much into the spirometry. In this whole morning and the afternoon. We're reading too much into that. And that's why I said I wanted to rock the board. That it's just a reflection of an organ in whom the air is going in and out, and we are trying to capture the air at different rates and things like that. To read too much into it is going to be very difficult, and you're going to probably miss the wood for the trees.
I also think that basically we are not, the patient will not have the same clinical status at each presentation. It's very unlikely that what he presented initially, he might have presented with a little bit of symptoms, and when he has come to you, the presentation, the symptoms have gone off, or he doesn't have any infective process at present. So it's difficult to compare whether in terms of reducing the dosage at every repeat spirometry. So it has to be that the patient stability has to also to be taken into concern. I think I'm not able to exactly.
Dr. Parvez?
I think if I want to recount what my practice is, I would not do a spirometry more frequently than at least 6 months. I mean, in between, maybe even more than that, in a patient with chronic obstructive lung disease. And if he is doing well in terms of what Dr. Angira said, in terms of his symptoms, I would be happy. But I must also say that something that we lose sight of, that is addressing the risk factors, addressing the exposures. Those need to be concomitantly and taken care of in parallel. Because once we talk of COPD as preventable or treatable, you don't treat anything. I mean, the only thing is that there is a subset of patients with COPD, specifically in the COPD, the erstwhile COPD stage 1 or stage even 2, who have demonstrated that they have gone to a stage of PRISM or even to enormous spirometry. And how that has happened, nobody has an answer to that. But that probably is because we have taken care of the exposures. That is the probable answer, though nobody knows exactly how it has happened, but it has happened. So we must concurrently take care of the exposures, the risk factors, be it smoking or occupational exposures or whatever they are exposed to. That's one. And second, I'd be happy if the person is doing well in terms of symptoms. I would probably be happy and not intervene too much.
And I think there is a subset of COPD patients who somehow, for some reason, they do develop this late onset Th2 inflammation, where we start seeing the ACO. So probably those are patients, it's worth looking for such patients, isn't it?
Yeah, I mean, you would suspect them on the basis of their symptoms.
Yes.
They would always come up with symptoms, and it is not that you would not suspect them otherwise.
Yes.
They would come up with, yeah.
May I make a comment?
I agree. I mean, you talked about in the morning also once we were conferring about it, but that is a subset which is not recommended as ever. This is something that we have to have data on. And as of now, the current guidelines don't say that we have this subset that has to be looked for in every patient of COPD. Yes, I mean, it's worthwhile looking at their sputa and finding out whether they have a phenotype, eosinophilic phenotype. It's worthwhile, certainly. And because now there is a confusion about steroids and anti-inflammatory drugs as well.
Dr. Anirban Sharkar, he wants to tell us something.
What I wanted to tell, I've already been told by Dr. Kaul actually. See, our aim of COPD treatment mainly is to give the symptomatic relief
and to make the patient exacerbation free as long as possible. So I don't think this follow-up PFT, follow-up spirometry is that much important in the management of COPD. Our aim should be to make the patient mostly symptomatic benefit, to give symptomatic benefit as much as possible, and to make the patient exacerbation free as much as possible. I don't think that is very important, because by definition COPD is a progressive disease. FEV1 will be progressed, will go down and down. If we make us that FEV1 static or just a little bit of worsening, that will be enough, I think.
No, our point is that when the previous question, where we were saying that we would not like to stick to just one spirometry test to diagnose COPD, then we are actually, even amongst us, not just the patients, even for us physicians, we have a glimmer of hope that there might be a subset of patients who may get better. So is it worthwhile to look for those subset of patients? That is basically the crux of the question. Okay, time, yes.
Thank you.
So I'll go on to the next question. How useful are peripheral eosinophils in COPD management decision? This question goes to Dr. Shoikat.
Regarding our patient management in COPD, the peripheral eosinophil level is a very important factor because it is very important to decide whether this set of patient, subset of patient who will receive inhaled corticosteroid or not. As we know that if the eosinophil level is more than 500 per microliter, we call it eosinophilia. But as per GOLD, if it is more than 300 eosinophil per microliter, it should be considered as eosinophilia, and that is to be taken for consideration of inhaled corticosteroid. Now regarding the exacerbation of COPD, if the patient eosinophil level is high, then we can predict that this set of patient, this type of exacerbation will respond to systemic steroid.
No, Shoikat, see, considering the fact that in India, in Indian context, where we see a lot of peripheral eosinophilia, probably this question is...
In Indian context, the exact value of eosinophil, it is not known to me, which should be the cutoff level of eosinophil, so that we can decide this patient will respond to inhaled corticosteroid or not. Because there are so many that eosinophil levels are quite higher than the temperate climates.
So if I may say, I must also add that in order to address the normal eosinophil counts of our patients, normals as well as those who have obstructive airway disease, there is a study which has been completed, led by Dr. Rajadhar, and it is, I think, total around 8 to 10,000 people have been recruited from various centers across the country. So we should be having soon normal eosinophil count distribution of the country available from a reasonably large sample, and I think that will aid us in our further tailoring of our therapy as regards the eosinophils and the inhaled corticosteroid is considered.
The next question, sir, this is for you, Dr. Parvez Kaul. PRISM.
Okay, I talked about it during my talk, and yes, it has been shown that small airway dysfunction has been a concomitant finding in patients with PRISM in a significant percentage of patients as compared to those who had a normal spirometry, and it was small airway dysfunction documented either by spirometry or by FOT or the impulse oscillometry or even by CT. So all the three had an evidence of an increased risk of SAD in patients with PRISM, and those patients who had a small airway dysfunction had an increased chance of progressing, transitioning to COPD and having a higher mortality.
No, my question is, where we are looking at FEV1 by FVC, the forced vital capacity. So you are not taking into consideration the dynamic closure of airways.
That's what
so
I said.
Is SVC?
I think the last slide was one of the concerns that was actually raised by Peter Burney. I said that they say that once you're talking of a forced maneuver, you would rather take a slow maneuver into consideration, and that is the caveat about PRISM. And that's why many people say that this term should be abandoned. But nonetheless, it is present, and it is present very prominently in the current GORD guidelines as well. So yes, this is a caveat with the definition itself, because if you do it on the basis of a forced maneuver, you have a risk of an early airway closure.
The next question will be directed to Dr. Shujan, and then it will. I hope Dr. Arup Haldar, who brought up this question just a while ago, we can possibly bring ourselves to a particular point. Why is inhaled corticosteroid and LAMA combination not yet available?
I haven't found out the exact answer to this, but as far as I think, if we are using just the ICS LAMA in this overlap set of patients having, which might be asthmatic, then I think we are putting them up for exacerbations. That might be the only reason that we are not getting this combination.
But we have the inhaled corticosteroids to take care of that. Take care of the exacerbations. Yes. Arup asked the question and he's probably going to answer it.
So actually, we have more of the beta-2 adrenergic receptor in the lung than of the anticholinergic receptors. That's for bronchodilation, we always need a LABA or a SABA. But for the other parts, parasympathetic innervation of the lung is the main innervation, not the sympathetic. Sympathetic has a very little role to play in the lung, except the receptors which have different functions. These beta-2 adrenergic receptors are there in the lung to remove fluid from the lung,
yes.
not for the bronchodilation. But we use this beta-2 receptor for bronchodilation in asthma or in COPD sometimes. But the glands, many of the muscles, the bronchial smooth muscle cells, and many of the goblet cells, these are being innervated by the parasympathetic innervation. So blocking the parasympathetic innervation will just defer the progression of the disease or not. That was my question. We do not have LAMA ICS because LAMA is not a good bronchodilator. LABA is a good bronchodilator. That's why we do not have a LAMA ICS. LABA ICS we have. But LAMA, whether that can prevent the progression of the airway disease in asthma, that was my question.
Another point I think is because, see, even now GINA 2023 is talking about MART is a very big concept, and that has been there for the past few years, but they are concentrating on the fact that LABA has to be, if you are using LABA, it has to be formoterol, so that you get the reliever effect along with the maintenance. So possibly that effect we might not get we are not getting with the LABA and ICS. So maybe that could be one. But why LABA and ICS is not being used in COPD again? Because they don't want ICS much in COPD. Probably that is why the combination has not been made yet.
So I am not a proponent for combination drugs anyway. So if you need ICS, if you need more of it, if it's in a combination situation, you won't be able to do that. Because if you put up the ICS, you also put up the LABA, LAMA, whatever it is.
Yes.
So I think it's always good to have the drugs separated out, bronchodilator with bronchodilator, and anti-inflammatory in anti-inflammatory.
So Dr. Angira is going against the GORD guidelines. GORD clearly says that combination therapy is better, combo inhaler is better than individual therapies. But nonetheless, it's an individual position a person can take.
If you add LABA to ICS, actually you're decreasing the effective ICS dose. And if you add lama to that, you are still decreasing the ICS growth. So your total.
That's exactly the reason why you should not be using a combination.
That means your effective ICS, that means mometazone, if you consider. If you use it singly, it is 800 is the high dose. Then if you use the LABA, the 400 becomes the high dose. If you use LABA, LAMA and ICS, 200 becomes the high dose.
Yeah. So the reason why combinations should not be used.
So in this plus minus note, we shall end the panel discussion. Thank you so much
I think the success of this panel has been to create more and more confusion.
Thank you.
Thank you so much. With this, we move forward to our next session, that is heart-lung interface with AE and comorbidities. To chair the session, I would like to introduce Dr. Arup Haldar and Dr. Supriya Shorkar. Dr. Haldar is associated as a prominent consultant with Institute of Sleep Sciences and Woodlands Hospital, Kolkata. His areas of prominent interests are sleep medicine and pulmonary critical care. He has several publications in indexed national and international journals to his credit. I welcome you, sir.
Thank you.
Our second chairperson is Dr. Supriya Shorkar. He is a professor and head pulmonary medicine, College of Medicine and Sagar Datta Hospital, Kolkata. He has published more than 50 articles in national and international journals. He is an eminent speaker in various state and national level conferences and has published book about 101 cases in respiratory medicine. I welcome you, sir, and request you to kindly proceed over the session. Thank you.
Good afternoon. Our first speaker is Dr. Dharmesh Patel, and his topic is heart and lung disease interface. Dr. Patel you can start. Hello. Let me introduce Dr. Dharmesh Patel is MD and Diploma in National Board, practicing respiratory consultant as Baroda, Gujarat, and a visiting consultant at the Rajmore Hospital. His primary interests are pleural disease, but today he will talk on heart-lung interface.
Thank you, Chairperson. And you realize the fact that the major interest is pleural disease, very far off from the airways. So may not be the appropriate person here to talk on airways, but nonetheless, it's topics like this stimulate you every time to try and connect the dots. And thank you, Parthada, for stimulating me into this topic. I was trying to look for a particular logo for this, and then I realized that the romanticism in Partha and the logo which is used for his own institute was probably more appropriate, because the lungs are looking at the heart, and two organs ensconced within a very limited space of the thorax. And yet, they don't talk to each other as much as they should be doing it. Pulmonologists look at cardiologists with suspicion, and the cardiologists ignore the pulmonologists completely. So this is the dilemma in real time where you see this. So it's befitting that we talk about lungs and the heart, and especially airway disease and the heart. And this is something which we day in and day out have been seeing, cases like this, where you have an X-ray chest with florid shadows, you're called upon to see, and once you sort of go through your checklist and you do an echo and you see that there is a hypokinetic left ventricle, septum is quite sort of thickened and hypertrophied, and you see the interaction that the actions of the heart are reflected onto the lungs. Similarly, you would see like a
patient of mine, a gynecologist, who comes with acute breathlessness, with a normal looking X-ray and a thrombus sitting into the right ventricle, clearly suggesting that the impact of what is going on in the heart is felt in the lungs in the form of symptoms. And similarly, you see cases like this: a retired colonel, an ex-smoker with a heavy pack here, breathlessness and reduced tolerance, an X-ray which is showing some sort of lungs which are hyper-expanded, an FV loop showing quite extensive, quite severe airflow obstruction. But in the same time, when these patients don't improve and you look at the echo of these patients, you see that the right ventricle is quite a lot dilated, the septum is bulging into the left ventricle, the tricuspid valve velocities are quite high, suggesting that there is associated pulmonary hypertension. So day in and day out, we've been seeing as pulmonologists the close interaction between our neighbor and ourselves. And this was well established by Eratius, the very celebrated Greek physician living in, at that time, in Asia Minor, which is the current day Turkey. And what he says 2,000 years back, actually, saying that the lungs suffer and the parts which assist in respiration, namely the diaphragm and the thorax, sympathize with them. But if the heart be affected, the patient could not stand out long, for in it is the origin of respiration and of life. So the heart-lung interaction has been demonstrated clearly years and years back. In the modern times, James Hope in the 19th century, English physician who actually has been attributed to be the first modern cardiologist, really, talks in the treatise of his disease of the heart, where he explains asthma as 5. And number 4 is from the organic disease of the heart, is there where the origin of asthma, and then the term cardiac asthma actually comes forward. And he goes on to describe very, very astutely the symptoms and why the pathophysiology of how the heart affects the lungs, with one particular statement saying, hence, want of breath is a necessary consequence of an excess of blood in the lungs. How true, which is this? So we've all clearly looked at the close association that exists between the heart and the lung, and particularly as far as the airway disease is concerned. And Harrison, Tinsley Harrison, the editor of the Harrison's book, which we've always read, talks about the symptoms of cardiac asthma and how gratifying it is to treat that. So the relationship is quite well established, the heart and lung. Only is we've forgotten to look at our neighbors, either the heart or the heart doctors looking at the lungs and seeking each other's help. In the process, there is a fair amount of mismanagement which goes on, which one or two cases might very well elucidate. Let's talk about asthma and heart, the connection between there. And cardiovascular disease is the first thing which impacts. And this is something which is actually an eye-opener, and which to me at least was an eye-opener. The premise is that inflammation plays a key role in atherothrombosis. Asthma has its basis on a milieu of inflammatory processes. Chronic inflammatory diseases like rheumatoid arthritis, SLE, HIV, patients are at increased risk of coronary vascular disease. A potential link, therefore, asthma and coronary artery disease exists. So that's because it has an inflammatory milieu. In order to find out whether this really exists, I have just 2 studies mainly to show you. One was a more recent one, which is from the United States multi-center topic, where they looked at the Framingham offspring study. In other words, the children of patients who participated, or not patients, I would say, of individuals who participated in the Framingham study, along with their relatives. And they looked at a prospective population-based cohort of 3,612 individuals aged 17 to 77 years. And they followed up every 4 years for 3 decades. So there is a fair amount of long follow-up of these patients. Clearly, those at the onset who did not have any cardiovascular disease were eliminated or were not taken into account. And the cumulative incidence of coronary vascular disease, or cardiovascular disease, I would say, with asthma was 36.9% compared to those without it, which was 9.1%. That does not mean that asthma and cardiovascular disease are linked, but then an unadjusted analysis shows a hazard ratio of 1.40 for those asthmatics to develop cardiovascular disease of some sort. And when you adjust this for other factors of cardiovascular disease, which would mean smoking, dyslipidemia, obesity, etc., you still find the association strong, because the hazard ratio is somewhere between 1.28 to 1.31, depending upon the models which you try and use. And this relationship seems strongest among women because of hormonal effects and smokers. So here is a study which tells you that the risk of developing cardiovascular disease in asthmatics is definitely there and high. A more earlier one was a meta-analysis coming from China, where they looked into, their whole idea was to try and see for cardiovascular disease in asthmatic patients. A subsequent study has actually looked at 11 studies, but this one meta-analyzed 7 studies with 12 asthma cohorts from United States, from one of the Scandinavian countries, from Taiwan and Canada. And it was community-based population as well as hospital-based admission. And here too, you find out that the greater risk of coronary artery disease in asthmatic patients was 1.42 folds higher in those with asthma as compared to those without. And in addition, the risk was more profound in females, as it was found in the Framingham offspring study. So these 2 studies are enough, and there are a number of others. For example, the Copenhagen study before that has also shown the risk of developing cardiovascular disease in asthmatics as having a hazard ratio of somewhere around 1.40, which is there. Let's look at the situation in India, though no studies have come out, and we all know this data quite well, that the prevalence rates of coronary artery disease is anywhere in the rural 1.6 to 7.4, and the urban population is from 1 to 13.2, shown by colleagues in 2008. The standardized cardiovascular death rates is highest, with a global average being 235, ours is 272. It strikes younger individuals in our part of the world. Migrant Asian Indians living abroad have higher prevalence, 3 times more than the natives that were there. High burden of risk factors, which are not always and clearly understood or properly investigated in our part of the world. So we have a substantially important non-communicable disease in the form of cardiovascular disease or coronary artery disease in our part of the world. And on the other hand, asthma, which has a crude prevalence of, this was from the, I think the INSERT study, or the global burden of disease study of 1990 to 2016, showing the crude prevalence to be about 2.9%, which I'm sure has increased. The disability adjusted life years is asthma is impacting is 1.3%, and the 42% of the global asthma deaths are attributed to asthma in our part of the world. So on one hand we have cardiovascular diseases, and on the other hand asthma, both of them of substantial importance for us to notice. And if there is a crosstalk between these 2 partners in crime, then you can understand how important it is to study this as a risk factor in patients with cardiovascular disease, rather than, just assuming that these are 2 separate, when this is very important, because in a large number of patients of coronary artery disease, we still don't know the risk factor. So I'm not saying that these studies clearly point to asthma being the causative factor, but it makes you just stand up and say that should we not look at patients of asthma, of coronary artery disease, and try and screen them for patients with associated asthma, because the link is fairly strong and the pathogenesis is not completely obscure, because both is an inflammation disease, and we know through various important studies and well-established studies that inflammation does lead to atherothrombosis, which is there. A second point over there is cardiac dysrhythmias, asthma and the heart. And again, the same premise stands, that inflammation plays a very key role in initiation and progression of atrial fibrillation especially. Elevated systemic inflammatory markers have been shown to be associated with the incidence of atrial fibrillation, along with markers of coagulation, fibrinogen and the D-dimer, which are independent predictors of atrial fibrillation. Asthma subtypes are characterized by an increase in markers of both systemic inflammation and coagulation, and therefore asthma is associated with increased risk of atrial fibrillation. Again, a premise which is very similar to the one we saw that. How do you prove that? And that comes from Tattersall and their colleagues from Wisconsin and other parts of the country, where again they were looking at how asthma leads to dysrhythmias, and they analyzed 6,615 cohort followed for a median of 12.9 years. The majority of them were without asthma, 150 had persistent asthma, and 497 had intermittent asthma. And if you see the number of atrial fibrillation events per 10 years, then for persistent asthma, it has been high, giving a hazard ratio of 1.66 compared to the non-asthmatics or intermittent asthmatics. It doesn't surprise anybody that the persistent asthmatic patients had a high degree of inflammatory markers, namely interleukin-6, CRP, D-dimer, tumor necrosis factor, and fibrinogen. And therefore, the persistent asthma, according to the authors, was associated with a 1.5-fold increase in the risk of incident atrial fibrillation. Greater relative risk of atrial fibrillation, sadly again, had been found more in women. However, this association was not reduced or attenuated by adjustment for baseline inflammatory markers. So in other words, if you try and find out the patients of subset which have high inflammatory markers, by trying somehow by reducing them with therapeutics, does that sort of reduce the risk? This particular study does not show that, which is there. So the asthma-heart interface the take-home messages, if that's what I have to give, is asthmatics are at risk of cardiovascular disease, namely coronary artery disease and arrhythmias. Identifying patients with asthma as a high-risk group that may benefit from targeted interventions to prevent the onset of cardiovascular disease is a contentious issue. Like the whole morning we've been discussing airways and contentious issues. Cardiovascular disease patients should be assessed for asthma as a risk factor, which I strongly feel so after reviewing the literature for this particular talk. In acute exacerbations, we all know and we should diligently monitor the heart and address hypoxemia and hypertension because they are risk factors for ischemia and arrhythmias. And of course, an important factor which is for another day in Kolkata would be anti-asthma drugs and impact on heart, which needs a separate symposium by itself. Before I push on to COPD, which is the second airway disease on the heart, I was pretty much impressed by this particular study, which I think was again a very big eye-opener because not very much keenly interested in airway disease as my forte. Naturally, these things later on in life have been eye-openers to me at this particular age. Nonetheless, as we know, it's not too old to never learn. And this study comes from Don Sin and his colleagues from Vancouver. And they have shown that how reduced lung function and cardiovascular mortality are linked with. So they looked at a cohort of 1,861 patients of the health and nutritional assessment surveys which they carried out on 71 to 75. And out of that 14,407, they looked at 1,861 participants. They divided their FEV1 into 5 quintiles, which is on your table, which is from left to right. The first quintile was 63% predicted, for the fifth quintile, which was 109% predicted. And their follow-up studies showed that 19.1% died during that follow-up period, which was up till 1992. So again, 2 decades of follow-up, which is there. 45% of these deaths were due to cardiovascular disorders. 76.4% of these 45.4% deaths, that is 123 number, were due to ischemic heart disease, and 9.3% were hospitalized with the principal diagnosis of cardiovascular disease. More importantly, what does all that tell you is that in all cases, they reported that the risk of cardiovascular mortality and hospitalization increased with decreasing FEV1, which is there. And that's tables which clearly shows you, and this is the weighted analysis that shows that a quintile of FEV1, we'll see the quintile. Now, the first one with 63% predicted, the relative risk was 3.36. For hospitalization was 1.69. And for mortality from ischemic heart disease in those with lower FEV1 was again high at 5.65 compared to the other quintiles, especially when you compare them with 5. When you adjust this for smoking, because you might feel that, when you adjust this for smoking and other maybe contributing to risk factors, but when you look at the non-smokers, here also the relative risk was high in those with a lower FEV1, namely the first quintile of FEV1, which was less than 63% or 63% mean, was 2.68 as far as cardiovascular mortality was concerned. Hospitalization was 1.71, again compared to the fifth quintile, which had normal or supernormal FEV1. And similarly, the mortality from ischemic heart disease was higher. And therefore, the conclusion is that reduced FEV1 is a marker for future cardiovascular morbidity and mortality. The relationship between reduced FEV1 and mortality from IHD is particularly striking as you've looked at the data. And even among lifetime non-smokers, this relationship held, indicated that reduced lung functions independent of smoking is a significant factor or marker for cardiovascular mortality. So here we have a study which shows that if you have your low FEV1s, your chances of cardiovascular mortality is high, which is there. Again, the possible causal mechanisms, lung diseases lead to airway inflammation, especially the obstructive airway disease. The interstitial lung disease is also something to be included, but because of the nature of the issue today which we are discussing, airways and heart, we take obstructive airway disease into account, and airway inflammation, namely the CRP, fibrinogen, interleukin-6, etc. They are local airway inflammation which we know incite and propagate systemic inflammation, and systemic inflammation has been found and well established cause as a risk factor for plaque genesis, progression and rupture. So that's where the low FEV1 or reduced lung functions and cardiovascular mortality link is trying to be established. Now add all this to somebody who is smoking, as in a COPD, and you'll clearly see that it's a double whammy for these patients. You start off with, even with when you adjust for other risk factors, that low FEV1s make you more prone to cardiovascular mortality. Now if you have people smoking, which further impacts your FEV1, which has clearly been shown by Richard Petto and Fletcher, by the famous Fletcher graph or curve, which says that patients with COPD who continue to smoke have dramatic drops, progression drops of their FEV1s. As I said, you already have that those who don't smoke and have low FEV1s can be affected from cardiovascular risk. When you smoke, you further add to this, which is there. I mean, that's intuitive, and it's clear that therefore these patients of COPD are at more and higher chance of cardiovascular risks and mortality. Is this a probabilistic chance? In other words, you have a common risk factor, smoking, which is affecting COPDs and affecting the heart as well. Or is it a reciprocal influence, one neighbor influencing the other? And which we've seen with pulmonary arterial hypertension in COPDs, or cor pulmonale, or just left ventricular failures, which is there. Just one slide, and not going into too much of data which is out there in literature in abundance, but 10 to 40% of COPD patients have left ventricular dysfunction. 42% of first admissions of COPD is due to actually cardiovascular disease and not due to respiratory failures. Anthonyson and the colleagues again from Canada have shown that. Cardiovascular disease is the main cause of death in mild to moderate COPD, not respiratory failure. And conversely, 10 to 32% of congestive heart failure patients have COPD, and mortality rates are higher in these patients with those who have concomitant COPD, 21%, 9%. And therefore, there is a submission that screening is vital in patients of congestive heart failure or for lung functions, and vice versa for those with COPD to screen them for echoes. We pulmonologists are very good at asking our patients to do, COPD patients to do echo for a number of reasons. But are our cardiologist colleagues ever asking their patients to do spirometries? That's the question we need to ask. And I think it's important. Let's look at this case one, just one case to enunciate my point. CP is a 71-year-old electrician with a heavy pack year smoking history, comes with syncope and breathlessness and normal examination features, and as is all the cases, any patients of breathlessness is booted to the cardio department which is there. Misfortunes, he lands up with the cardiologists in the cardiac institute, has a slightly abnormal ECG. That's enough for a cardiologist to shift him to the cath lab, do a coronary angiogram and say there's a double vessel disease, stent the patient, and then put him on cardiac drugs, which is the usual jazz of antiplatelets and statins and beta blockers and some diuretic as well in this case. The next 3 months he continues to be breathless with reduced effort tolerance. Just 2 minutes. Chest x-ray and antibiotics, a repeat angiogram to say that are my stents blocked is being done. Continue the cardiac drugs. Emergency admission, he lands up with hypoxemia and extensive V's. He comes to the A&E, given oxygen, furosemide, salbutamol nebulization. Normal bloods are done, a chest x-ray which is normal, and echo is the first thing which people do jump to, and it shows that there's not much change compared to the echo which he had done 3 or 4 months back. And it is continuing breathlessness and need for oxygen at 3 liters per minute, and that's where the COVID has taught us that investigating breathlessness, first you do a scan rather than, and my radiology colleague behind, by the way, enjoyed your talk. You've set a very bad precedent, I'll tell you. Well, it's not your fault, but at any given moment, don't do sputum in a case of suspected TB. Do a CT just to get pretty pictures of a cavity sticking around. You know, that's the state of affairs, what's happening, at least in my part of the state which I come from. And I can't see Gujarat being totally different from Bengal, really. So that's the sad part. So HRCT is telling us emphysema here, you know, in this patient, rather than a lung function which should have been done over the last 5 months or so. So this is his lung functions, basically showing severe obstructive airway disease, which was driving his symptoms. Maybe he has got cardiovascular disease. I don't deny the fact. I mean, you have to accept the coronary angiogram saying double vessel disease. But the point is, when your patient does not sort of improve, you need to actually take time out, step back and see. You can't have a tunnel vision. Neither we as pulmonologists nor my cardiologist colleagues. You have to step back and see, is there anything else happening? Because of the common risk factor of smoking, it is quite normal for an individual to have COPD and cardiac failure both. Underuse of spirometry, stroke echocardiography, addressing common risk factors which we fail. Of course, then the impact of COPD and heart disease will be approached to drugs, beta blockers and its impact on the lungs, beta-2 agonists and their impact on the heart, theophylline, inhaled steroids, long-acting beta-2 agonists, underutilization of cardiopulmonary rehab and palliative care in these patients, and finally, cardiopulmonary unit establishing where colleagues can work together to approach airway disease along with the heart disease, which I think is the need of the hour. I'll leave behind with just last 1 or 2 slides, mainly cor pulmonale, which is better now defined as pulmonary heart disease, which is the group 3 PAH, and the pathophysiology is the same. Remodeling over time in COPDs, will leave the ILD part apart because of the airway emphasis, leads to remodeling of airways, lung parenchyma and vessels, and that leads to pulmonary hypertension. However, the prevalence of mild pulmonary hypertension is of severe pulmonary hypertension in COPD patients is only about 5 to 10%, well established, more through right heart catheterization, as people say, with a PVR of more than 5 Woods unit. However, the transthoracic echo is our day-to-day test to try and look at this. Non-severe pulmonary hypertension is common in severe COPDs, but can impact symptoms and recurrent admission. 1 to 5% of the cases have severe PH. They have worse outcomes. Low oxygen, carbon dioxide, and low DLCO are the risk factors for developing this. Overlapping symptoms in patients post-diagnostic challenge. Right axis deviation on ECG and elevated BNP are markers or red flags which make you think that your patient of COPD will have right ventricular dysfunction. And echo using a composite score of tricuspid regurgitation velocity, right atrial area, LV-RV ratio, etc. is preferred to diagnose this state and in a stable state rather than an acute state. What can be treated with COPD with PAH is optimize the pharmacotherapy, oxygen, in some cases non-invasive ventilation and pulmonary rehab. is what sort of things. Could we use vasodilators, whereas the new guidelines just come out last year mentions that there is absence of large randomized trials. The evidence is insufficient to support the general knee-jerk reaction of adding sildenafil to our patients of COPD with CORP. It's a no as the data stands right now. Last but not the least, heart airway disease and drug impact, which, as I said, Partha, should be one of the most important issues to be discussed at some other time or the next meeting. How does drugs of each disease impact the other one? We know beta-2 agonist can cause arrhythmias, angina, heart failure, and sudden death. What's the action? Probably use long-acting muscarinic agents. Theophylline causing tachycardia and arrhythmias. Therapeutic drug monitoring may be important, or use an alternative. Azithromycin, often used in COPD, increases the QTc interval, so monitoring is going to be important, and review its use if it's causing that. Beta blockers is the big thing to be discussed. It leads to asthma exacerbations. Cardiologists refrain from using it. The New England Journal of Medicine study of 2019 has shown that it is relatively safe with a very small percentage. However, it looks stupid on a prescription to give beta-2 agonist and beta-2 blocker in the same thing. When you ask yourself, what are you doing? You're stimulating the receptors at one end, and on the other you're blocking the receptors. So anyway, but that's using cardioselective beta blockers is not a problem. AC inhibitors and the effect on the cough, and aspirin can lead to asthma attacks. You know the Samter's triad, in which case my cardiologist, when I asked him, he was confounded. He said, no, what else can I use? And then he came up with, you could use either desensitization or you could use clopidogrel or tacrolimus. So I think I'll end this this is the airway lung interface. We could discuss a lot of things, but maybe the panelists, etc. can. And before I end, this is well in advance, but next year, we're looking forward to our fourth respiratory update from our society in Vadodara, which is in Goa. And please stand by for further exciting news. It's the fourth update every 5 years, which we have. Thank you once again, Parthada. Thank you, chairpersons. And should we take any questions now? Or? Okay, thanks.
Thank you, Dr. Patil, for giving a nice lecture and describing the catastrophe of super specialization. Now I have some points for the PGTs. Now the so-called the pericardial PET is not due to involvement of the pericardium, because pericardium is not sensitive to pain, and it is due to adjacent pleural disease. Number 2 point, though anatomically our sweetheart is in between two dark lungs, but physiologically our lungs are compressed by two sides of the ventricle. So when there is left ventricular failure, there is lung congestion, pulmonary edema. On the other hand, when there is a lung disease, we can have the pulmonary hypertension, and that leads to right ventricular hypertrophy. But it has been found. One question I want to ask here for the PGTs, that in high altitude dwellers, there may be moderate to severe pulmonary hypertension, but they usually do not have right ventricular failure. On the other hand, in COPD, in mild to moderate pulmonary hypertension, there may be right ventricular failure. What is the cause? Any PGT can answer this question? They are not hearing actually. It is the additional factor, that is the hypercapnia or infection, that is required. On the other hand, the most important cause of left ventricular disease in COPD is the age and the smoking habits. But even if those confounding factors are excluded, then also there is 2 to 3 times more risk of left ventricular disease in COPD, and that may be due to systemic manifestation. With this comment, I invite our second speaker, Dr. Aditya Jindal. He is DNB Medicine, DM Pulmonology, and Critical Medicine. His area of expertise are advanced interventional pulmonology, critical care, sleep disorder. His experience in multiple interventional pulmonology procedures, and he has extensive experience in critical care And with this comment, I invite Dr. Aditya Jindal to talk on.
We have a tea counter open outside. So if anybody interested can go outside, have tea, or take the cup inside and listen to the lecture.
And his topic is exacerbation of OAD. With this, please.
Thank you, Chairperson, and I would like to thank Dr. Parthasarathi Bhattacharya for inviting me here to this beautiful city, and this is the first time I'm coming here. So my talk is on obstructive airway disease, exacerbations of obstructive airway disease. And we have been discussing since the morning about obstructive airway disease. The two major sort of conditions constituting obstructive airway disease are COPD and asthma. And there is a considerable overlap between these two conditions. And the current thinking or maybe future thinking would be like whether these are actually two separate diseases, or are they a single disease with variable spectrum, with a large spectrum, or are they all part of a single sort of constellation or syndromic of OAD or obstructive airway diseases. So we'll just discuss a little and specifically focus on exacerbations of both of these diseases, and what are the similarities, what are the differences, and how to go about treating them, and are there any new approaches to treating exacerbations. So there is as I've already mentioned, diagnostic confusion between COPD and asthma is quite common in our routine day-to-day practice. Unless there are very clearly defined phenotypes or very clearly defined patients, there are lots and lots of patients in whom it is very difficult to make the diagnosis. The so-called ACOS or the asthma COPD overlap syndrome complicates the picture further, and it was introduced a few years ago by the guidelines and then withdrawn abruptly within 2 to 3 years. And we see these kind of patients in our clinical practice. A modern approach to phenotypes and endotypes also creates a lot of confusion. And it is, as I've already mentioned, it is important to decide whether the difference between these two diseases is valid or not, especially in the acute settings of exacerbation. So what is COPD? COPD is a heterogeneous lung condition characterized by chronic respiratory symptoms like dyspnea, cough, expectoration, and exacerbations due to abnormalities of the airways and or alveoli that causes persistent, often progressive airflow obstruction. While asthma is a heterogeneous disease, usually characterized by chronic airway inflammation, it is also defined by the history of respiratory symptoms such as wheeze, shortness of breath, chest tightness, and cough that vary in time and intensity, which is very important. So the airway obstruction and the symptoms are variable in both time and in intensity as compared to COPD. Nowadays, both of these diseases are considered to be syndromes and have different sort of constellations of maybe diseases under them, different phenotypes under them. And here we can see the different pathophysiological sort of genesis of this. So asthma is basically atopic or allergic disease, and is you can see the main components involved are the eosinophils, the mast cells, which produce all the different inflammatory markers, and this is predominantly a Th2 type of inflammation, which is sort of increased by cigarette smoking, pollution, biomass fuels, etc. While the COPD patients is basically a neutrophilic or macrophage predominant inflammation or a Th1 type of inflammation. In cases of allergic sensitization, there might be some overlap when this is called the ACOS or the asthma COPD overlap syndrome. Early on, maybe in the 1960s or so, there were two main hypotheses regarding the origins of these diseases, the Dutch hypothesis and the British hypothesis. And the Dutch hypothesis was postulated first, and it said that maybe asthma and COPD have a mutual origin, and subsequent expression of each disease is explained by the individual variable factors, like maybe different genetic factors or environmental factors. And contrary to this, there was a British hypothesis which postulated separate origins for asthma and COPD, and both of these said both of these are driven by their own individual genetic traits, inflammatory profile, and separate treatments. So over the period of time, in the last 60 years or so, the British hypothesis had generally won out. But now we'll see that since the introduction of ACOS, the Dutch or the Netherlands is making a comeback. And you can see that there is a significant overlap between COPD and asthma. So asthma is mainly eosinophilic inflammation, airway hyperresponsiveness, a good bronchodilatory response, and steroid responsive, while COPD patient is neutrophils, no airway hyperresponsiveness as such, less bronchodilatory response, and limited steroid response. But there is a significant sort of overlap in which we can call them by a variety of names: ACOS, remodeled asthma, VZ bronchitis, a lot of things. So now we'll go on to the acute exacerbations. So people, clinicians who are practicing know that the person who's coming to the emergency with breathlessness, once you diagnose that it is a sort of an exacerbation, rule out other causes, then the treatment more or less is quite similar in both asthma and COPD. So acute exacerbations of COPD are defined as transient periods of increased symptoms of dyspnea, sputum purulence, and sputum volume. And there may be other minor symptoms like nasal blockage, discharge, wheezing, sore throat, etc., limited physical activity. And the exacerbations are triggered mainly by infections with respiratory viruses, commonly the respiratory syncytial virus, adenoviruses, and recently the COVID virus. And some bacteria, especially the mycoplasma and Haemophilus, are also quite common in causing exacerbations. And of course, presence of pollution directly leads to airway epithelial damage and can cause inflammation and exacerbation as such. So this leads to inflamed COPD airway, then again increases the inflammation, which causes all the bronchoconstriction, formation of edema and intraluminal mucus, causing, as already discussed in the morning, dynamic airway inflammation, hyperinflation, exacerbation symptoms, etc., etc. And along with this, in COPD, we have, as Dr. Dharmesh has also already mentioned, the systemic inflammation, which causes cardiovascular mortality, and most of these patients, or a large number of these patients, will present in the cardiology department as compared to the pulmonary medicine departments. In general, COPD exacerbations are more severe as compared to asthma. Each COPD exacerbation is associated with decline in lung function, quality of life, and increased mortality. And here you can see these are two graphs. So on the left side, you can see this is the time period after the first severe exacerbation. This is only the rate of severe exacerbation, while the B part is telling the rate of severe exacerbations or death per 10,000 patients. So this was data from almost 70,000 patients. And you can see that the rate of exacerbation significantly increases after the first severe exacerbation, while the rate of, if we include the death also, then it increases within the next 2 years. So these patients are at high chances to die in the next 2 years. And the rate of exacerbation also increases. While if we look at asthma as such, so asthma is the exacerbation is generally interaction of the host factors and environmental factors. So the environmental factors again are the same respiratory viruses, allergens or bacteria, while the host factors include maybe more sort of predominant in this case as compared to COPD, like allergic sensitization, different immunity levels, eosinophils, risk factors such as increased BMI, higher severity of the disease, and more SABA use has been associated with frequent exacerbation, and all of this leads to airway inflammation and asthma sort of exacerbation. So generally speaking, the asthma exacerbations are milder as compared to COPD exacerbations, but they're much more common. And some studies have questionably labeled that 40 to 70% of patients might have exacerbations each year. So, but the thing is that might be difficult to differentiate these patients from improper control of asthma rather than exacerbation. So it is sort of a difficult job, but still these are mild exacerbations are much more common in asthma. And here we can see some data from the US about the incidence rates of acute exacerbation. So the upper part is the exacerbation of COPD, Well, this is severe asthma. So nobody has done a detailed analysis on mild exacerbations of asthma, but severe exacerbations of COPD are much more common. But the mortality of both of these diseases is significant. And in recent years, the COPD exacerbation mortality is coming down while the asthma mortality is going up. So I don't know what the reason for this would be. It's not really mentioned in the article, but this is the data from at least sort of 20 years of ICUs. So is there any point in actually treating these patients separately, especially in the exacerbation stage? And I think most people would agree with me that whenever we see a patient in the emergency, standard treatment is start nebulization, give steroid, give antibiotic, and if give oxygen, all those things, and if needed, shift to the ICU. So this is generally called a unified airway approach to the management of exacerbations. And so the exacerbations can be divided generally into 3 types: the mild, moderate, and severe. And this is basically for clinical purposes. Even the GOLD guidelines also sort of classify exacerbations based on this, but exacerbations can be classified based on the response to treatment, based on clinical presentation, based on some laboratory parameters, and sometimes they are classified after the exacerbation has been treated. So as a basic clinical sort of thing, you can just check the you can say the vital signs, maybe if a CRP if you have, or oxygen saturation, and these things are generally enough to treat all these patients. So I'll not be discussing too much about this because this is all routine clinical practice. This is just to give an overview of and emphasize the point that both of these patients or both of these diseases are treated in a very similar manner. So if it's mild exacerbation, generally the patient can speak in full sentences, have a normal mental status, and no accessory or mild accessory muscle use. While in cases of moderate, there will be increase in the heart rate, increase in the respiratory rate, oxygen saturation will be above 90%, the mental status will be normal, but there will be more symptoms. The visual analog scale for dyspnea will be higher, there will be more wheezing, there will be more accessory muscle use. While in cases of severe exacerbations, there will be significant increases in the heart rate, respiratory rate, low oxygen saturation, and if you can do an ABG, which shows a significant hypoxia or increase in the carbon dioxide with low pH, then it is definitely a severe exacerbation. So just a general protocol, some of these things are done here, some of these things are not followed in our country, but just to show that in a severe exacerbation, generally in all cases where there is hypoxia, we give oxygen, give nebulization with salbutamol or ipratropium to control the acute episode, give steroids, give antibiotics. Magnesium has been used in these patients both in asthma and in COPD for refractive wheezing. Non-invasive ventilation again is used in both of these diseases, more commonly in COPD I would say as compared to asthma, but still used in both of these diseases, and maybe other sort of intravenous drugs. And then if it is not sort of working, go forward with intubation and mechanical ventilation, go for ECMO. So this is again a chart to show the therapeutic options at the different stages of patient management. And so all of this is just to sort of show that this is sort of a unified approach to treatment, and especially at this stage of disease, there is no point in differentiating whether there is a COPD or asthma. So I have just a very short presentation, last few slides about newer approaches to sort of managing these patients or to the approach to asthma and COPD. So there is a concept about treatable traits. So what is a treatable trait? Treatable trait has 3 major sort of points. It is clinically relevant, it is identifiable and measurable, and it is treatable. So clinically relevant means that it requires the trait to be clinically important and associated with specific disease outcomes. Identifiable and measurable, that it should have a biomarker which can be checked and it can be sort of followed up, and it gives a good, if you follow the biomarker, then it gives a good response to treatment. And of course it should be treatable. So this should be effectively treated, and the effect should be measurable in randomly controlled trials. And Of course, some of these things are not available. They are research opportunities for future, for the future. I think Dr. Dharmesh was talking about Dr. William Hope, so I think everybody has heard the name of Dr. William Osler, who was one of the most famous early physicians and has given rise to given many quotes in medical literature. And the general approach we follow today, the sort of disease approach is called the Oslerian approach. And the advantages, there are certain advantages, and for almost 100 years or so, this approach has been followed by all doctors across the world. So the advantages is that it leads to making simpler definition patient groups. For example, COPD, asthma, bronchiectasis. So this is a very simple definition. It is easy to use in some studies and useful syndrome identification. So you can easily label that this patient has COPD, this patient has asthma, or this patient has bronchiectasis or ILD or whatever. But disadvantages are that it may fail to provide optimal care because all of these diseases include a vast variation of phenotypes and endotypes, and all of these patients may not respond similarly to similar treatments. There is no appreciation of common patterns of disease. For example, if you look at all the diseases I'm mentioning, asthma, COPD, bronchiectasis, bronchiolitis, all of these are obstructive airway diseases. So it's it's not necessary sometimes to treat them differently. So then sometimes it inhibits research progress. So the new or the proposed treatable traits approach does not make any diagnostic assumptions. There is a precise definition of the traits. It's based on disease mechanisms, and there is an expected larger therapeutic response. For example, in asthma, if we look at severe asthma endotypes, where we have all these IgE levels, all different biomarker based. So that is basically a treatable traits approach to diseases, and it stimulates research and helps in identification of criteria for future RCTs. But it also has some disadvantages, that it is nowadays only partially based on evidence, and there is uncertainty among all the practitioners and difficulty in acceptance of all these things. So this is just some food for thought for the future. And some of these treatable traits in patients with asthma, COPD overlap would be some of these are pulmonary treatable traits like airflow limitation, airflow smooth muscle contraction or hypertrophy, eosinophilic airway inflammation, severe and frequent exacerbations. So some of these traits can be treated specifically with different medicines. And there are some extrapulmonary treatable traits and some behavior lifestyle risk factors which are also treatable traits. So this approach can also be there to treating as compared to the traditional approach in treating these kind of patients. So I think in summary, I would say that the original Dutch versus British hypothesis debate is still ongoing. There are lots of similarities and also differences between COPD, asthma, and exacerbations. Exacerbations are much more common in COPD as compared to asthma, and much more severe, though mild exacerbations might be more in asthma. Approach to treatment in exacerbations broadly follows a similar path, and new approaches to treatment may yield better results in the future. Thank you.
Thank you, Dr. Jindal, for your nice talk. And the pre-exacerbation and post-exacerbation lung is not same. This has to be remembered because the FEV1 will decrease definitely. There will be increased risks of viral infection. There will be increased colonization by bacteria, and all factors responsible for the chance of exacerbations, further exacerbations. Now I invite Dr. Hariharan and for his talk on comorbidities in OAD. Now he is MBBS, MD in pulmonary medicine, DNB, DM, pulmonary and critical medicine. He is an interventional pulmonologist and a renowned teacher. His primary area of interest are severe asthma, endobronchial ultrasonogram, and pleural disease, He has more than 25 publications. Presently, he is an assistant professor pulmonary medicine in AIIMS, Jodhpur. Dr. Hariharan, please.
Thank you, sir, for the kind introduction. And at the outset, I would like to thank Dr. Parthasarathi, sir, for giving me this opportunity. So, without quickly wasting time, I have been asked to speak about comorbidities of obstructive airway disease. So, I will begin my talk with this case of a 56-year-old gentleman who was a heavy smoker with progressive breathlessness and intermittent cough, with also history of intermittent swelling over the feet. He is obese on examination and has a reduced intensity of breath sounds. This is his X-ray on the right, showing probably hyperinflation, and a PFT showing obstruction. His labs were within normal limit. So this is the standard prescription the resident had written, that he was advised smoking cessation and a LABA-LAMA combination. Although the patient had partial relief in breathlessness, but still it persisted. So what could be the unaddressed issues? There could be an underlying cardiac issue contributing to his breathlessness. Is there a risk for this patient to develop lung cancer? Was coexisting anxiety and depression evaluated? Should we evaluate for osteoporosis? Because he's obese, are there any risk factors or are there any metabolic derangement which are prevalent? And in view of his obesity, could he be also having an underlying sleep disorder? So that brings us to the talk, that is obstructive airway disease and comorbidities. So the problem statement. So if you look at the obstructive airway disease, they are complex diseases with multi-system involvement, and comorbid conditions, they could arise independently or they may be causally related, like for example smoking. And if you look at some of the studies, the presence of clinically relevant comorbidity, at least one of them, was seen in as high as 80% of the patients of COPD, with more than 3 being seen in almost half of the patients. These influence the prognosis and contribute to exacerbations. When you now talk about COPD and asthma, which are the major chunk of obstructive airway disease which we deal, these are some of the common comorbidities which could be affecting both the pulmonary and extrapulmonary systems, like asthma, lung cancer, skeletal muscle problems like osteoporosis, sarcopenia, frailty, neurological problems like depression, anxiety, cognitive impairment, cerebrovascular disease and sleep apnea, and the cardiac problems which has been extensively covered by Dr. Dharmesh Patel, like atherosclerosis and heart failure and myocardial infarction. There could be metabolic problems like obesity, diabetes, and peripheral arterial disease. If you look at asthma, if you see allergic rhinitis, obesity, and obstructive sleep apnea contribute the major chunk of comorbid conditions affecting asthma to the tune of almost 45%, with others being chronic rhinosinusitis, vocal cord dysfunction, and anxiety and depression. On the contrary, it is very surprising if you see this paper, the referrals for comorbidities has been dismally low, with as much as only 50% of the patients who have some other comorbid conditions are being referred to specialists. This is especially relevant in case of anxiety and depression. Now, how does this impact the control of the underlying asthma or underlying obstructive airway disease? So this paper from Hungary evaluated more than 12,000 patients, and the asthmatics were divided into well-controlled or partially controlled as one group and uncontrolled as the others. And when they looked at the comorbid conditions, they found that almost all of the comorbid conditions had a statistically significant impact on the asthma control, and the odds of having a worse outcome or an uncontrolled asthma was more, especially if you had a concomitant COPD, ischemic heart disease, or a cerebrovascular event. This is same. These patients also frequently have more exacerbations, worse ACT and AQLQ scores. As a result of this, they also lead to more exacerbation. So in this cohort of a Korean population, which analyzed more than 12,500 patients, they found that asthma, lung cancer, and heart failure were independently associated with a higher risk of exacerbations amongst patients of COPD. So now coming to some common comorbid conditions. So this has been part of this has been discussed in the earlier, but I would just quickly tell you that the predominant or the core of a problem is systemic inflammation in COPD, which leads to two important problems, which are the components of Virchow's triad. Number one, endothelial dysfunction, and number two is the procoagulant state. What these two do is that by altering the, altering the endothelial function, they lead to events like venous thromboembolism, pulmonary hypertension, heart failure, and systemic hypertension. And by the procoagulant state, they lead to obviously coronary artery disease and thrombosis. And mind you, it has been shown in studies that the clot which forms due to this inflamed state is resistant to fibrinolysis as well. Now, just a quick word on heart failure. You all know heart failure is common in COPD, but the most important entity which has been frequently neglected is heart failure with preserved ejection fraction, also referred to as diastolic dysfunction sometimes, which is typically characterized by elevated left ventricular filling pressures. This may be associated with signs and symptoms of heart failure or may not be. And sometimes you might have a raised natriuretic peptide levels. And this heart failure with preserved ejection fraction is commonly caused by COPD, obesity, diabetes, which are also the comorbid conditions commonly coexisting with COPD. And this heart failure with preserved ejection fraction manifests with the same symptoms as COPD, making the diagnosis difficult and often delaying treatment. The diagnosis is simple. It requires a baseline echo in all patients to look for the elevated diastolic pressures, and symptoms. The main treatment is to control the underlying disease with drugs like dapagliflozin and empagliflozin showing promising results in some recent trials. Sleep apnea, you all know, is very commonly associated both with COPD, where it is referred to as the overlap syndrome, also with asthma. Now, if you look at the common pathophysiology or the common mechanisms or the contributing causes in COPD, the rostral fluid shift, especially with heart failure, the skeletal muscle weakness, and use of corticosteroids leads to or is a risk factor for OSA, whereas in asthma, the upper airway manifestations like rhinitis and obesity, by altering the caliber of the upper airway, tend to develop or are risk factors for OSA. And this intermittent hypoxia, which OSA adds to in the diagnosis of COPD, leads to again endothelial dysfunction and increased risk of cardiac comorbid conditions. In case of asthma, it is very interesting to note that the obstructive sleep apnea and the hypoxia which sets in shifts the TH2 balance towards TH1 and makes the treatment of asthma by inhaled corticosteroids difficult. It sort of introduces a resistance to inhaled corticosteroids by this remodeling effect on the airways. So the evaluation of sleep apnea is standard, along with standard treatment with CPAP and BiPAP. But I just want to emphasize this fact that patients of COPD overlap with OSA. When they are treated with CPAP, the mortality is better if in comparison to those who are not treated with CPAP. So this again makes it essential to evaluate for this comorbid condition. Lung cancer, as you know, both COPD and lung cancer share the common risk factor, and the relationship again here is bidirectional. COPD independently increases the risk of lung cancer, and the other way, patients of lung cancer have a higher incidence or a higher prevalence of having an airflow limitation. This also influences the prognosis. The study has shown that the survival outcomes in patients of lung cancer who have COPD are worse at the end of 3 years in comparison to those who do not have COPD. So currently there are no clear-cut guidelines of screening for lung cancer in diagnosed COPD patients. We have the guidelines for healthy smokers, but whether we need to do a baseline CT for all COPD patients is an issue. I would just like to emphasize this case which we encountered recently. This patient presented to us with COPD. As a rule, we or my institute, we do a baseline CT for all COPD patients. This nodule was incidentally picked up, and on biopsy it turned out to be a squamous cell carcinoma. The other issue is oncologists do not screen for airflow limitation by spirometry in lung cancer patients. It has been shown, so this important paper looked at the post-hoc analysis of the patients recruited in the National Lung Cancer Screening Trial. Amongst 10,000 patients who were recruited in this study, they found that patients who had gold group 3 or 4 were, when they underwent a CT screening, that they were not having an added advantage of reduction in mortality, emphasizing the fact that airflow limitation can negate the effect of CT screening, and hence it is important to manage them. So my take would be that I would do a baseline CT in all of these patients and also evaluate for airflow limitation, because by giving them bronchodilators, I can also make them eligible for chemotherapy by improving their functional status. Osteoporosis, again a common. Pooled analysis of studies have shown that the incidence is as high as 38%, with underweight and low BMI and sarcopenia being independent predictors for development of osteoporosis. An important parameter is the cycle of frailty, that is a vicious cycle where COPD patients, due to their inflammation, have sarcopenia, which leads to reduction in physical activity, which in turn again perpetuates sarcopenia. So this is the vicious cycle of frailty which has to be broken. Anxiety and depression are independently associated with increased risks of exacerbation of hospitalization, and they most importantly affect the symptom perception. So your patient might be complaining of dyspnea, whereas the real reason why he is having this could be his anxiety and depression. So there is no point in escalating his treatment for obstructive airway disease, because in reality you have to treat the anxiety and depression. Similar is the case for metabolic syndrome. I would just skip these slides in interest of time. Now, bronchiectasis is another important comorbidity, with prevalence being around 54%. And a systematic review has found that patients with COPD who have bronchiectasis have greater sputum production, are more prone to exacerbations, have poor lung functions, higher rates of colonization with Pseudomonas, and increased mortality. To complete the list, we have some comorbidities like anemia, polycythemia, cognitive impairment, which is very important, gastroesophageal reflux, pulmonary hypertension. Lastly, infection could be an independent comorbidity, because we have seen that patients of COPD who have TB have early onset of COPD diagnosis, a lower FEV1 value, and they are less responsive to treatment. So how do we assess these comorbidities? So traditionally, you have the Charlson's comorbidity index, but this has been criticized to be not very specific for COPD. It has been for a generalized hospital setting. Two recent indices have been developed. One is the COAT index, which is basically assessing the specific impact of comorbidities on mortality. And other is the COMCOLD index, which basically collectively assesses the impact of comorbidities on patients' health status. So these are two comorbidities indices which you could use and sort of prognosticate your patient. We also have questionnaires in asthma for evaluating the sinonasal disease, the vocal cord dysfunction, the Berlin questionnaire and the GERD questionnaire. But the problem of these questionnaires is that it has been found in studies that it takes on an average of 40 minutes if we have to fill each of these questionnaires, thereby not suitable in a busy OPD, especially in a public sector setting, I would say. So, does treating comorbidities actually help? So, this has been answered in this study from 2017, if I'm not mistaken, where they did a multidimensional assessment and addressed the comorbidities, and they found that treating or addressing comorbidities increased the control of asthma, as was seen in the ACT and AQLQ score, and also reduced the number of exacerbations. So my take would be we need to have a holistic assessment and management of comorbidities. We could use screening questionnaires in context of a clinical history, and we can make tailor-made evaluation and referrals, especially for diseases like upper airways in asthma, the vocal cord dysfunction, GERD, obesity, and importantly, psychiatric assessment. In COPD, I would go a step further, have a baseline cardiac assessment in the form of ECG and echo, and a referral in case necessary. Do a DEXA scan, because we have to rule out osteoporosis, and God forbid if the patient has a vertebral fracture, that might further decrease his physical mobility and contribute to sarcopenia. I would do a baseline CT scan of the chest in all my COPD patients, A, to look for lung cancer, 2, to look for underlying bronchiectasis. And the emphasis has to be on rehabilitation and maintaining mobility, because otherwise sarcopenia sets in. So to conclude, I would just say that comorbidities are frequently associated, and the figures are surprisingly very high, as to the tune of 80%. They should not be ignored and should be a part of standard management. The unaddressed comorbidities can hamper disease control, and it is important that we rule out these comorbidities before blindly escalating the treatment for underlying COPD or asthma. If unaddressed, they can lead to worsening of lung functions and increase the mortality, and hence multidisciplinary management is a key to well-being. Thank you.
Thank you, Dr. Hariharan, for your nice talk. I invite Dr. Parthasarathi Bhattacharya for his talk on. I do not need any introduction, so I invite Partha to start his talk.
Thank you, Dr. Supriya. Basically, I requested people to go for a tea to charge them and come back. Where is the talk? Questions? But on the contrary, what I found that many people have left, and what I feel that naturally, with time the attention is down and the energy is also down. So what we decided of late to change the program and shift to my talk. After that there will be common question answer come panel discussions. So my talk is precision treatment of obstructive airway diseases, a new proposal. So actually regarding precision medicine, the discussion evolved after this publication of a book by national academics in 2011. It was motivated by explosion of molecular data on human, particularly data associated with individual patients, and the sense that these are large, as yet untapped opportunities to use this data to improve health outcomes. So we understand by now, more than 12 years after the publication of this particular book, that endophenotyping and phenotyping is very important. Phenotypes, here you can take a airway disease where you have different phenotypes. Diagnostic phenotype like blue bloaters, pink puffers, comorbidity phenotypes, susceptibility to precipitants, then exacerbation phenotype, functional capacity-wise phenotype, severity phenotypes, and others. These are descriptive, and these phenotypes are something which we can make out. If you consider etiopathology, you may be needing some investigation. Even then you can tell them these are phenotypes, like COPD, smoker phenotype, non-smoker phenotype, allergic phenotype and non-allergic phenotypes of asthma. But once you go to pathophysiology, then you can reach endotypes where you are discussing the mechanisms. Here it is for asthma, Th2 high or Th2 low endotypes. And if you go on, you may have a lot of different endotypes. There is a huge list. I thought that I will also be able to present the different endotypes and phenotypes to you through a talk by my colleague, Dr. Jindal, but because the mood, I thought that I should drop it out. But if you're interested, he is ready. So what is the most important issue to me as a clinician? How to treat my patient the best way possible. So I have to know what are the treatment opportunities. So I have bronchodilators and anti-inflammatory drugs at my hand. There are two classes of bronchodilators, beta-2 agonist and anti-muscarinic agents. And anti-inflammatory, luckily, mostly the inhaled corticosteroids. Now take an imaginary scene where you have got known susceptibility for beta-2 agonist, for anti-muscarinic agents, and also for steroids. And if you make a matrix on that, you can find that you can have different type of combinations treatment for different combinations and permutations. And finally, if you just see them through a filter, it will be reaching 5 pharmacotherapeutic phenotypes of obstructive airway diseases. The first phenotype is universally responsive phenotypes, where you have responsiveness to inhaled corticosteroids, inhaled beta-2 agonist, and inhaled anti-muscarinic agents. There may be universally non-responsive pharmacophenotypes, and in between there are 3 ICS responsive mixed phenotypes, non-ICS responsive phenotypes, and only ICS responsive phenotypes. So basically you can have 5 phenotypes on pharmacoresponsiveness. This is about different, okay. So how do I approach to determine reversibility to a bronchodilator? I have to know, I have shown you the matrix, reversibility to bronchodilators. There are 2 classes of bronchodilators. I know about the salbutamol reversibility test. It's a proactive assessment, but we don't have any anti-muscarinic agent reversibility test. So I'm not going into details of the preamble. This anti-muscarinic agent reversibility test is important, but this is not possible because of the pharmacokinetic properties of the available anti-muscarinic agents, because their onset of action is delayed, and because the peak action is much delayed. If you take a patient, give a puff of inhaler and let him wait for 2 hours, he won't. It is very difficult for office practice. But we can do it if we combine with salbutamol, a anti-muscarinic agent, and give it for 7 days, 10 days, 2 weeks, 4 weeks, 6 weeks, and have a comparative limb where you are giving salbutamol alone. You can see that this combination may be working better. So that's a way of prospectively on long term evaluating the responsiveness to a drug. So what we found, what we thought as a new anti-muscarinic agent glycopyrronium came to market, it has got a short onset of action, 3 to 5 minutes, and by half an hour it has got a reasonable good level of function. So we thought, let us see what is the effect of glycopyrronium if I use it as for reversibility. So this was a plan. This is open absolutely. First we tried standard salbutamol's reversibility in spirometry. After 20 minutes, we give the glycopyrronium inhalation, 50 micrograms through dry powder, and then after 30 minutes repeated the spirometry. And this is my first experience where I have, the left hand side you can see a patient, there is severe obstruction with 320 ml and 22% reversibility. This is salbutamol. So there is, of course, we'll stamp it as a case of asthma. But once I had given glycopyrronium to him, there is extra 640 ml of reversibility. So had I have not given glycopyrronium, I would have been blind absolutely about the sensitivity or reversibility to glycopyrronium. So this patient has got altogether about 1 liter of reversibility, huge change, huge. And that means a huge change in the quality of life if I can give 2 drugs to him. So naturally the patient was very happy. Even after the test he was feeling happy. He said, I'm feeling very free. Well, this is my second patient where I have given salbutamol. There is 240 ml and 28% reversibility. That too we tend to diagnose as asthma. And once I had given GP, there's hardly any extra reversibility. There's no extra reversibility. So we have seen 2 patients, one asthma with more glycopyrronium reversibility, much more glycopyrronium reversibility than salbutamol, and a case of COPD where there is no glycopyrronium reversibility. We always say the LAMA is the treatment of choice. Here LAMA is a failure, but LABA is working. LABA means long-acting bronchodilator, beta-2 agonist. So then we just compile our experience, had a few dozens of patients and seen how the lung function behaves. You can see a improvement of lung function after salbutamol reversibility by adding glycopyrronium. So you can see the dots, and the conclusion says that testing the reversibility with glycopyrronium appears feasible and safe in obstructive airway diseases. It induced add-on reversibility once used after standard salbutamol reversibility. The observation demands further evaluations. Okay. So we had next thought, what is the effect of the agent depending upon the order of use of salbutamol? We have given glycopyrronium after salbutamol. There can be spillover effect of salbutamol that influence the glycopyrronium reversibility. So we just reversed the order, glycopyrronium followed by salbutamol. And this is the result where on FEV1 we can see that the 2 groups had similar reversibility 72 versus 60 ml. And it was almost near similar, but salbutamol had a little better reversibility. This is again published in this month's Lung India. Here you can see two colors. Red is the change from glycopyrronium and blue is the change from salbutamol. Here you can see the number of patients below. There are 4 groups of patients. One group of patients is essentially salbutamol sensitive. So they are beta-2 agonist sensitive. The number is 48. And the amount is 155 ml. Mind, these are all COPD patients. Second group is glycopyrronium sensitive. The number is almost same, but reversibility is little less. The third group is both sensitive, a small number, where you can have a total of nearly 300 ml of reversibility, which is quite big. And these patients are definitely going to do better if you combine the 2 drugs. And the fourth group, which number is the biggest, 70, which is roughly 44%, 40% plus, where the total reversibility is less than 100 ml, total combined reversibility. So there is a group of patients which are not responsive to any of these bronchodilators. There is a group which is responsive to both, and there are groups that either responsive more to beta-2 agonist or more to glycopyrronium. So this is one way we can preemptively or proactively understand the responsiveness to the bronchodilators for further treatment. Well, with that, we also did a salbutamol add-on reversibility to see whether the spillover effect is there. It is not there. So I'm not presenting that. Let me see what is the anti-inflammatory responsiveness. So anti-inflammatory drug is steroid, and we have taken all kinds of OAD patients, and we have looked for peripheral eosinophil count, we have looked for FeNO, and we have looked for high IgE level. If all three are present, it is sure type 2. If 2 are present, this is our assumption. It is likely positive, likely Th2 high. If all 3 are negative, it is a negative, absolute negative Th2 or low. And if any 2 of them are negative, it is likely negative. So there are 4 subgroups again here, and we have added our informations from our salbutamol reversibility and glycopyrronium reversibility to it to make a composite matrix to see which drug to be given to a particular patient. So the algorithm is like that: you diagnose an obstructive airway disease, you do reversibility test with salbutamol and glycopyrronium, then you determine the inflammatory phenotype, then plan and determine the treatment, and then go for additional considerations. This is a matrix where if purely Th2 high can be treated well with corticosteroids. Ideally, non-Th2 high OAD is likely difficult. One needs to rule out infections. This is a dictum. Bronchodilators have to be chosen according to the sensitivity. Suppose in the upper row, the patient has got sheer non-Th2 high, no ICS, and there is no glycopyrronium or no salbutamol reversibility. This is the most difficult patient. These patients will go for special clinic or referral. It should be further evaluated for the reason of refractoriness. He or she may be treated with long-term antibiotic or bronchial thermoplasty or kind of thing which is very different than our common treatment. What I mean to say, the common basket of treatment is not applicable to this patient. On the contrary, the bottom row where the eosinophil count is high, the FeNO is high, IgE is high, and we have got responsiveness to salbutamol and glycopyrronium, you can understand he will demand 3 drugs and the patient will do far, far better. So I can preemptively understand which patient requires what treatment, and I can also possibly predict his treatment response. This all requires further research. So this is the algorithm of the new precision proposed by me. I propose that we should be simplified. We should simplify the whole thing. Now it's been enough. It is enough of complication. Asthma itself is a big heterogeneous disease. COPD is heterogeneous. ACO is a lump, and nobody has defined ACO so clearly. We should forget all that. We should take them all together under one umbrella of obstructive airway disease. And this is umbrella approach, and this approach with the philosophy that it's the first priority is the airflow obstruction, not the name. Second is do not discriminate. Same policy and strategy to all essentially for primary pharmacotherapy. For complications, for exacerbations, for comorbidities, you may have different treatment according to the requirement. But the primary therapy should be non-discriminatory. It will be based on your matrix that I have shown. Avoid exposure to allergens and noxious agents, that is common to all, and to the best possible treatment beyond conventional pharmacotherapy. Do not see the color or character. Treat as per one's susceptibility to response to the agents as bronchodilators or inhaled corticosteroids. So with this, I wish to end. This is just a proposal. We have been doing glycopyrronium reversibility test for quite some time, from 2016 onwards, and we have got only 2 publications. By now, I have got quite a bit of data with me. I have done more than 1,000 patients, more than 1,000 patients, and what I find that this is really a good test to follow. I will try to do my best to publish them one by one, and there are so many new issues cropped up while doing that. I feel they are very important. I presented my experience in a very small way so that you too get interested and can try it. We have been trying for a pan-India sort of validation of my observation. So I'd be very happy if anybody wishes to join me. So this is what is my presentation. Thank you very much for your kind attention.
So, good afternoon. So now we'll start the panel discussion for heart-lung interactions, exacerbations, and comorbidities. May I request upon the speakers here on the stage, Dr. Dharmesh Patel, Dr. Ajit Jindal, and Dr. Hariharan, and Dr. Somnath Mitra, please. Dr. Ajay Sharkar, not present, I think. Lieutenant Colonel Dr. Shafeen Babu. Dr. Anirban Sharkar. Dr. Shelly Shamim. Dr. Arunabh Datta Chaudhary. And Sir, Dr. Supriyo Sharkar. He has left, okay. So you all are panelists, as audience is 10 now. So let me start with Dr. Dharmesh Patel. Oh, he's not there, okay. So let me start. To any of the panelists, you can answer. So when we will suspect presence of cardiac comorbidity in COPD? Should we suspect in all COPD patients, or whenever they are presenting with certain symptoms, we suspect a cardiac comorbidity? Any one of you can answer. Yeah.
Basically, I will suspect, for me actually, I will suspect a cardiac comorbidity, especially if a patient is too breathless for his FEV1. If that FEV1 is not explaining his severity of breathlessness, one. Another, some other signs and symptoms, say pedal swelling, orthopnea, too much of breathlessness on exertion, chest pain, this sort of things. So one is symptom-wise, and other is lung function-wise. If his shortness of breath is not explainable by that grade of FEV1, I will suspect some other disease, especially cardiac disease, or some presence of other disease, other signs, especially pedal edema, arrhythmia, or some other symptoms like chest pain. Then I will suspect possibly some cardiac disease.
So FEV1 have some caveats. So there are few patients who have a combined restrictive and obstructive disease. Their FEV1 will be less, and they will have both the disease. So out of proportion FEV1, both in the upper terms or in the lower term, we have to think about the cardiac diseases. Very true. So my next question goes to Dr. Jindal.
There are two small points.
Oh yeah, please.
Yeah.
on at the very look of the patient, if you find patient has got likely cor pulmonal, so you have to investigate that on clinical examination. Second, if the patient has got disproportionate heart rate, I think that patient should also be investigated. And third, if you look at the chest X-ray and you find that, say for a COPD, the cardiac silhouette is not non-tubular, to be frank. So you may have you should investigate concomitant cardiac problem. And I think the disproportionate fatigability, getting out of breath, walking a few steps, or getting wheeze on walking or working, that is also another indication for, this is to me.
So I think also we have to look at the risk factors.
Yes.
So what risk factors patient present with to us, because if the patient is a smoker or obese, so he may have a concomitant cardiac disease also. So my next question goes to Dr. Jindal. So you have told about the exacerbations. So how mild exacerbations, repeated mild exacerbations are actually different from severe exacerbation in terms of long-term lung function or decline in the FEV1?
Yeah. So, in fact, any exacerbation is associated with the decrease in both FEV1, quality of life, and even with sort of long-term mortality and exercise capacity. Of course, severe exacerbations are associated with more immediate and medium-term, you can say, sort of complications and effects. But even mild exacerbations, if they keep on occurring, again, they will lead to, in the long term, definitely they will lead to a decrease in all these parameters.
Right. So do you think, in our previous session there was a discussion about the mild asthma. Do you think this mild asthma term is misnomer? Actually, mild asthma can have a severe exacerbations, and we are undermining the disease by calling it mild. Should it be called a stage 1 asthma?
Yeah, I think that was a sort of what I was trying to say about when I mentioned about treatable traits. If you go into the phenotypes, as Dr. Parthasarathi was mentioning, there is a specific phenotype of asthma called frequent exacerbators, in which they might not have sort of day-to-day symptoms which are very severe, which are mild, but they might have severe exacerbations. So they generally present with exacerbations. So asthma is a very heterogeneous disease, and it is very difficult to categorize and sort of encapsulate all the variability of this disease into simple, mild, moderate, and severe. So I think the best would be to go for good control and poorly controlled and not controlled. That is, I think, the best way of going about it.
Rightly said. So, Dr. Hariharan, this question goes to you. What are the dreadful comorbidities in asthma and COPD, and why?
I think if you ask about most dreadful, I would say they are the cardiovascular complications, namely myocardial infarction, angina, and heart failure. And
in asthma or COPD? In
COPD predominantly. If you look at the statistics point of view, in asthma, if you look at the number of comorbidities, they are mainly the upper airway ones, like the allergic rhinosinusitis, coexisting obstructive sleep apnea. And why these are important is that, if you don't recognize these comorbidities, you would feel that the disease is uncontrolled and you would just escalate the therapy rather than addressing them. So in reality, this is what is causing problem and not is airway disease. So that is why it is important to address, because they can lead to sudden cardiac deaths, especially in COPD. If you don't address the cardiac comorbidities, they can lead to sudden cardiac death, which will be most dreadful for the patient.
So asthma patients have several comorbidities.
Yeah, most of them, but if you look statistically, I feel that the
So what is the most dreadful to you? So whenever an asthma patient comes with that comorbidity, you are very much anxious that this patient may land up with some problem.
I think even if you look at data, I would still say cardiac comorbidities are also common in asthma patients.
But in young asthma patients? Do you fear obesity with asthma? Because obesity comes with many of the comorbidities.
Yeah, true. I would agree with you on that point, because obese patients may have coexisting sleep apnea as well. So, if uncorrected sleep apnea can lead to multiple comorbidities, including sudden death and cerebrovascular events in young patients, like uncontrolled hypertension as well. So, you're right, I would...
metabolic disorders as well.
Exactly.
Dr. Mitra, do you want to add anything here?
Actually, on a lighter note, comorbidity was, known to everyone, irrespective of doctors, when the COVID strike by center or state. Every patient of senior citizen like us, we were dying of comorbidity, not of COVID. But here, actually, comorbidity plays an important part. The only comorbidity which pulmonologists actually do not look into it, one is OSA, and another is, psychological these things.
Yes.
Depression, anxiety, is very much.
Having an independent risk factor with mortality.
Right.
So even that has to be actively screened, and we have to take a multi-modality referral to correctly diagnose them.
So the psychiatric problem...
In COPD, patient usually they are aged. As such, they were active. Maybe they were smokers. When they get old, they do not like to do some exercises like that. So gain weight. So they are aging, gaining weight. They are getting breathless. And so they are getting more depressed and anxious. And this is a vicious cycle. That's the problem.
Dr. Shafin Babu, do you want to add anything?
In COPD patients, apart from cor pulmonale, I'll be cautious when the patient is having bronchiectasis.
Right.
Because in my practice, I have seen the emerging phenotype COPD bronchiectasis overlap. So that patient generally go into type 2 respiratory failure. And it's very difficult to bring them back. And if NIV fails, then we counsel the family, and if they still insist, we put them in invasive mechanical ventilation, and it's very difficult to bring them out from the ventilator.
Right.
So that is one thing, apart from cardiac bronchiectasis.
So COPD with bronchiectasis, they have frequent exacerbations, infective exacerbations, and they have a rapid FEV1 decline and higher mortality than plain COPD alone. Rightly said. So the next question goes to Dr. Jindal again. So you have told about the exacerbations. Do you think the exacerbator phenotypes in both asthma and COPD are different from the usual clinical phenotypes or the symptomatic phenotypes? Because there is a dissociation. We have seen that those patients who exacerbate, they exacerbate more in terms of exacerbations, then they may not have that much of symptoms. Do you think they are two different phenotypes? Actually, we should have an exacerbator phenotype separately from the disease phenotype.
I think you are right in this, that and we have already talked a little about this, that there are separate phenotypes in both asthma and COPD which are frequent exacerbators, and they may or may not have daily symptoms. So I think that is a point, and that is well taken.
Right. So regarding asthma COPD overlap, we do not have that term at present, because in 2021 Gold they have abolished the term. So now it comes with different names. Asthma, that is in the recent Gold, that is called COPD-A. So the name is changing, but the disease is remaining the same. So what is the current status of asthma COPD or the COPD with eosinophilia? So does it fit into the echo? Or it is a different treatable trait actually? We should not actually distinguish between these namings. So anyone of you can answer.
What I feel is the entity is still there, and now nobody is able to differentiate whether it's an overlap syndrome or what. Because if it is asthma COPD thing, ICS will be giving if the eosinophils are more than 300. If it is simple asthma also, ICS we have to give. So now Gold is also not able to say that we are stopping this terminology or we are continuing. But Gold has withdrawn the overlap.
Right.
Farooq, can I make a comment on this? See, I think it's an artificial and completely manufactured term, this asthma COPD overlap. Asthma and COPD, or obstructive airways disease, have always had an overlap. If you look at that Venn diagram from 50 years, you have a group of patients who have features of both. And that's very simple to answer, you know. So you could have an asthmatic who smokes or who's exposed to environmental pollution and obviously develops features of COPD. And you could have a COPD patient, as per the Dutch hypothesis, who basically has characteristics of asthma. So I think this term really is a very manufactured term. And I'm very happy that Gold has discarded this term because it conveys no additional information in terms of pathogenesis or in terms of treatment approach also. If you have a patient who has got good reversibility, eosinophilia, you treat that patient as an asthmatic, you know. You don't treat that patient as a COPD patient.
Right. So do you think COPD with eosinophilia and echo are different?
I don't know. First of all, I don't think there's something called echo, you know. I don't
Or asthma COPD overlap and COPD with eosinophilia are two different subsets?
We don't know. I don't think there
Because
are...
why I'm asking you the question, the biologics doesn't work in COPD with eosinophilia.
Well, there is some evidence now that some of the biologics may have a role in and in fact, I think there is a trial underway looking at the
That is the
role of-
tezepelumab you are telling about, not the tezepelumab which acts on the hierarchy and the pathogenesis.
No, even
go for the IL-5 inhibitors.
No, I think there was a
there
trial
is
in
also-
NEJM which is just published on Thursday.
There is
where
also
they
a
have investigated
IL-5
mepolizumab.
Yeah.
So they have just, the NEJM has published an article this Thursday where they have looked at the role of mepolizumab in COPD patients with high eosinophilia. So there is evidence of, which is upcoming with regards to the biologics in COPD as well, especially the eosinophilic variety. And with regards to echo, I would just like to add that it's very confusing term. Second, there is no consensus amongst different societies as well as to what is echo. The Japanese define it slightly differently. They have included FeNO, they have included radiological criteria, whereas if you look at the other consensus definition which came out in 2016, they have slightly different varieties. So what I would go ahead is like what Dr. Partha sir said in his lecture, rather to phenotype and take it under a common umbrella and treat them as a whatever is the phenotype, rather than we need to have a separate different entity for treatment. That is what I feel.
If you look at the GINA guidelines, they have still not let go of this term.
Yes.
So in the latest terms, the latest guidelines, they're still mentioning ACOS or ACO being treated.
So shifting from this topic, so we often get a COPD patient with pulmonary hypertension and diastolic dysfunction. So what is your take on that? So it is whether the pulmonary hypertension due to diastolic dysfunction or due to COPD. So how do you differentiate that, or how you manage those patients? Anyone from the panel can, or anyone from the audience also can answer.
See, I think pulmonary hypertension in COPD is multifactorial, So if you look at heart-lung interaction, so if you have huge swings in intrapleural pressure in patients with, say, for example, severe COPD in exacerbation, there will be problems with cardiac function. And this has been there for years and years. It's nothing new. We've just coined terms to describe that. Now, whether you will treat the pulmonary hypertension in COPD with drugs meant for pulmonary arterial hypertension is another question. If you leave the drugs aside, you just have to treat patients with for COPD, optimize their treatment and optimize their heart treatment separately. So obviously you treat the patient as a whole. Now, whether you give them pulmonary hypertension drugs, I think we don't really have the data. That does not mean it may not work in everyone. So if you look at the phenotypes or endotypes of COPD, there is one phenotype endotype in which pulmonary hypertension is supposedly much more common. And maybe they have pulmonary vascular inflammation, these types. They have systemic inflammatory response. So I think it's too premature to answer this question in terms of a specific therapy apart from optimizing their heart condition and optimizing their COPD. Practically, I don't think what else you can do.
So what I'm trying to just tell that COPD is associated with some pulmonary hypertension. On top of that, patient has a diastolic dysfunction. So that produced another set of pulmonary hypertension. Is it additive, synergistic, or what?
I don't have an answer to that, you know. I mean, I suppose it will be additive, you know. If you have heart failure with reduced ejection fraction, it will be additive.
I'm going to speak my experience. I do a little bit of research on pulmonary hypertension. I've done roughly 60, 70 patients right heart catheterization. And to my utter surprise, almost all patients have got high PCWP. They've got high left ventricular filling pressure. I have asked my cardiologist colleague to look for direct left ventricular catheterization to see the left ventricular end diastolic pressure. He said, is it also high in few patients? So class 2 pH is common in COPD, if not universal. I do not know exactly. Second, if you have to see pH from COPD, you have to calculate the pulmonary pressure. You have to look for the pulmonary vascular resistance.
Right.
That is dividing pulmonary pressure by cardiac output. So you need to have a catheterizations. You can isolate these as class 3 pH. This segment, to my mind, is coming smaller. Regarding treating these patients, well, guidelines say nothing. No to sildenafil. But we have of late published 2 small papers. One is a concept of treating these patients, and second is endorsement to it. A very small number of right heart catheterizations. But I think it is possible. We have to make way out. Some patients require treatment, not all. But before treating these patients, we have to give optimal COPD treatment. We have to give long-term oxygen therapy and very good rehabilitation.
Right. So do you consider the pulmonary artery pressure in treating the pulmonary hypertension in COPD? That means most of the COPD patients have a pulmonary hypertension which is moderate. PASP or pulmonary artery mean pressure doesn't cross 40. So if you have 65.
What I do is, first I look at the quality of life. Second, I look at the desaturation max in 2 chair tests. This is a small test, we have evolved it. If a patient gets dyspneic and shows desaturation after that, and if that desaturation is more than 3%, that is very important. So first thing is diagnose pH. Second is look for WHO classification of pulmonary hypertension. It is only class 3, class 4, WHO. And if these patients, despite all treatment, have got desaturation on movement, so they are possibly the candidates who should be treated with vasodilator, that too with a very cautious note.
Okay.
I totally agree with Parth sir. What I follow is the class 1 patient with some pedal edema, which they are not observing. I will start them on diuretics. There is no guidelines, but I am just telling you what I am following. From class 2 onwards, depending on the quality of the life, depending on the WHO functional class, I will think to add some monotherapy. 3 also, sometimes monotherapy or dual therapy. 4, generally they are on LTOT and all, so double drug I give.
Right.
And there is no guidelines.
So that may be related to a group 2 pulmonary hypertension because of the left heart disease patient is having pulmonary hypertension. No. So the guideline doesn't give us permission to use sildenafil in this, but in certain subgroups have. Okay. Yes. So this is my last question to the panel. So sometimes we get a patient with acute exacerbation of breathlessness in emergency, and it is it becomes very difficult to distinguish between whether it's a COPD exacerbation or a heart failure exacerbation or a mixture of that. So how do you approach these cases? Anyone can answer. Or anyone from the audience can also give some ideas about that. Heart failure, AECOPD, or a mixture. So how a junior resident should approach these cases to differentiate between the different causes?
First of all, the resident doctor should take a clear history. Then after that, the clinical examination. So in heart failure oblique COPD exacerbation, the auscultatory findings may be same. Like you will get wheeze. Like cardiac asthma, you are getting wheeze. And in COPD patients, sometimes there may not be the pietal edema, raised JVP and all, like a cardiac patient. So like that, the resident doctor should clinically, historically, have to distinguish and treat the patient.
So do you think ultrasound here comes into rescue?
Yeah, that will help. In all settings, there will not be ultrasound in the emergency department.
Yes. So if you have an ultrasound, if you just can show the B lines, or if you can show pleural effusion, so that comes into rescue. Or many times we have a mixture of both. So before I finish, as the previous speakers, sessions of the previous speakers also present here, so let me ask one last question. So in COPD, we do not give steroid to all patients. We give steroid to a certain subset of patients when they fulfill certain criteria. But why in exacerbation of COPD we give steroid to all patients? So do we have to separate them based on the peripheral eosinophil count, do you think?
Absolutely. There are studies which are coming up that in fact we will be, if you are using steroid to all patients of COPD, we will be doing injustice and we will more steroid.
COPD exacerbation.
Exacerbation also. There are studies, few studies, which have shown that if we can just see the peripheral eosinophilia and give steroid, we will do more justice to them by using less steroid also.
Right, because most of the...
But again, there are studies before that. This is a recent study which I have done before that, which has shown that all benefits of COPD exacerbation if we are giving steroid and early steroid.
So most of the exacerbations are due to either viral or bacterial infections. So there is no justification of giving steroid blanketly. Yes.
May I have a comment? I think that the reason behind giving steroid in acute exacerbation of COPD is that in acute exacerbation of COPD, whatever be the basic cause, be it infection, be it whatever, the inflammation is the primary thing. So because as of now, we don't have a better anti-inflammatory drug other than steroid, that is why probably we are bound to use this steroid as an anti-inflammatory drug. If we get a specific anti-neutrophil inflammation specific anti-inflammatory drug, neutrophil specific inflammation, which will be acting against neutrophilic inflammation, then we will probably be safer to use that agent. But till then, we are bound to use this anti-inflammatory drug, and we don't have any other choice other than steroid.
So do not have a biomarker for that. Yes.
Which you sometimes use for pure asthmatic, sometimes you use very high doses. So maybe 40 milligrams of prednisolone equivalent should suffice for most patients with exacerbation of COPD, rather than sometimes going into 40 milligrams sometimes.
Also the duration.
Duration is for up to 2 weeks you can use actually.
So as a question of thinking came, I use low dose steroid. I have a idea in my mind. I thought of looking at it scientifically. I couldn't. That COPD is a frail state, and there could be relative hypocorticism at the time of exacerbation because it's a stress where you require more steroid. So if you give a little bit of steroid, it might be helping the patient. From that viewpoint, I support the concept. I also agree to what Dr. Agarwal has just said. But as such, the question is correct. If we don't have evidence, steroid should not be given. This area should be investigated, whether this patient has got relative hypocorticism at the time of exacerbations.
If the audience have any questions to any of the panelists, we can discuss, or then we'll finish the session here. Thank you, sir. Please conclude.
There's one more small thing is left with. In fact, in our plan, we had a session for discussion on research issues. I see all my friends, colleagues have come from all along. We have got 3 scientists, eminent scientists, sitting from the morning till now. And I understand with how much vigor they listen to us, and they want to support us. So this is the morning started with a comment by Dr. Souvik Mukherjee. If China can do, why can't we? And if we stop it here, I think we should be failing. So I invite Dr. Supriya Sarkar to have a sort of general panel amongst this panelists and the audience regarding how to go ahead for research, because this is very important today. As a nation, we are faltering. And if you look at 10-20 years back, I remember I listened to a Chinese expert. I was laughing, in fact. I was thinking, this guy is talking so poor, data is not that good. I didn't find anything special in them, and I found that his English was also not at all impressive. And after 20 years, if you open any international journal, you find Chinese people have grabbed it. In fact, they have been dominating. They have money power, they have willpower, they have political power, everything they are applying on science and research, and they're developing like anything in a phenomenal speed, where we are lacking, perhaps because we are not giving adequate attention to science and technology development. And I think we should not stop here. We should have a little discussion, and then we'll just close it.
I think, Partho,
yes,
there should be here, turn left. Yeah. Philosophy and dedication, these things are dignity.
Yeah, that's definitely. Once you have will, things will come gradually. I think so. Shukri, please.
As Parthoda is talking, India is lacking in research activity, particularly in the eastern part of India, in Bengal. This is because of lack of resource, lack of ecosystem, and lack of mindset. So I invite Dr. Rajat Banerjee to tell something how we can improve our research activity.
Thank you for giving me this opportunity, but it is difficult to explain everything. But as a university researcher, as I can say that we are really running out of resources for a long time. And as I am discussing with Dr. Many of our colleagues, that nowadays, if you look at US universities, all universities have a medical center. So everybody has a medical center attached to the university, and there is no need to do paperwork, to do a move, to do a research with another. So in our Calcutta University, all the medical center once is under Calcutta University. Then it is separated around 1990, so they become separate part. So if you want to do research together now, you have to do a lot of homework, paperwork, and that is go to somewhere, and then it will take so long that you probably forgot what you have thought of at the beginning to do research. So it is sometimes we are actually lost in paperwork and everything, and so it is very. But still, if we want to do something meaningful, we need to do a collaboration between the basic research and the applied research, because we are not sure about how to get the patient data and how to apply them from our perspective. But if we do not attend most of the symposium done by the physicians, also the vice versa is also true. So there is no connection between us actually. There is no common bridge. So first we have to establish that before doing anything meaningful, I think. Thank you so much.
Okay. Dr. Souvik Mukherjee, your comment?
Yeah, See, I come from an institute that is solely, the mantra is accelerating genomics for health. And when this institute was built up by Professor Partho Majumdar, the main idea was to bridge the gap between clinicians and researchers. And how we have accelerated in it is that we have signed a MOU, of course, with IPGMER, and we have opened a center called Genetic Service Unit on the premises of the PG Polyclinic building, where free of cost we give them the screening of the monogenic kind of diseases. And by that we build a biobank. And they also give us a space to do research. And the clinicians, so when I joined the institute, I first went to the clinicians in IPGMER OPD and went to them and told, we are NIBMG, we are in Kalyani, but now we have come to PG Polyclinic. And if you have any problem, because you see many patients, if you have any problem, please come to us. We would like to give a genomic solution to it. Most of them came up, and then we shaped up some research proposals, and not all of them, few of them, and submitted to DBT and DST. Now, there is another bottleneck. If we only depend on government fundings for research, it's not going to help. It is high time we have to go to private funders, philanthropists and private funders. I have a decade-long collaboration with Unilever, and it is a fantastic collaboration with skin diseases. And we are also opening up some collaborations with other private partners. So the genomic science we do, also they need youngsters, young researchers. When they come up, when they come with MSCs, they don't know anything of genomics, because genomics is very understudied. So we also have started, like Dr. Rajat Banerjee told, we also have started PhD, integrated PhD programs. They come after BSc, we train them in MSc, and the last year of MSc is in research. And while we plan our research problems, we also collaborate with the clinicians jointly to ask the question, because questions has to be asked by you. We will give them solutions by multi-omics or some other kinds of thing. Until that gap is, answered, we will not be able to do anything. But I think we are now, there are several institutes in Eastern India also, and there is no lack of resources. Please come to NIBMG, please come to Bose Institute. There is no lack of resources, lack of willpower is there, lack of coalescence is there. So as a young scientist, when I started my work, I found that if two persons in India are in the same, they are working in the same disease, they don't see each other's face. If that is going to happen, we are not going to collaborate. You see the Nature publications, they have 30 authors. They don't struggle who will be corresponding, who will be first. They think about the research. That thing we have to come up with. Otherwise, resources are plenty, we are not using them. Thank you.
Thank you. Dr. Sudipta Saha.
I think, so Vik and Rajat has already addressed many of the things. So what I can say is that we have a platform to do science at Bose Institute. We have a human resources. We have multiple GRF, SRFs. We have 5 to 7 PhD students for each faculties. So what we don't have. So what we don't have is maybe the funding, and we don't have the samples, clinical samples, and also the questions from the clinical onset. So we, as a basic scientist, we have a question from our own perspectives, but for clinical perspective, question is also important. So that question can be addressed by the physician only. So me and Angiradi, we published at least 1 or 2 papers, and we also published with Parthasarathi Bhattacharya a few papers. But still, what we feel is that we need a questions which should have a translational aspects, which we can translate into, suppose, a discovery, which you can give an example that it's not only paper, we need to give some product. So that attitude has to come from both, from the basic scientist as well as from the clinical aspects, from the physician's aspects as well.
So we have to sit together. Parthoda, please arrange for that. I invite Dr. Jindal to tell something about the research activity, how we can improve it.
Yeah, thank you, sir. I think personally speaking, I don't have too much experience in research as such, but I think the basic problem, as I see it from a private sector, because I'm working in my own clinic, is a lack of cooperation among all of us. And there are silos, government departments separate, unwillingness to work with the private sector. Private sector people are more busy with doing the clinical work, and maybe don't have time for research, and maybe don't want to do research. So I think multi-centric studies like they're already going on in the medical fraternity. For example, if you look at the Embark study, which looked at bronchiectasis, I think it was a multi-centric study, and it has been published in the European Respiratory Journal, I think, or Lancet, I'm not really sure. And I was part of that study, and we collected data from, I think, maybe 30 centers from all over India, and it was collated in a single center, and then it was published, and it is a very good study, and it has become the landmark study for bronchiectasis in India. And same way, if you look at the ILD registry, which was done from Jaipur by Dr. Virender Singh, and again they collected a huge amount of data on ILDs. And work has already been started in this, but I think collaboration with basic sciences is something, because most of the basic sciences people are working in government facilities, and somebody has to take that leap to collaborate with the private sector also, and with the government sector also. I think government to government, it is more of paperwork and permissions. And with private, once that roadblock is, or that block is gone, that you have to collaborate, then you can have lots and lots of samples. Of course, each site, maybe a larger hospital might give you large amount of samples, but there will be multiple smaller sites. For example, there are OPD clinics. If you're looking at COPD patients or asthma patients, if you just want, maybe for example, you're just looking at eosinophilia or blood samples. So each small site, there'll be multiple small sites, and they'll be able to give you large amount of samples. So I think a collaboration platform is what is needed, and how to go about it, I'm not really sure.
There is no lack of clinical material. Dr. Huriharam, your comment.
So I totally agree with what Dr. Jindal has said. We also need collaboration is the key, I think. And when we have to integrate the clinical research and the basic research, I also feel that as clinicians, especially I'm talking about myself, there is lack of understanding of the basic research as to how it can be applied or how it can solve my problems. So I would expect, a more collaboration and what you call a hands-on, or at a similar platform, if we can have integration of both these. And then only we could move ahead in that direction. Because right now clinicians are not really aware about how these omics and how these type of studies can answer their question, or develop new molecules for that matter, or new drugs. So I think it needs to start somewhere, because that will only lead to major discoveries. Because otherwise what we are doing is we are just copying whatever evidence, like we are copying the GOLD guidelines, we are copying the GINA guidelines, and just incorporating it into our practice. So we need to have Indian specific data. So maybe our population might present in a different way. So we are not sure about that. So that thing can only be answered by this research, I would feel.
Ungira is our inspiration. So Ungira, your comment regarding research activity. And Parthoda is also. Sorry, yes, please.
Work with basic scientists. So I have experience with National Institute of Malarial Research. I have done with proteomics. So what problem we faced is basically I don't have any problem from conducting study, giving samples. The problem arises when the transportations, the cost of transportation, it has to be the sample in minus 20, 40 degrees. So that is difficult to arrange money from our aspect. If anybody, basic scientist, wants to work with me, I can give the samples. There's n number of samples. Whatever the clinical data are there, it is theirs. And what happens, basic scientists don't understand what we want. That is, they want something and we want some clinical relevance. That doesn't match. So we have to think that clinicians will not, every time they will get the clinical response. It is our duty to support the basic scientists to get something else. I always agree to help them. Whatever they want to do, you can do. I can give you the sample. It is likewise. So
there is no need for permission.
No, permission is there. That is,
yes.
No, permission is there. We have to go for the ethics committee. That is not talking about. We have to take the permission ethics committee. Get samples. And nowadays it is even in the, you go collaboration for the Western population, then again it is the issues. When I published my data, ICMR did not allow me to GLI equations. I published my data, they did not give me the permissions. Again I asked for second times in our institute. They again said, no, you have to take permission from the government of India. Just simple spirometry data or lung function data, that is not permitted. So that is the way problems happens. So basic scientists, we have to send the samples, and sending samples nowadays is very difficult in the, outside India. So if basic scientist wants, they can do, they can try their research, that is not a problem. But we should also try to forget that we should not get the answer. If you say the clinical relevance, what is this, that may not be the answer, but it is our duty to support them. That I believe.
Ungira, your comment?
So most of the experience of doing research was during the time I did PhD from IGIB. So when I went there for 6 months, the initial first 6 months has to be on site. মানে ওদের ক্যাম্পাসের মধ্যে থাকতে হবে, ওদের হস্টেলে থাকতে হবে, ওখান থেকে কাজ করতে হবে. So when I reached there and I saw all my co-PhDs, they were all my children's age. So I used to tell them, and they automatically called me madam. I said, you can call me auntie, you can call me grandmother, whatever you want. And the way I reacted in their class was unique because I was at par with the teacher who was teaching, whereas the people whom I was surrounded with were the students. And I always used to think, people must be thinking, এই তো ডাক্তারি করতে পারলো না, কিছু পারলো না, সেজন্য এসব করতে এসছে. But that was not really the goal. ওদের ওখানে গেলে you will realize, if you go hand in hand with a non-clinical বা basic scientist, you will realize that the basic scientist and our clinical physicians who do research, there is a basic difference. Like Dr. Dey just said, that it's very difficult to expand your thinking and make the basic scientists understand what you want to do, and we cannot understand what they are saying. That is a problem. And the way out is to do some reading. It's not that they are going to come and teach me something and I will learn and then I'll go and deliver. You have to have the interest. You have to solve your own problems. If you can solve your own problems, you are through a little bit for your research purpose. And the research question that you are asking is the most important thing that has to be framed correctly. We can do the methodology, it's not an issue, we can always do that. But when it comes to writing up the basic scientists' work and telling them that this is I want, we will realize that we have a lot of problems there. So recurrent discussions, onek alochona, onek kichu kore thore, then you have to come to a point. Jeta sample, I have faced this, so I'm saying this. Samples get lost in institutions. I had 33 samples to 45 samples, and I ended up completing my thesis with 30 samples. The rest were lost. And the advantage of sputum being a research tool is that you don't need a -80 to carry. You can just put it on a crater and send it off. ওটা আমরা তো জানি, আমরা আগে যখন ব্যাঙ্গালোরে পাঠাতাম for culture of tuberculosis, we used to send it just through the plane courier. No temperature control was required. এটাই আর কি, মানে there are certain things that we have to understand. Blood also doesn't, কারণ ব্লাডে আবার অনেক সময় প্রবলেম হচ্ছে যে ওই ডিএনএ আরএনএ এর গল্পটা. So you have So these are certain issues which need to be discussed, and the answers will come around that point. And it requires a lot of dedication, it requires a lot of ambition, and you have to complete the entire thing, get your answer and publish it. Thank you.
Indranil?
So keeping in line what Ongira was telling, so I'm working in a government hospital and also as doing my clinical practice. A lot of clinical and research questions are there which arises in us. But I have worked with NIBGM in 2 projects, and I have seen that convincing my patients and giving a lot of patients is an easy task for us when we are working in a government hospital. For example, they wanted tuberculosis with HIV. Just doing this, giving the sputum, the blood, it was not a difficult task for us. But when we think of writing it down, tabulating it, doing the statistical methods and all those, this is something difficult and we find it cumbersome. And that's the reason practically we don't go into much of research. But yes, if some basic clinical scientists work with us and just implement what our clinical questions are and how they can be implemented, it will be easier for us. So we have to work together. That's the thing. So I have worked in 2 projects with NIBGM, and I have seen that when we convince the patient to give the sputum, the blood, they used to draw 20 ml of blood and the patient used to round up. But when we tell them and convince them, they used to comply with us. So when we work together, it is easier for us, and doing the project becomes very easier and we get very good result out of it.
Parthada, your final comment and conclusion.
I am really novice. What I feel that all clinicians should keep a research attitude, because I feel when I look at a problem, as a researcher, I become more analytical, I learn more. And I feel that every clinician, if they are bent to do a little bit of research doesn't mean that you have to do big things. You can do small things. Small things also matter a lot in long term, may matter a lot in long term. It makes you feel happy. It fulfills your a different kind of desire. And moreover, you turn a better physician. That is the first motivation. I wish that it should be dissipated. And second, that research means that you have to be at equal length with your colleagues and have a good rapport. Of course, in this country, there's a lot of problem for private sector to get fund. Without fund, you cannot do. We cannot do anything. Not of late, for last 10 years or so, I have taken a policy to light my own house, generating our own electricity. So it's a very difficult job, very, very difficult job. But I cannot change the system. What I can do, I can try my way. Whatever small I can achieve is okay. I do it for my personal happiness. And I feel that everyone should try it, and that makes a physician more complete. This is my answer.
Thank you. We conclude this panel discussion.
Thank you.
So this is a final hour for the pleasant job for giving vote of thanks, and I am the only person on behalf of my institute to do it. My good friend Dharmesh is not here. I was asking him when will be reaching his home. So he said that he will start from here at 10 o'clock. He will stay at Mumbai airport the night, and early morning he will catch a flight to Vadodara. So this is a kind of commitment and love been shown by our colleagues to come and join. Aditya has come. I call you by name. He's much junior to me. He's my teacher's son. He has come from Chandigarh. It's a pretty long journey. And I understand for a clinician to sacrifice and manage, not sacrifice, manage the job and come for a conference is how difficult. Same implies to Dr. Hariharan and all others. I'm very impressed that our revered scientist colleagues have been with us from the morning till now. Dr. Sajal there has immediately agreed to my invitation, and I'm really grateful to you. I am really grateful to all my colleagues in Kolkata, and everyone has come so to make this small conference successful. I would have been very happy if I could see more and more postgraduate students. Somehow I'm in disconnect with them because I'm not in the mainstream teaching. Maybe that would have helped. I'm grateful to my colleagues in my institute because they take the institute as their own, and frankly, they always treat the institute as their... I tell them that this is our family. So you have to thrive on it, you have to flourish on it, and whatever you do, you'll be actually reaping the harvest. So this I openly declare, I tell everyone, those who work with me, and I feel that they have taken it, and I always feel the warmth once they work and they look at small important issues which I miss, and most of the time they pick it up and come to me, sir, this should be done, this should have been done, this is missed, this would do better, and then take care of small items and save them from being wasted. A simple switching off a light or a AC in time, it means a lot to us because we have to pay the bill at the end of the day. My junior researchers, my research fellows, they're also very keen to do work. We have a lot of logistic difficulties, but despite that, I know they do their best. And they are being always guided by our researcher colleagues and the faculties like Dr. Banerjee, Dr. Saha, and Dr. Mukherjee, and many others. So I have a very lucky status to my mind. I'm very happy that way. All the other, I have not bothered about this small symposium because almost everything has been done by my people, my family members. So I thank them profusely. I thank the sponsors. Without them, it is not possible. And finally, I thank all others who have helped us, including this hotel authority and all, because ambience, the place, space, food, everything is somebody's effort. Without effort, nothing goes. So at the end, I thank everybody and thank those, if I've forgotten to mention particularly the names to make this successful. I hope that we'll be able to continue such efforts in future, and I will be having much more better academic sessions. So with this, I thank you once again. Please come if we call you again.